Welcome, everyone. I want to first thank every single person that came today to the scientific workshop for Limb-Girdle Muscular Dystrophy. I know so many of you had to fly long distances and traveling can be difficult. We're so grateful for you to be here. Thank you, all of our academic experts, the FDA, industry, and all the many people in the LGMD space that are doing drug development as well. We are honored you're here. I am Kat Bryant Knudsen, and I'm the founder and the CEO of an organization called the Speak Foundation. And we are a patient-led and patient-run organization for all subtypes of limb-girdle muscular dystrophy. And when you see events that we run, such as the scientific workshop, the International LGMD Conference, the LGMD News Magazine, all of those events that you see are run by patients and the caregivers of those patients, which is really unique and unusual. And so I tell everyone that because I think I'm so proud that we can do this in our community. I want to also let you know that I live with LGMD 2I-R9, and I've had to undergo some really great difficulties as a woman living with a disability. And so I know firsthand what it's like to live with limb-girdle. I've been running this organization for 15 years, and I talk to families, I talk to kids, I talk to teens, I talk to parents, and I am 100% committed to seeing treatments get to the finish line. Our community previously conducted an externally led PFDD meeting. PFDD, for those who don't know, stands for Patient Focused Drug Development. Today's scientific workshop is a natural step as academic disease experts, individual patients, patients advocacy groups, regulators, and drug developers with an active IND will all be discussing many important scientific aspects of limb-girdle muscular dystrophy. Today, you're going to also hear compelling stories from our patients and their caregivers. And I really want you to take heart and listen to what they're sharing, because they're going to be telling you their personal stories, what they want to see in clinical trials and in drug development. And they've taken their time, and it's taking a lot for them to be here. And I'm very proud of them for being here today to share. And I urge you to also contemplate the decades of scientific knowledge that we have for limb-girdle. This disease is well researched, and the experts that you're going to hear from today are going to share some amazing information. And at this time, I want to introduce Keith Flanagan, and he'll be talking about the nuts and the bolts in the housekeeping for today's workshop. Keith?
Yeah, our agenda for today is we're going to ground the discussion in the pathophysiology of the disease, have a little break, talk about what's important to patients clinically, and then we're going to dive into the details of accelerated approval, surrogate endpoints, and innovative clinical trial design. That's the agenda for today. We're trying to pack a lot into a small amount of time, so please try and stay within your allotted time. Bathrooms are around down the hall. If you exit to the left, there's a water dispenser right there. If you need to step out, please exit quietly to avoid disrupting the live stream. For online participants, use the chat to converse with others and ask any IT help desk questions. The chat will be captured in the record, but there's not enough time for live Q&A. If you have questions concerning the venue or other such topics, Marty is out. Ask Marty, who's at the table outside. And finally, with any follow-up inquiries, please email Kat, who just spoke. In the tiniest nutshell ever, here's what we're talking about today. How well do we understand the disease? What's clinically meaningful to patients? How can accelerated approval using surrogate endpoints enable a first wave of approved therapies and subsequent therapeutic advances? How specifically should trial designs be tailored to limb-girdle muscular dystrophy clinical contexts, and what's a pragmatic approach? I echo Kat's gratitude for patients and caregivers who traveled, and I need to uh, reinforce some standard FDA caveats. We're super grateful for FDA folks attending, but this is not the right forum for individual companies to get feedback from FDA concerning their individual programs. And FDA, can't make formal binding regulatory commitments in discussions today. So with that, we have a senior statesperson in the field as our keynote speaker today, Dr. Mendell.
Thank you for the opportunity to be here today. My talk was, I was assigned in some ways by Kat and others, and mainly about the history and importance of nosology and classification. And as I looked at the program, it fits in beautifully for those who follow with individual talks about different forms of limb-girdle muscular dystrophy. And these are the goals for my talk today. One, to define the basis for the LGMD nomenclature, address how the nomenclature and classification contribute to the advances in molecular biology or molecular therapy, and address if the nomenclature enhances a path to clinical diagnosis and treatment. So with that, I think what I'd like to start with is the definition of limb-girdle muscular dystrophy. And this was defined, and it's the current one that we use for limb-girdle muscular dystrophy. It was defined in the 229th European Neuromuscular Center International Workshop, of which there were several that I'll discuss. But limb-girdle dystrophy, by definition, is a genetically inherited disease. Most of you know this. It affects predominantly proximal muscles. It is inherited, and in order to get a place on the classification, it has to be described in two previously unrelated families, has to have a CK elevation, degenerative changes by muscle biopsy, and muscle imaging, and that will define the disease. Now, what's interesting about this 2017 workshop and published in 2018 by Volker Straub was that it was largely based on something that happened 60 years before, and that is a classic article by Walton and Nattrass in 1954 that appeared in Brain. And the definition is largely the same as what was defined 60 years before. It had the one unique thing about Walton and Nattrass definition is that they made it distinct. They described that there were 4 forms of muscular dystrophy, not surprising: DMD, FSHD, and myotonic disorders. It had to have proximal muscle weakness with a waddling gait, and we often use that, and I'll show you an example. The typical, what we call the Trendelenburg gait, had to have frequent falls, trouble stair climbing, and had to be distinct in the sense that there was no pseudo-hypertrophy, or it was very uncommon. It was genetically based, largely, as Volker Straub said in 2018, affecting 2 unrelated families. And one of the things I admired about their observation was that it affected proximal muscles, but they allowed for the possibility that it could extend to affecting distal muscles. And this is the typical Trendelenburg gait. It's often, from a historical point of view, it's also interesting that the Trendelenburg gait was described almost 60 years, 50 years before for Walton and Nattrass. And here's basically what they showed, or what they described. And many of you are familiar with this. This is the typical waddling gait, one hip up, one hip down, and it shows better when there are two canes involved. As you'll see here, that you can really see the waddling performance. And this is what characterizes limb-girdle muscular dystrophy. When many of you see patients with this, you appreciate the waddling gait. The other thing about this is the observation that that proximal weakness can extend. It's not entirely defined by proximal weakness. It can extend to distal muscles. And this is a very good example of that. As you see, he basically has muscle atrophy below his knees and has bilateral foot drop. So characteristically, what you see here then is proximal and now superimposed distal weakness. And many of you are familiar with this. This is really characteristic. We don't know what this patient has, but he looks like he has a dysferlin-related myopathy. And this is what Walton and Nattrass really described, and it's observational. It wasn't dependent on any genetic identification. They described FSHD, as you see here, with shoulder girdle weakness and facial weakness, the myotonic disorders, the typical facial appearance, and tongue myotonia, and a typical Duchenne patients, contrasting with what's over on the right-hand side with the limb-girdle patient. Now, what changed dramatically from what Walton and Nattrass described was the discovery of DNA, and this is no surprise to any of you. DNA had an interesting history itself. It was the first evidence of a transforming factor was done by Frederick Griffith in 1928 when he described that bacteria would change their phenotype from harmless to severe lethal bacteria. And then the very famous contributions by Rosalind Franklin, the photo of DNA that then extended to Watson and Crick's very well-described double helix in 1953. Now, consider that from 1953 to what we consider now as classification, what happened was that once the DNA was described, we had, one, the discovery of dystrophin by Lou Kunkel. Then we had characteristic changes that were well described with the dystrophin glycoprotein complex by Kevin Campbell, and then individual diseases that were added, but with basically no organization. And then in 1992, Kate Bushby decided that the field needed organization, and she proposed that there be ENMC workshops that did exactly that. And from 1992 to 2018, there were 5 workshops. The first one was in Soest, the Netherlands, and then the others were in Naarden, as you probably well know. But let's look at the first one. The first one was what kicked off the program, and it involved 37 participants from really international contributions. It established a further distinction from what Walton and Nattrass had described was some, I would say, some refinements, but it wasn't really terribly different from what Walton and Nattrass described or what Volker Strobe put out in 2018. But they'd started discussing the molecular genetic work that was in progress to coordinate future work required to identify relevant genes. So it was a good kickoff, but it was really the 1995 ENMC that established the classification for LGMD. And you'll find this interesting because the classification has gone through several evolutions. In 1995, it was proposed that the autosomal dominant LGMDs be designated type 1, and the autosomal recessive be designated type 2. And they further defined LGMDs by adding on an alphabet designation that was based on sequential chromosomal linkage, but not gene identification, and that's an important distinction. And it established a framework for classification that had been defined by Luke Kunkel and Kevin Campbell on their dystrophin glycoprotein complex. So in 1995, this was the shape of classification. There was one LGMD that had been linked at 5q, and there were 4 LGMDs that had been linked they were all sarcoglycans, and not surprising because Kevin Campbell had described the sarcoglycans as important components of LGMDs. And the first one was adenylin or adhalin that was later changed to alpha-sarcoglycan. So the field is evolving, and we used this classification to show you how it contributes to molecular therapy. That's when we did our first gene therapy following that. That was in 1999. And we did this in LGMD 2D, not surprising. That was one that had been molecular characterized. We knew the size of the gene. We knew it fit into AAV. And we asked the FDA, since there was little known about gene therapy except mostly fear and anxiety, and that was, could we inject a small muscle in the foot? If we injected this muscle, then it would give us a safety domain, because if there was a problem, we could remove the entire foot muscle. And I injected there 2 ways, one vertical and one horizontal. So we had a very high incidence or prevalence of the gene in the middle of the muscle. We removed the whole muscle. It was a double-blind study. One side received the gene, one side received placebo. And most of you know whose foot muscle this is, and I won't say any more than that as a reflection of HIPAA rules. But here's what we found. It was remarkable that we found any gene expression. We were at least 2 or 3 logs behind what we know today as the proper dose for gene therapy. But we did see a small segment of the muscle that was expressing the gene, and on the opposite side, we didn't see any. So here's the first goal. We did contribute by classification to molecular therapy. And then we went on to the 1999 ENMC. This was mostly to further characterize genotype and phenotype as they were being characterized further and further, because we knew more and more about the molecular basis for identification. And then in 2002, what you'll see here is that we had, by then in 2002, we had 5 genes or 5 diseases that were linked to chromosomes. Not all of them were identified with genes, and we had 10 disorders that were outside the dystrophin glycoprotein complex now. FKRP, Titin, Calpain, and so this was expanding and done as Kate Bushby hoped. It was doing what it was described to do. And then we took advantage of that again with our first LGMD, with our second LGMD gene therapy, and now we had further refinement. We could use a codon-optimized sarcoglycan. We could add an intron to enhance gene expression. We used an MCK promoter, and we knew enough about the safety of so we could fit this into AAV, and that was AAV1. And again, it was in LGMD 2D. And again, adding to the science of gene therapy, we did 6 patients, and we had robust gene expression in 5, but the 6th patient had no gene expression. And we were perplexed by this, thinking that maybe it was the technician who made a mistake, used the wrong side, and so forth. But when we looked at this further, again, what we found was pre-existing immunity, and we were surprised about this. This was really the first observation of what later became a critical factor in gene therapy. And the first 5 patients had pre-existing antibody that was less than 1 to 50 dilution. And by the end of the first week, it had elevated to 1 to 3,200. The 6th patient started out with 1 to 1,600. And by the first week, had increased the antibody now to over 100,000. This was 30 times elevated. And it was really because there was pre-existing antibody and an amnestic effect. And those antibodies then found their receptor on the AAV and blocked gene expression. And now that has become an important contribution, because now we started out using 1 to 50, and now we're basically in the same range with pre-existing antibody less than 1 to 800. So let's move on and go back now all the way where we started in 2017 and see what we have. Because here now we have significant changes in what was defined as a sensible way to classify limb-girdle muscular dystrophy. We have now over 30 LGMDs. 4 are dominant, and the remainder, actually that's 5, should be 5 there, I think. I excluded 1. 5 LGMD that have been linked as dominant, and then the remainder are recessive. But what's significant about this is that instead of LGMD 1 and 2, it was the wisdom of the conference to change that to dominant and recessive rather than 1 and 2. Can't hear me? Thank you. To change it to 1 and 2, from the dominant and recessive from 1 and 2. So that was an important contribution or change. And then instead of using the alphabet, it was assigned numbers. So now we had LGMD D1, D2, LGMD R1 and R2, and so forth. And that way, there couldn't be any loss over time in classification of alphabet contributions. So I just have those 4 highlighted because those were the first, and now we have changed completely. And further goals are achieved by this in that it contributes to whole genome sequencing, whole exome sequencing, and certainly in gene therapy trials. Now, there are potential disadvantages. Certainly, it's not intuitive to call a disease LGMD R3 without any gene identification. If you're going to use this classification, and I see the next talks will maybe address that, you practically have to have the cheat notes in your pocket to know what is R21 or R17, or with no gene identification. So it makes it more challenging, but there is wisdom, but there have been, I think, some resistance to this classification, but that's really what it is today, and I wanted to review this. And so as the next talks, as the foundation for the following talks, it'll be clear where we started. And the final slide here, I see I have 1 minute left, so that's not bad. So I'm usually over, so I'm happy about that. So what you'll hear later on, and Louise Rodino-Claypack will discuss our gene therapy trial. But I also want to point out that there, here in the title of the paper, there's a little combination, LGMD2E, which is uses the old classification, and R4, which refers to beta-sarcoglycan as part of the classification. So you'll hear from Louise about the result of our LGMD trial, and I'll stop there and answer any questions. So thank you very much.
Thank you so much, Dr. Mendell, for your presentation. We would now like to introduce Dr. Kang, Dr. Matthews, and Dr. Wickland to share an LGMD subtype overview. Dr. Peter Kang is Professor and Vice Chair of Research for the Department of Neurology at the University of Minnesota Medical School. Dr. Catherine Matthews is a Professor of Pediatrics General Neurology at the University of Iowa Healthcare and Carver College of Medicine. And Dr. Matthew Wickland is a Professor of Neurology at the University of Texas, San Antonio. So we'd like to welcome you up at this point.
Thank you very much for the invitation. It's a pleasure to be here today. And I guess I'll just get started. We'll discuss the sarcoglycanopathies for a little bit, and then I'll hand off to my colleagues. I'll go through a little bit of history and genetics, and then clinical features, and then clinical course and outcomes to set the stage for the discussions that'll follow for the rest of the day. Let's start with some history and genetics. So Dr. Mendell talked about this a little bit, but just to frame it a little bit, the concept of limb-girdle muscular dystrophy as being a separate disease category really started in the 1950s. And he went through some of the symptoms that are characteristic. It's important to remember that the onset can be in childhood or adulthood. There is a very broad range of ages when it could start. And one of the key genetic factors is that it's inherited in an autosomal fashion, both recessive and dominant. And then there are muscle biopsy features that are not unique to limb-girdle muscular dystrophy, but characteristic of it, including fiber size variability, degenerating and regenerating fibers, and fibrofatty infiltration. So as discussed, the individual gene discoveries for limb-girdle muscular dystrophy began in the mid-1990s. So when Dr. Kunkel discovered dystrophin in the 1980s, that set the stage for a whole wave of gene discoveries that followed in the decades to come. And there are still new genes being described even to this day. So the sarcoglycan genes were among the early ones that were described, and these were in the 1990s. And alpha-sarcoglycan was also known as adhalin, and I still know a few people use that term adhalin, and but most people have started to adopt alpha-sarcoglycan. And then beta, gamma, and delta sarcoglycan were all described around the same time in the mid-1990s. It was quite a wave of discovery, and we started to understand much better where these diseases were coming from once we understood the genetics. There are a couple of other sarcoglycans. There are actually 6 of them. One other has been linked to a genetic disease, that's epsilon sarcoglycan. It's associated with a myoclonus dystonia syndrome, and so it's not typically discussed in the setting of muscle disease, but I thought I would mention it in case it comes up. And then there is a 6th gene, zeta sarcoglycan, which has not, to my knowledge, been linked to any kind of disease at all to this point. So as we've heard, the nomenclature has been a little bit tricky. Once the genes began to be discovered, a classification came up because people realized there are many of these subtypes. So what was originally adhalin became known as alpha-sarcoglycan, and then beta, gamma, and delta. And then they, with the previous classification, they were known as limb-girdle 2D, E, C, and F. And with the more recent reclassification, now they're known as limb-girdle R3, 4, 5, and 6. And so as you can imagine, just as an aside, this does get a little bit confusing because of the swapping of letters and numbers. And so to avoid confusion, some people are labeling these subtypes with both classification systems, and we try to do that just to make sure that we're being as inclusive as possible. But we realize that there are some difficulties with this classification system. So this is a diagram that puts the sarcoglycans in the context of muscle disease genes in general. And you can see how complex it is. And I remember back in medical school in the 1990s, I mostly learned about actin and myosin when we learned about skeletal muscle. And you can see how much has been learned since then. The thin double line going through the middle horizontally is the sarcolemma, the muscle membrane, and then above it is the extracellular matrix, and below it on the inside is the inside of the muscle fiber. And if you look on the upper left, you can see that the sarcoglycans form this complex, one of each, alpha, beta, gamma, and delta, form a complex that goes through this sarcolemmal membrane. And as you can also see, the sarcoglycan complex interacts with a number of other proteins, both inside and outside of the muscle fiber. So just looking at this diagram, you can imagine that any disruption to the sarcoglycan complex is likely to have some consequences for muscle function. And you can see some of the other well-known proteins elsewhere in the diagram, including dystrophin and others that are scattered throughout. Let's move on to some clinical features. Some of the key clinical features of the sarcoglycanopathies, childhood onset is more common than adult onset, and there are different limb-girdle subtypes that tend to be more childhood onset versus adult onset, but the sarcoglycans tend to have onset more during childhood. And as you can see from these statistics, which are compiled from multiple sources, which I have a reference list at the end, the mean tends to be during childhood, typically between 5 and 10 years, but the range is really much broader, and so it can be anywhere from 1 year to 45 years, depending on the subtype. And there are likely other outliers. One thing that I've seen is that there should be more literature on the sarcoglycans in general and more research on these subtypes. The CK levels can be quite high, and they can range from a few times the upper limit of normal all the way up into the hundreds, into the range that you would see with Duchenne muscular dystrophy. So depending on the individual, the CK level alone would not necessarily tell you which subtype an individual has. This is from one of the articles in the 1990s, and this is showing what alpha-sarcoglycan looks like when you stain for it on a muscle biopsy. And one of the interesting points about this is that the degree to which alpha-sarcoglycan staining is diminished can vary. So there are 2 patients shown in these biopsy slides, and the panel B shows a Western blot, and as you can see, one of the patients has only a little bit of a problem with alpha-sarcoglycan. It's diminished but not absent, whereas the other individual seems to have complete absence of it. And that indicates that there is some clinical variability that occurs. And this is from one of the descriptions of beta-sarcoglycan deficiency. And you can see this is a set of biopsy slides from a single patient. And dystrophin staining is a little bit impacted, but you can still see that there's dystrophin staining. One of the interesting points about this that was recognized back in the 1990s is that if you have a deficiency primarily in one of the sarcoglycans, it can affect the others. So this patient has a variant in beta-sarcoglycan, but not only does the individual have a deficiency in beta-sarcoglycan, but alpha and gamma-sarcoglycan are affected also, and that's been shown repeatedly over the years since then. This is a paper that we published in the mid-2000s with a group of patients with gamma-sarcoglycan deficiency, and as you can see in the 3rd row from the top, gamma-sarcoglycan is absent in the 3 patients. But you can see that alpha-sarcoglycan is also affected to varying degrees. And then the top row shows the hematoxylin eosin staining, which has basic structural information about the muscle. So then we can talk a little bit about the clinical course and outcomes. So these are pretty serious diseases, and all of the limb girdles are serious diseases, but the sarcoglycans, we should document that there is loss of ambulation in a high number of individuals. And one thing that I've gathered from looking at the patients that I see, as well as the literature that I've reviewed, is that we really need better information about the clinical course of the sarcoglycans. There are articles out there. There could be more prospective studies, which some of us are working on right now. So cardiac and pulmonary complications do occur pretty frequently in all of the sarcoglycans. And again, these ranges are pretty broad because these are retrospective studies, and depending on the age you look at and the number of patients, you can get numbers that can be pretty variable. But I think what we can gather from this is that both pulmonary and cardiac complications do occur with some regularity. And so when we diagnose one of these diseases, we really do need to keep a close eye on cardiac and pulmonary complications being possibilities, and and also counsel the patients about these risks. And the cardiac complications can manifest in several ways, and that includes the dilated cardiomyopathy, hypertrophic cardiomyopathy, and rhythm disturbances, as is seen in other subtypes of limb-girdle muscular dystrophy. So I think I'll end here. These are the references that I've drawn from, and also from my clinical experience. The main message that I want to convey is that we've known about the sarcoglycans for several decades now. They're a pretty serious set of diseases. We do need better therapies for them. There are no FDA-approved disease-modifying therapies, so it's really great to see so many people that are interested in pursuing further knowledge and new therapies for these diseases. And I'll be happy to take any questions. Thank you very much. Oh, okay. In that case, yes, I'll pass it on to Dr. Matthews.
Hello, I'm Kathy Matthews. I'm from the University of Iowa. And I was asked, these are my disclosures. You can read them. I was asked to talk with an emphasis on LGMD2I and with an emphasis on sort of pathophysiology. So what I'm going to start with is genetics and pathophysiology, and then I'll move on to clinical phenotype. So LGMD2I-R9, I'm also going to use the double classification system, is in the group of disorders known as a dystroglycanopathy. So the clinical classification is LGMD. The pathophysiologic classification is a dystroglycanopathy, and I'm going to bore you with details of what that means in a second. LGMD2I-R9 in many series accounts for 8 to 15% of LGMD patients. I will put in a little plug. MD StarNet is a CDC-sponsored epidemiologic study, and we should have some numbers from that about each of the LGMD types coming in the next year or two. So you've seen other versions of this diagram. This is the dystrophin glycoprotein complex. The muscle cell membrane is the black thing going through the middle there. The sarcoglycans that Peter just talked about are in yellow. And dystrophin is inside the cell. It connects through beta and alpha dystroglycan to the extracellular matrix, represented in this diagram by laminin alpha 2, or myrisin. Alpha-dystroglycan is heavily glycosylated, so it has lots and lots of sugar groups on it. That glycosylation is critical to the linkage with the extracellular matrix, which provides some stability to the muscle cell membrane. The stability that is achieved through the complete linkage from dystrophin connecting to the contractile mechanism through alpha and beta dystroglycan to the extracellular matrix, which is fibrous tissue and providing stability to muscle cell membrane. If you do not have appropriately glycosylated alpha-dystroglycan, so if the sugar groups are not on there appropriately, you get the disruption of this link between alpha-dystroglycan and the extracellular matrix, loss of membrane stability, muscular dystrophy, and then in the case of dystroglycanopathies, with or without abnormalities of eye or brain function. So the sugar groups, and I tried this out, so the sugar groups, the connection, these are long, so if my body is alpha-dystroglycan, the sugar groups are long chains, and if I had like 6 or 8 or 20 or 40 arms, I would have them out like this, and my hands are the little holder-honors to the extracellular matrix. If you have an abnormality in the construction of that sugar group, you have little shrunken dinosaur arms, and you don't have your little hand that can hold on to the extracellular matrix. So you screw up the construction of that long sugar group on alpha-dystroglycan, and you can't connect to the extracellular matrix. These long sugar groups create a flexible way to attach to the extracellular matrix, creating sort of a, your muscles move in and out, in and out. They provide a flexible attachment to that fibrous matrix. This is one diagram. Matriglycan is the name of that full-length thing with the little hands. So the full-length sugar, Kevin Campbell would not approve of this. Kevin does not describe this by doing arm things, but it's the way I think about it. So matriglycan is the name of that whole full-length sugar, and a diagram is shown here. So the glycosylation of alpha-dystroglycan takes place through the secretory pathway, and this is a really old image, but I like it because it shows the alpha-dystroglycan in red working its way through the cell to its final home at the sarcolemma, and having different enzymes act on it to create matriglycan. And we now know that there are more than 20 genes, enzymes that are required to fully glycosylate alpha-dystroglycan. And FKRP, the topic for today, is one of these. So the function of FKRP was unknown for a long time. We knew the gene, but we didn't know what it did. We knew it was important for glycosylation. This was clarified in 2016 by work in Dr. Toda's lab in Japan. So there are 3 enzymes that work together to add 2 ribitol molecules to the growing matriglycan chain. So I don't have a, so if you, first of all, ISDP, which is in pink on your right of the screen, in the diagram of the screen, prepares CDP ribitol, and then fucotin adds one ribitol to the growing chain, and FKRP adds another, so then you have this tandem ribitol, and then the chain is further added to full maturity if you have all the enzymes there. So you have to have each of the enzymes in this pathway working properly for full-length matriglycan. And this is our one of our more current diagrams of the process, where you start with alpha-dystroglycan at the very bottom right of the screen, have each of the enzymes act in the correct order, ISPD, fucotin, and FKRP, add the ribitols, and then the chain is extended. So, we now have a pretty good idea of the function of FKRP. The FKRP protein structure is I'm not going to address this, but the protein structure is in the process of being clarified and opening up some other new potential avenues of therapy. So it's important to note that everybody has some FKRP activity. If you have no FKRP activity, you don't become a human. It's required for embryonic life in Reichert's membrane, so everybody has some FKRP activity. So if we move now to what this looks like in a human, so the muscle biopsy will show, this is not a bad muscle biopsy, but it shows progressive loss of muscle fibers with fibrosis and fatty replacement. And the control is C, G, K, and O, and the patient is next to that. So you can see that dystrophin is normal in the patient. The glycosylated alpha-dystroglycan, so the alpha-dystroglycan that is stained just for the sugar groups, is decreased in a patchy fashion. Everybody has some FKRP activity, so it's not absent. Alpha-dystroglycan itself, the core protein, is present, and beta-dystroglycan is also present and normal. So the protein is present, but the sugar groups are abnormal. This is also shown with serial MRIs. So the white is bad and black is good. And in the graph over to the right, it shows the change in fat accumulation in different muscle groups over 6 years. This is work by Volker-Straub's group. So if we move now to the clinical spectrum of FKRP-related muscular dystrophies, if we think about the dystroglycans in general, dystroglycanopathies in general, there's a wide range of clinical severity from congenital muscular dystrophy through limb-girdle muscular dystrophy. And actually, and where you draw that line between the two is always a little bit arbitrary. FKRP mutations typically cause limb-girdle muscular dystrophy, but very severe mutations can cause a congenital form. The clinical severity seems to be related to the amount of functional sugar on the dystroglycan. Clinical presentation. So when a patient walks into clinic, I can't tell whether it's a sarcoglycanopathy that Peter talked about, a dystrophinopathy, Duchenne or Becker muscular dystrophy, or a dystroglycanopathy. Jerry might be able to, I cannot. They all have proximal muscle weakness. They often have calf hypertrophy. They have screaming high CKs typically. So they look very similar clinically. And you really need to do the genetic testing or muscle biopsy to know for sure. They all progress to involve muscle weakness, have cardiomyopathy. The LGMD2I patients have normal intelligence, although the very severe congenital patients can have brain involvement. We looked at the age at diagnosis by the initial symptom that led to the diagnosis, and the vast majority presented with chronic motor dysfunction, progressive muscle weakness. Adults tended to present with muscle weakness in the second decade. Children were more likely to be identified by intermittent symptoms, so symptoms of intermittent rhabdomyolysis, viral-associated acute rhabdomyolysis, intermittent muscle pain with activity, and then some people are identified by incidental elevated transaminases. We can't talk about the clinical picture of LGMD2I without talking about the genotypes. So there are a number of FKRP founder mutations that are known across the world, and this is from 2019, but there have been additional ones identified since then. These founder mutations arose in specific populations and are typically associated with a clinical phenotype. We're going to be talking today for the next several slides about CA26C-A, which is a European founder mutation. So the carrier frequency of A26C-A is 1 to 200, 1 to 600, depending on where you are. In Iowa, it's 1 to 333. We are a fairly Scandinavian population in Iowa. It is more common in the Scandinavian countries, and it's thought to have arisen and have been spread by the Vikings. We sometimes refer to it as the Viking mutation. The map to the right shows the Viking expansion through the 8th through 11th centuries. And so the map sort of reflects where the CA26 mutation is most highly represented in the general population. In the US, almost 100% of 2I R9 patients have at least one copy of this C826C-A. If you have 2 copies of 826C-A, you're homozygous for it. If you have 1 copy of that and 1 copy of some other mutation, you're heterozygous or have other genotypes. So the genotype affects phenotype in this disease. So this is aged onset of weakness, and you can see that those who are homozygous for the Viking mutation have later onset of weakness than all other genotypes. If we look at motor function, and this is work from our natural history study, you can see that, first of all, I need a pointer here, but if you look at the pediatric group, okay, first of all, in these slides, the homozygous common mutation are in red. All other phenotypes are in blue. If you look at the far left, this is 4-stair climb, how long it takes people to climb 4 stairs. You see that in the children, at the little sort of compressed thing on the far left, they are often stable or even improving if you are homozygous for the common Viking genotype. Others have declining function. Once you go over to the adult, the rate of decline is similar in our data, although the homozygotes are later when they achieve any given function. And that's more simply shown in the loss of ambulation slide on the right. The earliest age of loss of ambulation in our group was 9 years. The median for the entire cohort was 43. And you can see that those who are homozygous in dark black at the top are less likely to lose ambulation at any given age than all other genotypes in the light gray. The people with LGMD2I have respiratory muscle involvement too. This is overall, just showing not divided by genotype, although you can see again that the red dots tend to be progressing more slowly than the blue dots. And by our work and work of Volker Straub's group, the adult homozygous, the milder phenotype, tend to, I think there's a typo there, decline lose about 2% per year in seated forced vital capacity. So about across the European study and our study, about 2% per year loss of forced vital capacity. Cardiomyopathy is measured by echocardiogram. Again, you see the marked difference on the right between homozygotes and all other genotypes. And our work is similar to that that's been shown by others. Other genotypes develop cardiomyopathy at a much earlier age than the homozygous. The median age at first abnormal echo was 54 as opposed to 18 in our group. So in summary, I have no idea where we are on time, but I hope I've shown you that LGMD2I R9 is one of a group of dystroglycanopathies. They result from hypoglycosylation of alpha-dystroglycans. So unlike a lot of other limb-girdle muscular dystrophies, it's not a structural protein, it's an enzymatic loss. Results in loss of membrane stability and loss of muscle fibers, progressive weakness. FKRP which is the cause of LGMD2I/R9, is one of about 20 different enzymes required to fully glycosylate alpha-dystroglycan to form matriglycan. And decreased FKRP activity is the cause of the disease. The disease has significant impact on motor, respiratory, and cardiac function with a strong genotype effect. So thank you, and I'd like to acknowledge in particular Steve Moore, Kevin Campbell, and Dima Sade, my colleagues at Iowa. And most importantly are the families and patients who provided all this data. So this is a picture from one of our annual conferences. And I think we're doing questions at the end, right? Okay, so I will turn this over to Matt.
Good morning, everybody. It's a pleasure to chat with you today, and I'm going to cover two of the LGMDs. The two most prevalent LGMDs, R1 and R2. So to start off, I like this picture because this is 5 teenagers and young adults. And if you go from right to left, they are all distinct limb-girdle muscular dystrophies. On the right to the left, you have dysferlinopathy, calpainopathy, sarcoglycanopathy, FKRP-related, and Becker muscular dystrophy. And so the reason I like this is they all look a lot alike, but they have distinct differences. I also like this cartoon because this cartoon, for me, tells that if there is an abnormality anywhere within or near a muscle fiber, it can lead to a limb-girdle muscular dystrophy. And for the ones that we'll be talking about, you'll see on the left side, the yellow circles include LGMD2B, which is dysferlin, and then you can also see associated with the sarcomere down below, calpainopathy, LGMD2A or R1, associated with titin. So how common are these disorders? So we have relative prevalence data, and that came from the Jane Foundation and MDA genetic testing program from 2013 to 2015. And in the United States, calpainopathies tend to be the most prevalent, followed by dysferlinopathies, with the other subtypes following. That same data was also shown in the MyoSeq project in Europe, so Europeans are the same. Calpainopathies are the most prevalent, followed by dysferlinopathies. Our South American colleagues have found roughly the same. In Brazil, calpainopathies are more prevalent than dysferlinopathies, and then on the other side of the Andes, in Chile, there's actually slightly greater prevalence of dysferlinopathies than the calpainopathies. And then very nicely, Monko Lek's data, where he interrogated the Broad database, what he found is actually that anoctaminopathies genetically are the most common. But since anoctaminopathies have onset sort of in the 30s, 40s, and 50s, those that are clinically prevalent are still most commonly calpainopathies and dysferlinopathies. So let's talk about these two subtypes. So LGMD2A/R1, calpainopathies. It is the overall most common of the limb-girdle muscular dystrophies. About 1 in 100,000 people have a calpainopathy. That means there are maybe 3,000 or so in the United States. And there are pockets of enhanced genetic involvement in certain areas like the Indian Ocean, the Basque region of Spain, and then some Amish communities. Calpainopathies can be recessively inherited, but they can also be dominantly inherited. So the recessive have onset usually between 5 and 20 years of age, and the dominant usually is 20 years later than that, in the 30s and the 40s. And the nice thing about calpainopathies for diagnosis is they have a relatively distinct pattern of weakness. And so their knee extensors are much stronger than their knee flexors. Their hip flexors are much stronger than their hip extensors, and their hip abductors are stronger than their hip adductors. And you can actually see on that photo, if you look at the quadriceps on that gentleman, he has relatively preserved quadriceps. And if you look at his hamstrings, they're scalloped out and they're essentially gone. And we'll see that again when we look at cross-sectional images of MRI. Patients with calpainopathies lose independent ambulation 10 to 30 years after onset of symptoms. So the earlier the onset of disease, the sooner the ambulation will be lost. CK levels are usually 5 to 25-fold elevated, somewhere in that ballpark. And then the biopsy has this very beautiful lobulated fiber appearance in many of the biopsies. So this montage shows some of the other features. Scapular winging can be seen. There are contractures. There can be axial rigidity. And then the medial gastrocnemius muscle is significantly affected. There's no significant heart involvement in calpainopathies. And there is gradual respiratory insufficiency such that late in the course, perhaps 10% of patients will need some sort of respiratory assistance, nocturnal non-invasive ventilation. So as we talked about in this disparate involvement of different muscle groups in the thigh, this is an MRI of the thigh, a T1 weighted image. And you can see that on the top is the quadriceps. And then on the bottom, you can see where the white is, that's almost completely fatty and fibrous replaced hamstrings. And then somewhat medial to that, you can see the same thing with the adductor muscles. This then as a biomarker can be expanded through whole body MRI. And the nice thing about whole body MRI is that then we can do that across patient populations and develop heat maps so that we know which muscles are most affected in different disorders so that we can then have patterns of weakness. And then you can also follow that over time as a biomarker. The muscle biopsy in patients with calpainopathies, so going from A through F goes from early in disease till late in disease where you have a dystrophic pattern, you have variability in fiber size, fibrosis, fatty infiltration, and eventually that dystrophic muscle biopsy. So calpainopathies, calpain 3 is an enzyme. It's a calcium-regulated proteolytic enzyme, and it does have proteolytic activity, and that helps facilitate muscle remodeling. Calpain 3 cleaves sarcomeric and cytoskeletal muscle proteins, including titin and filamin C. And it also has structural roles which stabilize the triad complex. Calpain 3 interacts with and stabilizes RYR1. It promotes calcium calmodulin-dependent protein kinase 2 activity, and it's necessary for muscle adaptation in response to exercise. And even though we know a lot about calpain 3, we don't know the exact mechanism that it goes from calpain 3 dysfunction through to muscle death, but it's involved in proteolytic activity, involved in the secondary structure, stability, and ability to bind to titin. Mutations can affect any of those aspects of calpain 3. So the mechanism of action of calpain 3 is involved in assembly and remodeling of contractile proteins in the sarcomere, control of calcium efflux from the sarcoplasmic reticulum. It's involved in membrane repair, and it's important for muscle regeneration. Dysferlinopathies. So moving on to the second limb-girdle muscular dystrophy subtype that we'll be talking about, 2B or R2. This is maybe 8 to 10% of limb-girdle muscular dystrophies, perhaps 7 per million people in the US. It has a proximal-distal pattern, so the proximal limb-girdle pattern is there, and there's also distal involvement. And from about a decade ago, we looked at all of the cases in the literature, of which there were roughly 300, and half of them had a proximal predominant pattern at onset, and half of them had a distal involvement. Now we know that dysferlinopathies truly are a single entity, and it just is slightly greater proximal or distal involvement at the onset, but we consider it as one entity. Carriers rarely will be symptomatic, and there have been reports now of dominantly inherited dysferlin mutations. This cartoon just shows dysferlin in that sort of pink along the sarcolemma. Onset is later than in calpainopathies. It's generally in the teens so the later teens and then the 20s. We talked about that they have distal involvement early in the course, and the clinical pearl is that if somebody has difficulty standing on their toes within the first 2 years of onset of weakness, you have to think about dysferlinopathies and anoctaminopathies. There really isn't scapular winging, dysphagia, dysarthria, contractures, and from the COST studies, we know that there are P-wave abnormalities that are seen in about half of patients, but to our knowledge, there's not been clinical involvement with arrhythmias or cardiomyopathies. And in terms of respiratory involvement, there's about a 2% predicted reduction in respiratory capability over 3 years. But what that means is that maybe 30% of patients have an FVC that's below normal, but importantly, of this entire COST group of 188 patients, there was only one that required nocturnal non-invasive ventilation for a reason other than obstructive sleep apnea, so only for their respiratory insufficiency. This is a nice little finding that we tend to see in some of our patients that do have dysferlinopathies. There is atrophy of the proximal and distal portion of the quadriceps, leaving the central portion, which is a diamond on quadriceps sign. We talked about earlier in that both Miyoshi myopathy and LGMD2B or R2 are essentially the same disorder, and over a decade ago this was shown on MRI, this in the thigh and then also not shown in the calf. The involvement of the muscles in the limb-girdle pattern of onset and in the distal involvement, Miyoshi myopathy, are the same as you go through disease from the top, the most affected muscles, to the bottom, the least affected. The order in which muscles are affected is the same in the two disorders, so it really is one. CK level can be markedly elevated up to the 35,000 range. There is inflammation on many of the biopsies, but it's not the same extent of inflammation that you'll see in other cases of myositis, so it's just smaller, more scattered inflammation, and amyloid can be seen on the biopsies. In terms of how does dysferlinopathy lead to disease, the answer is mutations in DYSF or dysferlin lead to severe reduction or absence of the dysferlin protein in skeletal muscle, and you can see that on the left is the control patient with immunofluorescence staining of the muscle membrane, and on the right is a patient with dysferlinopathy. That lack of dysferlin in muscle leads to muscle cell death, which you can see on the right there with macrophages invading necrotic fibers, and then that eventually leads to fatty and fibrous replacement of muscle. The exact mechanism by which we go from lack of dysferlin to that end stage MRI, which you see just above there, is not precisely known, but dysferlin has been implicated in numerous muscle functions, including calcium regulation, membrane repair, vesicle trafficking, endocytosis, and lipid metabolism. And then 20 years ago, Kevin Campbell's lab nicely pointed out that there is a defect in membrane repair. And there also is involvement with of dysferlin with mitochondrial health, and then it also helps stabilize stress-induced calcium signaling in the T-tubule membrane. So management of these two subtypes and essentially all LGMDs currently, we do symptomatic management through multidisciplinary care. This is what a team usually looks like in that multidisciplinary care clinic. We are now adding this to that, which is a gene-based therapeutics team. And where we're at with dysferlinopathy is, one of the goals of many of the limb-girdle muscular dystrophies is to replace the protein. And to do that, you can simply, I'll use the word simply, do gene-based therapies. The problem with dysferlin is it's a little bit too big to fit in an AAV vector. So again, I'll use that word, use simply, bifurcate the gene, and then use those two transcripts in a viral vector. And then the native DNA machinery will put that back together. That's been shown up through the large primate model, and that's our goal going forward. It's been done in human, and so that's sort of where we're hoping to go. I'm going to leave you with that, and hopefully that was an overview of those two subtypes that you'll get a greater appreciation for, and I thank you for your time.
And we thank you for your authoritative insights. Thanks to your disciplined time management, we're a little ahead of schedule. So do all the three of you want to come back up and do the Q&A? It turns out we do have time for. And then Jessica can summon Dr. Johnson to the podium at 10 sharp.
So for those who don't know, MD-StarNet is a CDC agreement with now I believe 6 states or regions to perform surveillance. So it's not, a patient doesn't have to consent, but the state allows surveillance of medical records of people with muscular dystrophies. It started out just looking at Duchenne muscular dystrophy, but 10 years ago maybe expanded to all the muscular dystrophies. And now that data collection has been going on for a while across muscular dystrophies, one of the first things that will come out that I expect within, so what happens is that abstractors go to the medical record and pull key clinical elements out of the medical record in a de-identified fashion, put them into a massive database. So one of the things that we can do pertinent to limb-girdle is, and one of the earlier things that will happen is that we will be able to describe the period prevalence, so like over a 5-year period, how many people with limb-girdle were identified in these 6 surveillance areas, which we try to make as representative of the US as we can. How many people with limb-girdle, how many were genotyped, what genotypes are represented. So that information should be coming out within the next year. Ultimately, down the line, we've also collected a lot of clinical information and we'll be able to answer some questions about clinical progression of the disease. Did you have anything to add?
No, that was a really good overview. I started the Florida site for MD-StarNet a few years ago and then moved to Minnesota. So I'm still involved with the Florida site. And I think it's a really fabulous consortium. And I'm interested to see that we are able to keep asking more and more detailed questions, not only about epidemiology, but also now about disease progression, complications. And so it's a really wonderful consortium.
The question is, we do have genotype information. If it's in the medical record, we have it. There's a group of 5 of us clinicians who meet monthly, review all the records, go over all the clinical information, make a decision about whether or not this really is that disease and all of that. So it's curated to the best we can. Yeah.
Comment on the variability that we can see at the muscle biopsy in LGMDs. And sometimes, despite patient presenting a clear muscular phenotype, the muscle biopsy is not as bad as what we could have expected, and particularly maybe because of the localization of the muscle biopsy, tibialis anterior, it's a distal muscle. Is it important to select appropriately the muscle for the muscle biopsy to ensure featuring the right features of the disease?
Maybe I'll chime in just to start. The easy answer is yes. The other area that has struck me over time is there are times muscle MRI. And then I'll come back to biopsy. But there are times you'll do a muscle MRI on somebody that is incredibly weak, and it comes back remarkably almost normal. And then I distinctly remember a gentleman that we did an MRI of his thigh, and he's 60, he's still ambulatory. When I go to try to overpower his quadriceps muscle, I can't overpower it. When we got the MRI of his thigh, he had absolutely zero muscles in his thigh. They were all fatty replaced except for one rectus femoris that looked awfully good, and that's all he had. And by my exam, I had a hard time picking up that he had such extensive involvement. So if you have muscle that is too far along, you're going to wind up not getting useful information. And interestingly, if you're too early in disease, you may also, at least histologically, not get what you need. So imaging prior to biopsy will help you select a muscle that perhaps will allow you to see what you would want to see appropriate for what your study will be looking for.
And some of it depends on, H&E may be very different. It depends on the question you're trying to answer. So all the muscles will have immunostaining abnormalities, but the H&E may vary. Question over there.
How you doing? Thank you, guys. My name's Chris Carroll. I have LGMD 2D. My question is, with the clinical study on the group of sarcoglycans, if the study shows promising results for, say, 2E, can that be completely rolled out for then 2C and 2D, or does it have to go like each subtype by itself, since it is part of that same kind of cluster of enzymes there?
It's a good question. I think that's one of the things that's that we really would like to address, is that there are a large number of limb-girdle muscular dystrophies, and going gene by gene is difficult because we find the solution for one and then we wonder about the applicability for others. There are similarities among the sarcoglycan genes. They're roughly in the same ballpark in terms of the size. So for a particular technology such as AAV gene therapy, that's an advantage. But at this time, there's still a regulatory process that has to be gone through for each subtype. But yeah, if others have insights there, they'd be welcome.
Hi, I'm Michelle Campbell from FDA, so I'm going to give you that regulatory insight there. So I think this is when we would encourage, and there's some good sponsors here today who would probably going to say, yes, we hear this advice from the agency all the time, this is why we would say come early and have that conversation with us. So if there is something, as you're describing, where we can describe well the pathophysiology and the biology of the disease, and we have an understanding that we think there could be something that may translate to other subtypes, we want to have that conversation early of what would that trial design look like, right? Because we want to, make sure that we are utilizing our participants well. We don't want to waste your time. But we also want to make sure it makes sense from the study design. How can we measure outcomes? What is the safety data profile that's going to be needed on the therapy? And so this is why we would encourage this early conversation with us of how could this be done? What is a path forward? And so will always be unique nuances to development programs, but this is those kind of conversations we would want to have early with someone to really talk to us about what could that kind of program look like, because obviously we we know that we can, in diseases and disorders, they're kind of in the family concept. We are learning from one another, and so can we apply that? But this is definitely that example where we say, come talk early to us so we can start thinking about that.
Thank you.
So as long as you answered that question, the question that I think you're sort of getting at is, one, is it the same thing? And the answer is no, but the other side of that is if we have a well-established delivery mechanism and it's simply that the payload is changing, is there less regulatory hurdle that will occur?
I may not have the exact answer to that, but I think that's the example of why we would want, for example, to make sure we are bringing in then our colleagues who are the experts in that internally, right? So our CMC and our pharmaceutical quality folks that would be needed for that, we would want to bring them in early. And it may be, when I say early, they're always involved, right? But maybe someone wouldn't really talk to them until like the second or third meeting, right? And we want them at the first meeting scenario. And so I think that's why we would want to help identify those things, because that's an important thing. That's an important question. It's not just what do we know about our diseases and disorders, but how are we actually going to be providing this treatment? Is it a gene therapy, which has its own specificities? Is it going to be an ASO that's going to come to me in CDER with its own specificity? So this is why we want to bring in and be open to these questions and start thinking that out. And it may be a series of conversations, right? I don't think we will probably have the exact answer the first time, but it may be a series of conversations and dialoguing back and forth to really kind of collaborate together to see what the path forward could look like.
What's the prevalence on limb-girdle type 2B? And specifically, they're using a dual vector. And would it make more sense to tackle a simpler gene like a sarcoglycanopathy with a smaller gene? Question.
Two answers to that. I think the answer is already ongoing, meaning that that's where they're working right now, is with a gene that fits in easily to an AAV vector, so beta-sarcoglycan. But, and again, we have these specialists sitting in the front row that would answer the question about the dual vector, but it's, for me, exciting that we can maybe have other mechanisms, whether it be different delivery mechanisms or means to have portions of genes that then can re-anneal that will be effective. It's not quite answering your question, except for the fact that I think it is easier to do a gene that fits into an AAV, and that's why I think beta-sarcoglycan.
And FK.
Prevalence of 2B? And, versus the others? It's much larger. So again, we talked about from the Broad database, it probably is in that 7 per million ballpark.
Thank you to Dr. Kang, Dr. Matthews, and Dr. Wickland. At this time, we're going to welcome Dr. Nick Johnson to speak on the LGMD natural history landscape. Dr. Johnson is Associate Professor, Division Chief of Neuromuscular, and Vice Chair of Research for the Department of Neurology at Virginia Commonwealth University. He is also the head of GRASP LGMD Consortium. If we could welcome Dr. Nick Johnson at this time.
thanks. And thanks to my colleagues for hitting some of the topics to begin with, because it'll make the talk go faster and get you to break. So I have the auspicious duty of covering all of the work that's been done in terms of natural history studies in limb-girdle to set us up for treatment trials. So I will try and work through that. Here are my disclosures. All right, so this is actually taken from the FDA draft guidance on natural history studies for drug development. And so, this is really clinical research designed to capture the disease course, define the therapeutic population, develop outcome measures and biomarkers, and potentially serve as an external control population. So as I work through the smorgasbord of data that I have to share with you, I'm going to use these kind of topics to see, how we're doing on solving these. And I know we like to use the term natural history studies, but I really do prefer the term trial readiness, because we don't do it for fun. It's, it's designed to move towards a therapy. And so I think I'd love to see us continue to adopt that language. So this is a very brief summary of the prior and ongoing natural history studies in limb-girdle. And so you can see that there are a number of groups who have worked in this area. So I think for the GRASP LGMD Consortium, generally speaking, we have patients that are followed for 1 to 2 years. Many of these studies are ongoing. You can see it covers a number of different subtypes. Across these different subtypes, the proposed sample size is going to be over 400 individuals. We do capture clinical outcome assessments. Depending on the subtype, we capture muscle biopsies, muscle MRI, or blood biomarkers. We don't really measure cardiac outcomes particularly well, but we do capture pulmonary functions. Kathy spoke to the data in Iowa Wellstone study, which is really, the granddaddy of the limb-girdle natural history study. It's been going on for over 17 years with really over 136 different individuals. You can see covering the alpha-dystroglycanopathies, capturing muscle, MRI, and blood. Oh, yeah. I have an asterisk there because the studies where I have data that I'm pulling from for the rest of the talk, you'll see that I have an asterisk for that. The Genathon LGMD R9 study, it was ongoing and it lasted, participants are followed for 2 years. There's an LGMD 2A consortium that largely worked in Europe, and those patients were followed for 4 years. A lot of work and credit to the Jane Foundation and the Newcastle University for the CAUSE 1 and 2 studies, which really has over 330 individuals who've been followed for up to 8 to 10 years. The EU sarcoglycan study was a retrospective chart review that covered the sarcoglycans, and you can see 396 individuals. And then the Sarepta natural history study is ongoing and has 120 individuals. So really a whole bunch of different studies here that we have to pull from, to look at the data and see how ready are we to address the guidance for preparing for clinical trials. But before I start to move through that, it's also important to know that the LGMD patient registries provide an important role for collecting data as well. These really, they're listed there. There are many robust and active international limb-girdle patient registries. Many of these are managed by advocacy groups across several limb-girdle muscular dystrophy subtypes, and you can see them listed there. By and large, these registries right now focus as a contact registry. These are really a funnel or a pool to pull patients into these ongoing prospective natural history studies. But I think as the field matures, I'm hopeful to see these registries, become more robust and begin to collect more data that can be applied to trial design as well. And then what is important to individuals with limb-girdle muscular dystrophy? You're going to hear it in their own words directly after the break. But this is a survey that was done within the consortium, and you can see just asking individuals with a variety of different subtypes for limb-girdle muscular dystrophy. The most impactful themes included what you might expect: limitations with mobility and ambulation, hip, thigh, and knee weakness, and then inability to do activities. But if you ask those individuals who are no longer able to work because of the limb-girdle muscular dystrophy versus those who are continuing to work because of limb-girdle muscular dystrophy, I'll pull out a couple of different things that were statistically different. Number one, problem with breathing and respiratory issues, probably as a proxy for as the disease progresses. But then the other thing is problems with shoulders and arms, which I think, we do a very good job, I think, of capturing lower extremity outcome measures, but our ability to kind of capture that disability within the arms is something that I think we continue to need to work on. Hopefully, Lindsay Alfano has all the answers this afternoon about outcome measures that help us really capture that important and disabling feature. Dr. Matthews presented on this. So, how can natural history studies inform trials? So one way is, can you pre-specify or stratify patient population to better capture the disease in a more homogeneous group? And so one thing that we think about in limb-girdle muscular dystrophy is, can we stratify our populations by genotype? And again, as Kathy mentioned, work done by the University of Iowa group has really elegantly shown here, and she presented as well, that founder mutation, the FKRP, those individuals who are homozygous for that founder mutation do appear to have a modestly slower progression and function differently. So can you use genotype in LGMD R9 2I to begin to stratify your trial design? The answer is probably yes. What about in other forms of limb-girdle muscular dystrophy? So it's because in large part it's not been studied or it's been poorly ascertained. We don't have large founder mutations that have a lot of natural history data the way that it's been carefully collected in LGMD R9. But one way you can start to get at this is, can you look at the mutation and predict, is it a loss of function mutation or is it a missense mutation? And loss of function mutations are those that would be consistent with really complete loss of the protein. Missense mutations really, probably have some residual protein expression. And so in LGMD R1 or 2A, just in a small pilot here, you can see 3 individuals with a loss of function mutation versus 4 with missense mutation, and you can look actually at their protein function in these individuals here, and you can see particularly for some of these patients, there we go, where the between 2 patients, one with a loss of function and one with a missense, or those with a missense, and you can see that the age of onset does seem to separate a little bit. So I think there is probably an opportunity as we continue to collect these data and begin to classify mutations again into full loss of function versus missense. This might be an opportunity for us to stratify patient populations in clinical trials. This came up as well within the nomenclature. Can you use different disease terms to separate individuals in clinical trials? And here, this is work done by the Newcastle Group and sponsored by the Jane Foundation, looking at LGMD R2 or 2B compared to Myoshi muscular dystrophy. And you can see on the left-hand side, looking at the muscle MRI, that if you do the regression plot down there in panel C and look at the degree of involvement between those individuals who are diagnosed with Myoshi muscular dystrophy versus those diagnosed with limb-girdle muscular dystrophy, they're virtually indistinguishable. And then if you do a principal component analysis, either to look at the degree of muscle impairment, you can again see that there's a tremendous amount of overlap. So I think one of the challenges we have in our nomenclature system is that while these 2 disorders clinically appear indistinguishable, they are listed as 2 separate disorders, both due to the same gene mutation. So I think, should you separate allelic disorders on the basis of their terms? Probably not. And then the holy grail for something that is slowly progressive and heterogeneous is, can you begin to stratify patients by functional status? And this is data pooled from about 96 individuals who've been followed for 12 months in one of our different natural history studies. And you can see by subtype that across the group, using an outcome measure called the NSAD, or North Star Assessment for Limb-Girdle Muscular Dystrophy, that in general, again, in a large population, it is slowly progressive, about 5% progression per year. But if you separate out those individuals who have a progression greater than 10%, you can actually see there are 33 individuals in there who have a change of a mean change of about 20%, which is, I think, generally easier to study during the course of a standard clinical trial. The problem is that they are spread completely across that entire functional spectrum. And so I think we still have work to do to be able to capture those individuals who we think are progressing a little bit faster and perhaps more appropriate for early enrollment in a clinical trial. And this is just the same graph looking at the 10-meter walk-run. Again, the mean change across that group, about 6%. But there are about 20 individuals in there who have a decline of about 32% during that 12-month period, again, spread across a wide range of functional status. So I think there's work ongoing within the consortium about looking at either genotypes or biomarkers or muscle MRI that might help better predict who is more likely to progress during that time. So, defining the therapeutic population, how have we done so far? We think genotype-phenotype relationships may be important in subtypes with larger founder mutations, or perhaps continuing to classify the degree or the severity of the mutation by whether it's a true loss of function or a missense. And then that the genotype is more important than the clinical diagnosis in those allelic disorders, and that more work needs to be done to predict rapid progressors, I use that in quotes, using functional endpoints. In terms of developing outcome measures, I'm just going to touch briefly on the clinical outcome assessments because I know Lindsay will take us through in great detail this afternoon, but also to focus on the biomarkers. So we do think that the NSAD as a composite functional endpoint has the ability to serve as a key outcome assessment. It has good reliability between days within patients. And then if you ask patients how severe is their difficulty with ambulation, you can actually see it does a pretty good job of discriminating patient self-reported difficulty with ambulation in terms of the NSAD score. So I think it definitely is capturing the content that we want to, and it seems to be fairly reliable. It does represent a slowly progressive condition. So this is kind of the same data plotted a different way, looking at the percent change in the NSAD score over a 12-month period by subtype. And I think one of the key points here, and this came up earlier, is do we need to repeat these natural history studies or trial readiness projects across every different subtype? One of the lessons, if you look at the different subtypes, is that they tend to progress at about the same rate on average. And are really, to some degree, indistinguishable. And so I think I'm hopeful that we can come to a place where we can have an agreed upon set of endpoints, and we don't have to keep doing the natural history studies, particularly for those subtypes that are more rare. And this is just looking at ambulatory and non-ambulatory measures. So here, again, looking at the percent change on the 4-stair climb by subtype, and then looking at the 12-month change on the POL. Which does have more variation by subtype. But I think in general, you can see across these different forms of limb-girdle, the rate of progression is fairly similar. Okay, so that's the clinical outcome assessments. How about other biomarkers? What can we use to measure change in individuals with limb-girdle muscular dystrophy? I think quantitative fat fraction definitely represents an opportunity for us. This is data, again, Kathy showed on the left, looking at 23 participants with FKRP compared over 6 years, and you can see that in certain muscles, they do see a fair amount of change on that quantitative fat fraction. And on the opposite side, you can see an example of from our colleagues at the University of Florida measuring the quantitative fat fraction in panels A and D, and that the difference compared to Duchenne muscular dystrophy against limb-girdle muscular dystrophy. So this speaks to, for the limb-girdles here, while we may not be able to use the same muscles for each subtype because they are differently affected. You can see the soleus and vastus lateralis. As a modality, it may be sensitive in terms of detecting change. How about muscle-based biomarkers? So this is work using an ELISA looking at FKRP in individuals who participated in the GRASP FKRP study. 71 participants, and this is similar to what you might see in addition to the clinical data, that you can see a difference in terms of the between the homozygote and the heterozygote patients looking at the quantitated alpha-dystroglycan. And then this is work that was done by Jordi Diaz-Manera looking at the sarcoglycans. So this was a retrospective study and not controlled, but here you can see that when they quantitated the degree of sarcoglycan that was expressed in these muscles and used a cutoff of 30% more or less, there's a clear difference in terms of the loss of ambulation, both across all sarcoglycan patients, those with alpha, beta, and gamma is a little bit more mixed, probably because of sample sizes. But this is an opportunity where using the degree of protein that's both present or restored for the sarcoglycans may provide a meaningful insight as to whether or not those individuals will gain function over time. And then back to CalPain-3. This is again looking at 5 patients here. You can see with either 0% CalPain-3 expression, 10%, 50%, or 70%. With a wide variety of mutations, and you can again see that in general, that age of onset is separated by the percent of residual CalPain-3 expression, although it is a little bit of a mixed bag and probably due to the sample size, and that the progression on the NSAD is also a little bit separated based on the amount of CalPain-3 expression. So this is really an opportunity where these muscle-based biomarkers may provide a good insight in terms of both the rate of decline and the patient's functional status, both at the beginning of the trial and then may be predictive of how they do in the future as well. So in terms of developing outcome measures, the current clinical outcome assessments do demonstrate slow progression over 12 months. About 25 to 33% of that cohort demonstrates a faster 12-month progression. We have work to do in terms of pre-selecting or identifying those individuals and better characterizing them for enrollment in clinical trials. I would make a point to say that the progression is quite similar between different subtypes, and then that biomarkers in terms of both quantitative MRI and muscle biopsy biomarkers do offer opportunities to achieve an earlier signal of efficacy in what is determined to be a slowly progressive condition. Okay, and then I have just one slide because I don't think that we're here yet. It is mentioned in the FDA guidance that these natural history studies may serve as an external control population. You can see that within each individual study, there are up to several hundred individuals, but those are each individual study. And so I think we really, as a community, need to come together and begin to work towards aggregating these core data sets, really building the power, as they've done in Duchenne and other conditions, that you can use a predictive modeling to understand the progression for an individual. So I don't think we're there yet. But it may be appropriate to use multiple subtypes in these ultra-rare limb-girdle muscular dystrophies, where it's going to be very challenging to see patient enrollment of enough patients where you can truly detect efficacy in a small trial. And then the slow progression on those clinical outcome assessments does present challenges for well-powered placebo-controlled trials. So I'm excited to hear what the group comes up with this afternoon in terms of solutions to that problem. And with that, I will wrap us up. I need to thank all of our GRASP LGMD investigators, which you can see here on the slide, in particular, Chris Weil in the biorepository for providing some of those slides, and of course, those that support the network as well. And with that, I think we probably have time, about 5 minutes, according to this clock, or a longer break.
Hi, Dr. Johnson. Thank you so much. This was amazing. I was wondering to what extent clinical trial randomization rules permit you to ensure that, say, similarly, patients with a similar progression level are represented both in, say, equally in like the placebo control arm as well as the dosing regime. Because I think there was a recent clinical trial where due to kind of the way the different progressing patients were placed, the results might have been a bit skewed against the favor of efficacy. So I think there was like a fast progressing patients in the dosing regimen and maybe later onset slower progressing patients in the placebo control. And is there a way to use this data to make sure that doesn't happen and still comport with like the randomization rules? And
yeah, I think, and there's lots of other trialists in this room, so I'm certainly happy to pass the baton, but I can at least start by saying that those first slides where we talk about how to stratify patients, that's the type of work that can be garnered from a natural history study. So I think it is, particularly in some of these like FKRP, pretty clear that you need to probably pay attention to the genotype, and control for that and make sure that it's equally represented if it's appropriate within the placebo and the active arms. I think the other place where people often think about stratification is using those functional measures like the NSAD or 10-meter walk. I think there are, as I showed, still some challenges because our understanding of those is incomplete. But I think hopefully these data provide the kinds of things that you might control for. Jerry?
I just wonder, actually, to some extent, following Josh's question, you showed us five subtypes with NSAD that basically were the same. Is that the message, that the NSAD has little sensitivity, minimal sensitivity? Certainly 5 diseases would be unlikely to progress at the exact same rate.
Well, I think that's the question. I'm very interested in your colleague behind you's opinion as well. But I think that one message is that these are slowly progressive proximal conditions. And so to some extent, measures that capture slowly progressive proximal weakness are probably going to be appropriate no matter what the subtype is. I do think that we have more work to do in terms of understanding where we capture patients in that window of progression. And I think that's going to be one of the challenges. And I hope that as we continue to collect data, we can optimize the NSAD to increase the sensitivity, which is certainly a goal of the consortium.
And then I'll jump in since I got called out. But I would also say to that point that, outcomes are a measure of the cohorts that we enroll as well. And so I think it depends where these patients are in their disease progression. Jen and I were also talking about, these do progress in a stepwise sort of fashion when you talk to patients, but it's easier to draw straight lines between kind of a baseline and 12-month. And we do see some of those differences in rates as these trials get extended. So we've learned, I think, a lot in the COST consortium about some of those differences in which subgroups may be at that precipice that are most likely to have that large change versus those that are in a period of more stability.
We'd like to thank Dr. Johnson, and at this time we're going to take a break.
Hi everyone, welcome back. We are going to get started on the next session. So I'm Jennifer Levy, I'm the Scientific Director at Coalition to Cure Calpain 3, and it's my pleasure to talk with you all about the externally led patient-focused drug development meeting for LGMDs. So this meeting was held in September of 2022, and it focused on LGMD 2A, 2I, and the sarcoglycanopathies. I use the old nomenclature because many of the patients in our community were originally told that they had 2A or 2I, and that's still how they identify. So the purpose of this meeting was to educate the FDA and other stakeholders on what it's like to live with LGMD. So what's the burden of disease? How are people managing their symptoms? And what would a meaningful treatment look like? The video recording and the report summarizing the meeting are publicly available online, and I'll give you that information later. And this was conducted as a parallel effort with the FDA's PFDD initiative. This was a virtual meeting. We had over 600 individuals attend live and over 400 LGMD patients and family participants. So there were a number of ways that people could participate. We had people that recorded videos of themselves. We had people calling in and Zooming in live. There was also a survey that people were able to answer throughout the sessions, and people also submitted written comments. So lots of ways that people could share their perspectives. We also had observers from the FDA and industry and other researchers. So you could see we had people join us from all over the world, which was great to see. And we had pretty good representation of the different subtypes. So actually, the proportions of the subtypes really closely mirrored the prevalence that was mentioned earlier in the meeting today. So I'm going to summarize some of the insights that we got out of this meeting. It was really hard to just find one quote per slide, but I think that these really nicely illustrate what we learned. So insight number one is that LGMD affects multiple body systems. So like Dr. Johnson, we heard from lots of people who were very concerned about impaired mobility. So that's problems with walking and balance, but also difficulties getting from a sitting to a standing position and using stairs. We also heard from a lot of people about difficulties using their upper body. But aside from weakness, we also heard a lot about fatigue, pain. Some subtypes had issues with rhabdomyolysis, and certain subtypes have pulmonary and cardiac issues as well. We heard from many people that are severely affected by LGMD. In some cases, it affects every aspect of their life and robs them of their independence. This was a theme that came up over and over again, was talking about independence and loss of independence as your LGMD progresses. So some people rely on caregivers or family members for many of their activities of daily living, and some people require 24-hour care. So I want to read this one. This is from Vanessa. She says, "Weakness has taken away my ability to be self-sufficient. I can no longer toilet myself, bathe myself, dress myself, or prepare meals myself. I no longer have enough strength to put up my hair in my desired style since I can't elevate my arms. I slouch and prop my arms on counters to brush my teeth and to feed myself." So everybody with LGMD progresses, but it's at different rates. There are a lot of concerns about continuing progression, concerns about how much independence people will eventually lose. Adapting to that progression is challenging, and there's a lot of concerns about the future. Who will be able to provide care? How will activities of daily living be affected? And what's going to be the impact on school, on work, and on family life? This isn't always a linear progression, and things like catastrophic falls can really change the rate of progression. So Emily says, "My daughter was diagnosed at 7 and is now 16. We no longer can watch her run, walk, or stand up on her own. She needs help transferring from her wheelchair to her bed. I'm concerned about her loss of strength in her arms and how it will impact her independence even further." LGMD is stigmatizing. A lot of people have experienced discrimination and social exclusion. A lot of people express that they have anxiety or depression, and also feelings of grief and loss as their symptoms are progressing. Peter says, "I did not experience symptoms until I was 18 years old, so it's easy to remember what being normal was like. My bad days, I feel tired but able. My worst days, I'm still able but plagued with sadness and depression. In public, not only do I walk slower and with a gait, but I feel a deep shame and oftentimes feel like everyone is watching me." There are no FDA-approved treatments for any LGMD subtype. Everybody in this room knows that. That's why we're here. But we also heard a lot of feelings of anger, from frustration, because there's been research for years and years, but we still don't have a treatment that could halt or even slow progression. Dan says, "The progressive nature of it leaves me feeling terrified about what my future holds. As I live alone, I must face these challenges on my own. Any treatment or drug therapy that would even slow the progression of my condition would be considered a major victory in my book. Treatment approaches are supportive only, and they don't change the course of the disease. So we heard lots of people that are trying dietary supplements, also using medications that might manage the symptoms, so things like anti-inflammatories or medications for anxiety and depression. Really commonly, we heard about people using mobility aids, often needing home remodels or vehicle modifications in order to accommodate those. And what we did here was useful is exercise, aquatic therapy, physiotherapy. So Farron says, we supplement with vitamin D, iron, creatine, just anything that could potentially help him in any way. We're really trying. It's discouraging when you see the disease continue to take hold, but he's a strong little guy and he's doing a good job. People living with LGMD urgently and desperately need better treatments. Short of a cure, most would like a treatment that helps them regain strength, but many would welcome a treatment that just slows the loss of muscle function. We heard from a lot of people that said maintaining mobility is important, as well as cardiac and pulmonary functions. Kristen says, I would love to regain some of my lost muscle strength and function. However, I would be thrilled to simply stop the progression of the disease. Stopping the progression of this disease would be a huge success. The fear of things getting worse and increased disability from this disease is intense. And finally, individuals with LGMD want to participate in clinical trials. We did hear from people desiring shorter placebo durations or no placebo control, and that's really due to the progressive nature of LGMD. You know, when you have a progressive disease, there's a risk in doing nothing. And when you're talking about multi-year trials, there's a real concern about if I'm doing nothing for years, where does that leave me? Many people would accept a small number of trial participants. We heard from a lot of non-ambulatory individuals that would like to be included. And we also heard from a lot of people that said endpoints shouldn't be limited to ambulation. So certainly arm strength, upper body strength, and respiratory functions were very important to the people that we heard from. Stephen says, participation in a clinical trial is always voluntary and a patient can withdraw at any time. Are you going to stay in a trial for 3 years once you realize you're on the placebo? So what I'm presenting today is really just a taste of what we learned from that meeting. So LGMD PFDD is the website, and that's where you can get all of the information. We have a pretty thorough report. We have the video recording. Part of that video recording, individuals made recordings of themselves doing activities of daily living, coping with the disability that they're living with. So there's a lot to learn just by watching those. We also have all the survey responses, and I just included this as one example, asking about what medications do people use. But we have all of those, as well as a complete list of the written comments. We had such great participation that we really wanted to include everything we got. All of these are broken down by subtype, broken down by topic, so really easy for you to search for what you're looking for. And then we also have a benefit risk assessment framework. So please look at these, consider them as you're in the process of developing drugs or in the process of making decisions on behalf of patients. It's a resource for you. If you have somebody new joining your group, you know, statistically, they've probably never met somebody with LGMD, but this is a great way to hear the perspectives of a large, diverse group of people. So I really want to thank all of the patients and family members that participated that day and contributed to our success. And I also want to acknowledge the sponsors that we needed to make it happen. So that would be Sarepta, Edgewise, AskBio, ML Biosolutions, and Vita Therapeutics. Thank you.
Thank you, Jen. She did such a wonderful job helping with the PFDD. And at this point, we're going to do our patient panels. Before we start this, I also want to just acknowledge our patient delegates that are in the room. In addition to those who are speaking, we have patients that have come and taken their time off of work to be here with us today. And they're all throughout the room. I've known some of these patients since they were 12 years old, and now they're in their late 20s, looking and waiting and hoping still for treatments. And they're here today, and I want you to meet some of them. So when we have these breaks and lunch, make sure that you get to meet some of them and just hear from them. They've got wonderful stories. Today, you're going to hear a video. I'm going to start with a video because with LGMD, a lot of times you don't realize the burden and the early age onset for limb-girdle. But as you'll see in this video that I'm getting ready to show you, this is going to demonstrate that families are often finding out that their kids have LGMD at a very, very young age. Sometimes they're finding out that not only one of their kids has it, but all of their children have it. That's devastating for a family. And so this first video is just, it's hard to watch, but it just shows the burden and how early age onset can occur. So if you'll cue the video, and then there will be another video after that, and I'll share more about that one.
When we were in our meeting with the geneticist, they tell us about, our diagnosis of LGMD2I. And then he said, I think you mentioned you have another child. Through all of this, I wasn't even thinking that whatever was happening with Jackson could be something that would impact Chloe. He said, well, there's a 1 in 4 chance that your daughter will also have this.
And again, you're feeling pretty good, right?
1 in 4, I was like, okay, 25%. Like, all right. We already have one who has it, so really we should be able to have 3 more children without it before you have one.
Surely life isn't that cruel.
Yeah. So then when we got her diagnosis, it was just terrible. As soon as the kids went to bed, we would just like cry. And it was so awful. Parenting is hard to begin with, and then you throw this on at such a young age, and we try to spin everything kind of in a more positive light, where now we think of it as it'll be something that bonds them in a special way, and they'll have somebody who understands what they're going through. Since it is so rare, it's so hard to find somebody who's going to understand that, and they're gonna feel isolated and alone. So at least they can talk to each other as they start to experience some difficulties.
Every day we just hold on to the hope that there will be a treatment for our kids at all or in time.
Yeah.
You are doing it. I started dancing when I was 4, and it was the one thing that kind of helped me escape from the world.
I dance as well. I dance hip hop, and there was this, I remember it so distinctly, there was this one move where I was in the front row, and you had to like get down on your knees and then come right back up without using your hands. But I would always like use my hand to like push up on my knee to get up. And my teacher would always correct me and be like, oh, you can't do that. I'm like, well, I can't do that either, so I don't know what you want me to do. And I was young, and I was kind of like, okay, I'll try. It didn't work. When things started getting harder for me, I was definitely on the younger side, I would say, just from like the type that I have. It got harder quicker. I had to get a lot of things like modified for me. I stopped dancing and kind of doing like most physical activities. I played basketball, I stopped gymnastics, stopped softball, all of that. My whole family kind of did dance. My mom and my sister both did it. And my sister was on the competition team that was a couple of years older than me. But I feel like the one thing that did kind of help me out a lot was throughout kind of my whole life, like up until now, I feel like I kind of live like vicariously through her. Like I went to all of her competitions. I kind of got like the same rush just by watching it.
Sometimes you lose the ability to do something, and sometimes it's small. Sometimes it's realizing like, oh, the milk on the top shelf of the fridge, I can't easily get that down anymore. Like I'm noticing it's harder. It's letting yourself grieve that, and it's super difficult.
So I almost felt a sense of relief to have a name put to what she had, but then knowing what that meant was devastating. And we kind of made a choice. As soon as I found out the news, I let myself be upset, but then I decided we're having a good life despite this. Last year, she was really into gymnastics and cheerleading, and she was having a fantastic year strength-wise. And then in August, she lost her ability to do handstands and cartwheels, and she was devastated. She said, "Mom, I just want to be like everybody else. I'm scared, and I don't know how much worse this is going to get."
I used to like do cartwheels and like tumble and dance. It's tiring and it can make me very frustrated sometimes. And I think it's hard because everyone sees me like laughing and like talking and stuff, so they think like nothing's really wrong with me because like you can't really like see it. So it's definitely really hard to deal with that too.
She's definitely has a good spirit and attitude, but she gets beat down and tired of it too. And she just says, "I wish they can fix this for me. I wish I didn't have to think about having pain before I have fun."
I remember one time I was at a pool party and I had just had a sleepover. It was a birthday party. And then they were all like, "Let's go to a trampoline park." And I had to like feel like, I can't do that. I like want to do things. And sometimes I will do it, and then the next day I'll be in like so much pain I can't even move. So I also like kind of get left out sometimes with it, so that's also hard to deal with too.
There's plenty of children who have this. If we can halt progression or find a cure for them now, you're giving them back the rest of their lives to live right now.
You're speechless when you see these families and these children and these teenagers talking about the burden of living with a disability. And here's something that's become very difficult. We have a large limb-girdle community. These children, these teenagers, these young adults, they see the lifespan of what limb-girdle looks like at teenager years. They see it in their 20s. They see what it looks like with other people in their 30s. They see what other adults look like when they are in their 40s with it. And we see the lifespan of what it looks like to lose abilities every single year. And it's important to acknowledge that halting progression prevents a life of disability, and even reducing that by 50% for someone means you're getting back your life. And we want to do that for these kids. We want to do that for these teenagers. And I don't want anybody to have to be in my situation if we can prevent it. Donovan's going to be speaking next. We have a video, and I really want to highlight Donovan Decker. He was the first individual to receive any gene therapy for muscular dystrophy, and he received it in 1999. In 1999 for limb-girdle. It was an intramuscular injection, and it was mentioned already today by Dr. Mendell. I just want to highlight something to you. This didn't happen in 2020. This happened in 1999. And then from that, all of things you've seen that's happened for muscular dystrophy came, a lot of that knowledge later on came from limb-girdle. So there's a huge, rich history of science, and Donovan's going to share it. Let's give him his voice.
My name is Donovan Decker. I was diagnosed with limb-girdle muscular dystrophy 46 years ago. There are 8 kids in my family, and 5 of us have limb-girdle 2D. Plus, I have 2 nieces that have limb-girdle 2I. My family knows the heartache of this disease, as I've lost 3 sisters, Monica, Mary, and Jolene, to respiratory failure at the age of 53 57 and 65, respectively. Many COVID patients died in the same way my sisters did, from respiratory failure. With COVID, we learned that the companies and FDA can work quickly together to help save lives. We need the same urgency in the muscular dystrophy field. I urge the FDA to allow diseases like muscular dystrophy to use accelerated approval. I also urge the FDA to look at natural history studies and historical controls instead of placebo controls whenever possible. If trials are forced to use placebo controls, this will hurt for recruiting and the patients will lose muscle when they could possibly be helped. My forced vital capacity is down to 21%, so I might have 4 to 7 years to live because of my limb-girdle dystrophy. And it makes me angry when I hear limb girdles are not considered as severe as other types of muscular dystrophy. 24 years, 5 months, and 6 days ago, but who's counting? September 2nd, 1999, I was the first person in the world to do gene therapy for any form of muscular dystrophy with Dr. Mendell. Mine was the local delivery. And my sister June was the first person to ever do vascular delivery by an infusion into her lower limb. My sister June and I risked our lives when we did the gene therapy. Because of our family, most patients I've talked to are willing to take risk and even stop the progression, and getting help could change our quality of life. It would be great. Non-ambulatory patients need to be included in trials, and we must update outcome measurements. Thank you.
And what a wonderful testimony and kind of story from him. So for those to share a little bit, it's an intramuscular injection that he had in one muscle. So obviously it's not systemic. His sister had a tourniquet on her arm, and it was so it was not throughout her body either, where it went everywhere. I did actually get to meet a patient that actually had systemic delivery accidentally through his body, and that patient is I can't even tell that they ever had limb-girdle. So something there to think about. Now I want to share with you Pat Motion was supposed to be here today. He's an individual living with limb-girdle 2E. And Pat is an incredible person. I don't know if you've ever met him, but he is like one of those individuals, when he comes in a room, he lights up a room. And when he talks, you listen because he says stuff. And that's Pat. Well, Pat was supposed to be here. He has 2E, and he got sick. And he asked me to share this important thing with you. Patrick is a 51-year-old devoted husband and father of 2 to Tim and Ted. He has limb-girdle 2E. He's a beloved pillar of his community in southern New Hampshire. He thrived as a middle school music and band teacher for nearly 3 decades, building an untarnished legacy as a decorated educator who used music, culture, and performance to promote positivity and inclusion and creativity. In 2023, he transitioned out of education to take on new challenges, working in advocacy for Parent Project Muscular Dystrophy, which we all know, which is a leading organization in the global crusade against Duchenne and Becker. He's traveled the world as a speaker, and Patrick has modified his home. He drives a wheelchair van, and he's got an adaptive home as well. But I want to share with you the reason why he's not here today is 2E almost took his life. At the start of the new year, Patrick was hospitalized with a severe respiratory infection, and he had cardiac distress. He was sedated, intubated, and med-flighted from his local hospital to Boston after suffering from multiple arrhythmias. Over the past few weeks, he was unsuccessfully extubated twice to BiPAP, and like so many, finally found relief with a tracheostomy after the third extubation. It's estimated his heart ejection fraction was 15 to 20%. And as we looked at natural history for the sarcos, that's not unusual, but it's terrible. He had a pacemaker defib put in after his 2-week stay in ICU. Doctors are hopeful that this device will increase his ejection fraction. Today, he still has a collapsed left lung, and he's in a rehab hospital, but he's watching right now. I'm telling you his story so you know what it's like for a 51-year-old person living with LGMD2E. It is not a mild disease, but with treatments, it doesn't have to be this way. LGMD is not a mild disease. It's not something that just old people get. Pat is watching today via live stream from a rehab hospital, and we want to say to you, my friend, you are a brave warrior, and we are honored to be in your company. Get well. Yeah. And at this time, our in-person patient presenters are going to begin, and we are starting with Rachel DeConti, and she's going to come up to the podium.
Mr. DeConti, that's what I call him. I call him this because he is such a mature young man with a big heart and the utmost respect for everyone he encounters. I look forward to seeing Mr. DeConti enter our school because I know that he's entering with a "Good morning, Mrs. Lawrence." That melts my heart and makes my day. It is important to Mr. DeConti that everyone maintains a high level of respect for adults and each other. Mr. DeConti encompasses our Ruth Chafee cardinal rules of being safe, respectful, and responsible. We need many more Mr. DeContis in our world. I love him, and I look forward to watching him grow into an amazing young man. Good morning. I am Mr. DeConti's mom, Rachel. Those sweet words were sent to me by my son Jacob's school principal, Mrs. Beverly Lawrence. It is a great description of the child that he is and the impact he already has on everyone he meets at age 7. Our world forever changed when Jacob was diagnosed with LGMD type 2D-R3 just after his 5th birthday. He suffered a case of rhabdomyolysis during summer break. At the time, we thought the dark urine was from a scooter fall and rushed him to the ER. We had no idea of what was to come. After many tests, we learned that Jacob's CK was extremely high, and we were in the hospital for 3 days on IV hydration. During a second hospital stay shortly after, he had a genetic test which confirmed LGMD 2D. My first question, still in disbelief, was will this shorten my baby boy's life? Our doctor's response, it could, because LGMD affects respiratory and heart functions. After this diagnosis, my husband Josh and I pivoted from being just Jacob's parents to devoted advocates for Jacob and the entire LGMD community. I've been privileged to become the executive director of the LGMD 2D Foundation with a mission to support research and clinical trials to speed the development for treatments and a cure of LGMD 2D, and we're not slowing down. Jacob is currently in the 2 available natural history studies or trial readiness, like Dr. Johnson mentioned earlier, for sarcoglycanopathies. Jacob is contributing to the cure. Last year, we traveled 4 times to multiple states for these studies, which involved him getting blood taken, multiple hours of MRIs, cardio appointments, bone density scans, and physical therapy assessments. This is a lot for anyone, let alone a 7-year-old. He is doing everything that our medical teams ask of him, and he's doing it with encouragement. Last year, during our first trip to Nationwide, we told Jacob that he was trying out for Avengers training camp. How else do you explain to a 7-year-old Spider-Man loving boy what's happening to him? The hospital was so amazing, and at the end of our visit, he received the award of being the most brave patient by our hero, Dr. Mendell, in Spider-Man. We still try to protect his young mind from worry whenever we can. He knows we visit Nationwide and now Dr. Johnson at VCU to keep his muscles strong and to avoid dark urine, as he calls it. And there are so many LGMD patients that do the same. We know we are not alone. We heard from Dr. King earlier, and it is clear that LGMD, especially the sarcoglycanopathies, is not a mild disease. The sarcoglycanopathies are the most severe subtypes due to the impact on heart and respiratory function. SOCRs are often referred to as DMD-like. As a parent, that is extremely difficult to hear and comprehend. The age of onset is typically during childhood, and the progression during these younger years can lead to being wheelchair dependent by 15. Some of the scariest and most concerning complications for sarcos, as we heard, are breathing difficulties and heart problems. No patient should have to go through each day knowing tomorrow it may be harder for them to catch their breath, be stuck on machines all day and night for air, or worry about their heart muscle weakening. That is torture. These are the things that keep me up at night for all LGMD patients, now and in the future, and the fear that this will happen to my son if treatments don't come soon. There are several challenges that patients face. As a child and throughout teenage years, patients can feel socially isolated, picked on, or left out by friends and classmates because they cannot do the same things as their peers. Children and teens also need to navigate the feeling of loss much younger than they should. They often lose independence before they are old enough to gain it. Adult patients have challenges that only true warriors overcome. They worry about continued weakness, no longer being able to feed, bathe, or transfer to a toilet or bed. Things that most of us in this room do daily without even thought. They need to gain more reliance on family and friends to help them during these daily tasks and worry what will happen as these caregivers age. Our big ask for a treatment is to stop or significantly slow down disease progression. Improved mobility or leg strength is important, especially in ambulatory patients. For non-ambulatory, gaining arm strength would be a huge benefit. Additionally, increased lung capacity and reduced risk of heart complications are critical outcomes for all patients. Our patients are willing to do almost anything for a safe treatment. We are willing to take the risk as patients and caregivers to enroll in a gene therapy trial, knowing that the next generation treatment may not be far behind. We heard from Dr. Mendell, Catherine, and just now Donovan, who took that risk over 25 years ago, being the first patient to receive gene therapy for muscular dystrophy. The first patient to receive a gene therapy for muscular dystrophy was an LGMD 2D patient. And yet, almost 25 years later, we still have nothing approved for our community. As an outcome of this workshop, we urge you to please consider the following. Leverage years of data from previous natural history studies and gene therapy trials. Consider creative ways to for treatment development and approval, like umbrella approaches for sarcos, drug repurposing, or off-label uses. Treatment development shouldn't be slowed down because of natural history studies and patient surveys. We have years of data already. We ask everyone in this room to act with a sense of urgency. We cannot continue to wait. The words on this slide are some of the many examples of what treatment would mean to our community. Gaining independence, being able to breathe easier, or living the lifespan they deserve are powerful yet heartbreaking asks. I want to thank you all for this unbelievable and unforgettable opportunity. Being in this room today means that each of you are committed to the LGMD community and to the patients here and on the screen, including my son, Jacob. Every minute of every day that we don't work together to accelerate treatment means that their muscle is wasting. Thank you.
Brooklyn is now going to share a short story. And she's going to, I think, look at you. She's going to get at that podium. Because she's just awesome. Brooklyn's a teenager. And I don't know about you, but I would have been talking in front of this group when I was a teenager. But she's doing it. She's brave. You go, Brooklyn.
Okay. Hi, my name is Brooklyn Garza, and I'm 16, living with Limb-Girdle Muscular Dystrophy 2A-R1. The signs I showed before being diagnosed were going up the stairs funny, walking/running slower than other people my age, dropping my baby sister on the coffee table, not being able to catch myself while trying to learn a cartwheel, and trouble shampooing the top of my head. When looking through pictures of when I was younger before I was diagnosed, I was surprised to find ones that showed early symptoms. The picture you see of my back in my ballet costume shows the beginning of my scapular winging. The push-up picture shows the beginning of my lordosis. These were when I was blissfully unaware I had a muscle disease. I wish I could go back to that time. After age 9. At age 9, after 6 months of being tested for multiple things, I was diagnosed with Limb-Girdle Muscular Dystrophy 2A-R1. 4 years after diagnosis, the contractures in my legs had started to affect my walking. I walked uneasy and was falling a lot. The summer of 2020, I had double heel lengthening surgery. This helped me continue walking but forced me to spend one whole summer in hot Texas with double cast as I recovered and learned to walk again through extensive PT. Sammy has 2I, and I met her at the recent conference last October. We have very similar stories which helped us become friends very fast. It can be really lonely and isolating not having anyone your age who can relate to you living with a disease like this. I've done multiple things to further research for a treatment/cure. These include being in Nationwide Children's Hospital Natural History Study in Ohio, GRASP 1 and 2 at NCH. As of last week, I'm the very first patient for GRASP 3 at VCU. I was an FDA Voice of the Patient contributor, a panelist at the International Limb-Girdle Conference, a contributor in LGMD magazines, and a fierce advocate/fundraiser for the Coalition to Cure Calpain 3 every month of September with a goal of 10,000, plus a company match of 10,000 from my grandparents' business. As a 16-year-old, this disease affects my life tremendously. For example, I need help getting out of bed, dressing, bathing, and more. I miss a lot of school and fall behind in classes due to multiple appointments. I have to make sure there are no stairs everywhere I go, and if there are, I have to see if there's an elevator. I also check to see if someone is there who knows what I have so I can get the help I need. I miss out on social events with friends, and I have to leave early due to fatigue. I am always cold and often overly tired. I have weaknesses that affect how I carry things, drive, etc. I have to use a wheelchair or a scooter for any long-distance walking. Since being diagnosed 8 years ago, I've gotten weaker. It is now impossible for me to get up from the ground without pushing up on something or just having someone help me. It is extremely difficult to get up from my desk, toilet, bed, etc. Things that would help me are stopping progression and being able to stay where I am right now. Any type of treatment that would help with slowing down the disease. A trial to open up while I'm still able to walk so I would qualify, or my biggest dream would be a cure that allows remaining muscles to grow and strengthen for all who are struggling with this terrible disease. Please provide access to treatments and a cure for this muscle-wasting disease. Time is not on my side, and it would be life-changing to not live with the lingering worry of progression to not walking and not being able to use my arms. I humbly ask for your help today. Thank you.
Brooklyn, you are the rarest teenager I've ever met in my life. Give me your hand. We told her, I said, Brooklyn, you don't have to do this. And this child said she was going to. And I'm just so proud of you. And, you know, you're a brave warrior. Okay, our next person up is Kelly, and then Josh will close. And then we will actually go to lunch.
All right, I'm going to try to go after that. Hi, good morning. My name is Kelly Brazo, and I'm the president, CEO, and co-founder of the Cure LGMD 2I Foundation that my husband and I founded when our daughter Samantha was diagnosed with LGMD 2I at the age of 2. Oh, I hit the wrong thing, sorry. Hand's shaking. I'd like to give you a little glimpse into Sammy's life, how it's been challenged by this rare and progressive disease. 15 years ago, when Sammy was only 1 year old, we started seeing symptoms of tripping and falling. She was much slower than her peers and had trouble getting up from the floor. 15 years ago, we started on the path of finding her diagnosis. We met many appointments, pretty much all over the East Coast. Her first blood work revealed CK levels between 10 and 15,000. We heard, well, if she were a boy, I'd think it was Duchenne. So we just thought we had ruled out any form of muscular dystrophy at that point. Ultimately, we ended up at the NIH for a limb-girdle genetic panel revealing compound heterozygous form of LGMD2I R9. And probably many of you recognize Dr. Bonneman in this picture who diagnosed Sammy. Sammy's early years were very active. We kept her as busy as possible to let her enjoy the activities that we knew would not last forever. She loved playing tennis, softball, gymnastics, dance, you name it. But not long after, we had to give up those activities in exchange for physical therapy, a lot of physical therapy. Since that time, Sammy has participated in 3 different natural history studies. She's enrolled in the International FKRP Registry, which includes 11 years of curated data. There are also 15 years of rigorous, reliable, and unbiased natural history study data for the district leukonopathies, which has been collected by Dr. Matthews' team at the University of Iowa. And yet today, there are 0 approved therapies for LGMD2I. Sammy has done all that she can do to contribute to science. And still, there is no approved therapy for our 2I community. As a parent, I have done more than just caregiving. I've committed my career to advocating and supporting drug discovery and development. What more can we do? In the absence of significant change in the way drugs are developed and approved for the LGMDs, Jacob, Brooklyn, and many of the patients in this room today are going to be standing here like me today in 15 years. In the last 3 years, Sammy has suffered many losses. She falls often and is not able to rise from the floor independently. She's gotten stranded in the middle of her high school hallway, waiting for someone to pick her up off the ground. She can no longer enter her friends' homes, as they have stairs without handrails, which she cannot ascend. Although she's an avid sports fan, she cannot climb the bleachers to sit with her friends to cheer on her favorite teams. She can no longer independently sit to stand, which leaves her unable to use a traditional bathroom in any typical seating in a classroom or restaurant without someone to lift her. Sammy recently mentioned that the constant vigilance and planning it takes to make it throughout her day is mentally and emotionally exhausting. Additionally, in the past 3 years, Sammy has had to endure 2 surgeries due to significant issues with contractures. 3 years ago, Sammy had her first surgery for an Achilles tendon release, just like Brooklyn talked about, and was in 6 weeks of serial casting following that surgery. Just 10 months later, we found out that Sammy had a 75-degree curvature of the spine, and she had to undergo a complete spinal fusion, which required severing 5 of her ribs to make the correction. Due to the extreme blood loss from that surgery, she had to receive 2 blood transfusions following. She had an extremely painful recovery while she stayed in the hospital for over 3 weeks, where she basically had to relearn how to walk all over again. In addition, we're constantly concerned about the cardiac and respiratory implications of this disease. Sammy had her first cardiac MRI just a few months ago, which revealed early signs of fibrosis and a low normal ejection fraction. She also has diaphragmatic weakness, so anytime she has a respiratory infection, she has a very difficult time clearing her secretions, requiring a cough assist. She has extreme fatigue and often pain when she pushes herself too hard. On behalf of Sammy and our entire LGMD2I patient family, we urge you to consider utilizing the 15 years of natural history data as a control arm. We can't wait for the traditional 3-year double-blind placebo trial format. We ask you to consider accepting alternative designs for studies and use of novel technologies and surrogate endpoints to facilitate accelerated approval pathways. LGMD is often referred to as a late onset and slowly progressive disease, and that is simply not the case. We can't afford to wait another 3 years. Every day that goes by is a risk of falling with potential fractures, respiratory failure, cardiac failure, and irreparable muscle loss. I don't know why, I'm sorry. In 3 years, Sammy will be a freshman in college. Having an approved therapy available for her would be the difference between her going off on her own to college to study medicine versus losing out on the opportunity to follow her dream of being a physician herself. Thank you on behalf of our entire LGMD patient community for being here today to hear our stories and to consider our pleas for an opportunity to make a difference for all the lives affected by this terrible and heartbreaking disease.
Josh is going to come up now, and he lives with dysferlinopathy, LGMD 2B.
Do I just say next slide? I don't have a clicker, or do I? Yeah. Oh, thank you. I think I can. It's just the green. Yeah. Hello, everyone. I'm so honored to be here. To be here today with all of you, and feel almost selfish as a late onset patient, asking for inclusion of non-ambulatory patients, but it's obviously part of my ask. So I'm gonna discuss my journey with dysferlinopathy. And then I'll touch on certain clinical trial elements that I consider crucial for us. My journey as someone whose symptoms first appeared in their late teens to early 20s is fairly typical. I first started to notice weakness in my teens and early 20s, but I just figured I was out of shape. It wasn't until I was about 22 years old that it became obvious that it was more than that, and I sought medical attention. And I'm 58 years old, so when I was 22, it was 1987, and the genes hadn't yet been identified. So I was diagnosed with Miyoshi myopathy, and that tends to, as you've seen in, I think, both Dr. Mendell and Dr. Johnson said, it's a phenotype that tends to present more distally initially, but over the years, the symptoms merge, and we now know that it's the same disease. So in 1998, when I was 33, the genes started to be discovered, and the DYSF gene linked Miyoshi myopathy to LGMD R2. I'll use the new terminology. So I promised, I promised not to have any models who were unclothed or who were nude in my pictures. You saw enough of that earlier. But I will say that picture of the fellow with the pen t-shirt and also the dysferlinopathy patient in Dr. Johnson's slide is a pretty good representation of my gait and how my lower legs looked at those ages. But here's some slides from when I was 3 to when I was 37. You can sort of in hindsight see some progression, but so obviously by the time I'm 37, which is shown in my favorite picture, I think that I have of my wife and me, I was using a cane. So since I was diagnosed, I really threw myself into whatever I could to help researchers and clinicians learn more about my disease and potentially how to treat it. And I've listed here some of the activities that I've been involved in since I was 22 years old. I just want to quickly point out that on this slide, you can see some of the true heroes in my life. So Robert Brown, Bob Brown is become like a friend of mine. Dr. Mendell, I was thrilled to be able to reacquaint with at some of these conferences, but he was the PI back in 2000 of the gentamicin study I was in. Dr. Amato, who worked with Dr. Mendell years ago, is like separated at birth, wonderful patient-oriented doctor. And I got to not only be in the cost study, but I got to negotiate the contracts with Newcastle University for it. So that was fun. And I think, you'll find that you have ample testimony here, but we really want to be your partners in your search for knowledge about these diseases. And we want to help, even though at times we encounter what seems like a very unfriendly medical and research community. This really happened to me. I was getting an open biopsy to see, looking for the presence or absence of the dysferlin protein, and the surgeon looked into his incision, and this is what he said. And he wasn't smiling, he sounded angry, he sounded inconvenienced. The nurses were horrified. I was gagging in pain because in 1999, anesthesia was believed to damage the DNA sequence. So they didn't use it. But you know what? He was right, because absent a therapy, we no longer will have muscle tomorrow. Oops. Oh. Sorry, I hit the wrong button. Yeah. So my clinical trial design preferences are on this slide. You've heard already from Brooklyn and about Sammy and about Jacob, by their moms, why these are important. I'm also thrilled to see how central these elements are to the presentations and discussions today. So I just want to emphasize that, these are synergistic. They work together. And I urge you to validate them and use them together as soon as you can to protect us from tomorrow. We're not looking for a miracle cure, and there's ample testimony of that in the PFDD quotations from our presenters today that we know that gene therapies will work more effectively in pre-symptomatic patients, and we applaud a future without these diseases, right? But that's not the end of the discussion. The standard for approval is whether a drug's benefits outweigh its risks to the patients. And we ask you to take that seriously. So I'd say, well, it's certainly up to the FDA, the drug sponsors, and the clinical trial PIs to determine whether or not clinical endpoints have been met. It should be up to the patients to explain to you what endpoints are clinically meaningful in the first place. And I have a list of what I would love to see further measured and adopted for use as primary clinical endpoints for, well, certainly for non-ambulatory patients, but I would say for any LGMD patient, to the extent that they're, the group where these are measurable, because each of these is relevant to our continued independence, our continued sense of dignity, and our well-being. Rolling in bed without assistance is a huge thing. I woke up last night feeling panicked and claustrophobic. And, fortunately, Una was right next to me, and she was able to wake up and roll me on my side. But so these aren't just convenience items. And, you're using clinical endpoints now that don't measure things that need to be measured. And you've got to get this done, because otherwise you're looking for your keys where the light is bright instead of where you lost them. So I loved Dr. Johnson's presentation, and what I was going to sort of cover was the stratification topic that he already brilliantly covered. So I'm just going to repeat. I think all of you sort of know and agree with the factual statements on this slide. So I'd just like to repeat what my patient peeps here said, which is, you've been collecting this data for a long time. And, we've believed it was for a purpose through all of the years and hardship that we've experienced to generate it for you. So it's sort of the concept, the idea of the FDA not wanting to use it as a control is just heart-wrenching for us. Oh, you know what? I'm sorry, was I? Yeah, so I wanted to touch quickly on accelerated approval. So accelerated approval has been a powerful tool for years. It was actually the result of patient advocacy by AIDS patients and their families. And it is every bit as applicable to the LGMDs as it is to any other disease. Each of our conditions is caused by a known protein, an identified protein, due to mutations in an identified gene that's related to it. And so we know, therefore, that the absence of the protein or the replacement of the protein are the ideal surrogate endpoints for these diseases, for all of them, regardless of subtype. And so I guess I would just, argue, using something that I think the FDA is very familiar with is, in my opinion, the replacement of protein should be every bit as reliable a prediction of clinical benefit as tumor shrinkage in cancer patients. I could say so much more, and lawyers can never shut up. But I am a little bit more emotional than I thought I would be. So I just want to thank everyone who's here. I find this to be, unique in my life. There have been so many great conferences the Speak Foundation has put together, that the Jane Foundation has put together. But this one's just very different, I think, with all the different people, all the stakeholders, and fully clothed pictures of non-ambulatory patients with different forms, subtypes, although I have a bias. So that's me and Una with our little dogs. And then that's me and Bliss Welch and Brad Williams, who's a hero. We all have 2B dystrophinopathy. And Melanie Bowe, who has one of the sarcos, and she's with Yumi Shim, who's here today. That's Melanie's husband reaching up with the mic. We were doing a presentation on travel and leisure. So in the bottom middle, that's me moderating that panel. And then bottom left is more recent. That's Brad again. He's so nice. He gets to be featured twice. And he's with K-Tran from Toronto and Jessica. Both, and Kay also has 2B. And that's Kyle Harrington. In the lower left, we're trying to do our sailor impersonation or something. She also has 2B. So, thank you all very much.
Great job. Great job, everyone. Thank you for sharing your stories, and we will not forget them.
Good. All right. Good afternoon, everybody, and welcome. Before we get started, I would just like to first introduce myself. I'm Annie Kennedy, and I'm the Chief of Policy, Advocacy, and Patient Engagement with the Every Life Foundation for Rare Diseases. But I've also been in the neuromuscular space for a couple of decades. And I was reflecting when we were listening to the presentations earlier, and they were incredible. So I just want to thank everybody who shared their journeys with us previously. That the first time I ever set foot on NIH was 1999, and the Recumbent DNA Advisory Committee, is known as the RAC, was hosting a meeting around the downstream implications of gene therapy. And I believe it was April or May of 1999. And at the time I worked for MDA, and they had invited us to come and speak about the gene therapy implications for patients with Limb-Girdle Muscular Dystrophy. And so I went with a woman who was in her early 30s, who lived here in the DC area. Her name was Muffy Levine, and she was Spitfire. She was amazing. And we went with her service dog. We went onto the NIH campus. I'm pretty sure that RAC had no idea what they were going to get from Muffy that day. And what the RAC was struggling with was what the downstream effects of gene therapy would be for individuals who had received it if they were to have children. It's a concept that they were really wrestling with called gonadal transfer. So essentially, if there would be harm to the children of children who had received gene therapy someday, what would the implications be for future generations? And I'll just never forget Muffy taking the RAC to task around the fact that she, as a 30-year-old, was having a hard time getting a date, and she wasn't really going to worry about whether her future unborn children were going to sue NIH someday. But what then happened was that in September of that year, there was a really serious death in the limb-girdle, or in not the limb-girdle space, in the gene therapy space, and Jesse Gelsinger died. Many people, I'm sure, remember that. And so I share that to say that gene therapy has absolutely come a long way, and science has come a long way, and the limb-girdle community has been on the forefront of that for a very long time. And we're going to have a conversation. We're very honored to have you both here. But the gentlemen here have helped us overcome some very serious obstacles and challenges, as have many of the scientific leaders in this room, all of whom were probably in the space, many of whom were in the space in 1999 at the time. But so many of those catalyzing moments that helped us get from point A to wherever we are, point L, point W, were catalyzed by patients who really helped us figure out what were the questions that had to be answered. And so as I listened to those presentations just a little while ago, and people talk about what matters, what changes would really make an impact on my life, those are the moments that will catalyze change. And so I thank you for being here and for sharing that. And I really feel like today is a really important inflection point for limb-girdle muscular dystrophy. So thank you for convening this meeting and for including us all in this. So with that, we're going to dig into a really wonderful conversation. And I thank Dr. Peter Marks and Dr. Peter Stein for being here with us. I'm going to do just a super quick introduction so we can dig into the conversation. Dr. Peter Marks serves as the director of the Center for Biologics Evaluation and Research, and we refer to that as CBER at FDA, or the Food and Drug Administration. He started his career as an attending, as a clinician at Brigham and Women's Women's Hospital in Boston, and eventually served as the clinical director of hematology. He then moved on to work for several years in the pharmaceutical industry on the clinical development of hematology and oncology products prior to returning to academic medicine at Yale, where he led the Adult Leukemia Service and served as the Chief Clinical Officer. And then he joined the FDA in 2012 as the Deputy Center Director for CBER and became the Center Director in 2016. Dr. Peter Stein serves as the Director of the Center for Drug Evaluation and Research, Office of New Drugs. So we have representatives from both CBER and CDER here, which is actually very rare to have both centers represented. Dr. Stein began his career in internal medicine and in endocrinology and metabolism at Yale University and Yale New Haven Hospital. He's a nationally recognized leader in pharmaceutical research and development. He joined CDER in 2016 as the O&D Deputy Director. Before coming to FDA, he served as Vice President for Late-Stage Development, Diabetes, and Endocrinology at Merck. And he's also served as the Vice President of Head of Metabolism Development at Janssen. So we have clinicians, therapeutic developers, and regulators here with us. So thank you for being here with us. And most importantly, a very patient-centered approach to care. So we've had a lot of conversations throughout the morning that have really focused on outcomes that matter to the patients, regulatory pathways that might help us get to answers that will improve outcomes for patients. And we'd like to spend the majority of today talking about limb-girdle specifically, but in rare disease, we learn from one another, and we learn from what's happening in our sister ecosystems. And 2023 was really a landmark year for rare disease and clinical trial designs, the application of the accelerated approval pathway, the use of novel surrogates, which we've heard a little bit about this morning. And we saw strides and milestones that we had never seen before in rare disease. So could you each maybe start off by reflecting a little bit on any learnings or insights from these programs that might be important for those in the room to think about as we think about our limb-girdle space?
You know, I can start by saying it was quite a year this past year. We had a number of gene therapy approvals where we've kind of gotten to critical mass now. Each year we've had more and more. We now have, depending on how you count them, 17 or 18 approved gene therapies. I know that sounds funny, but it depends on whether you count one product that has two separate indications as one or two. You know, with 17, let's just say 17 products now, we've gained some experience. I think gene therapy, has shown itself in this, especially in this rare disease space, to have certain qualities that make it a little different from protein therapeutics and small molecules that are just that are important to distinguish, in some ways that make it a little easier on the development pathway. And these are things that we sometimes didn't leverage as much as we possibly could have previously, and we're coming to realize we can. And I'll just say two of these specifically. First of all, in the manufacturing world, many of the gene therapies use the same backbone vector over and over. You know, it's the different product, but same backbone vector of adeno-associated virus often. And as it turns out, we can hopefully start to expedite development some by starting to rely on kind of a platform approach where people don't have to constantly recharacterize a vector that's already been characterized with toxicology studies. And if they're using the same manufacturing technology for two different products, have to describe that again. So we're hoping to decrease some of the burden on that side of things. The other piece, though, that really I think has come into focus, you already mentioned it, is making use of accelerated approval. And gene therapies really do have something unique that, having worked in small molecules and protein therapeutics, they have something that you sometimes see in protein therapeutics, you don't see in small molecules, which is you can measure if everything goes well. You can't always do it, but you can often measure the gene product and have a good sense that something is likely working, because if you can measure the product in the tissue that's affected, and you can correlate that type of replacement with either an animal model or a human situation where getting to those levels lead to normal function, you have a way to use our accelerated approval provisions with something reasonably likely to predict. Still want to get clinical evidence at the end of the day, But those two things together, I think, combine to help us think that in the coming years, and we're hoping to really make some progress in the next year or two, there are quite a few rare disease gene therapies that, using that combination of helping reduce the manufacturing burden and help getting them across the finish line with accelerated approval, will hopefully have any number more products for relatively small populations of patients. We're talking about dozens to maybe a few hundred make it across the finish line.
That's great. I'm going to come back to platforms again in a little bit because there were some questions around that earlier, so we'll come back to that. You're hitting on all the themes that came up earlier today.
Yeah, well, first of all, it's a pleasure to be here. And just to step back for a second, I think these type of meetings which reflect the commitment of the community, of the patients and families, is critical because, as we're going to talk about natural history studies and understanding how one designs trials and understanding what endpoints matter requires a committed community. It's really the first step towards trying to get successful therapies, successful programs that can move forward. Without a committed community, without patients who just have a passion for getting to an answer that can be helpful to them, you really can't go anywhere. And the stories that you tell are stories that we can leverage and think about as we're thinking about, like, how do we design our trials and how do we figure out what endpoints matter to you that makes sense to put into trials. So it makes such a big difference to hear those stories, and of course that's why we have the patient-focused drug development sessions, but sessions like this also telling us about your experience of the disease, what kind of treatments you're looking for, really do make a very big difference. So just thank you for pulling together and being here. Of course, we prefer, if we can, to focus on exactly that, which are the design of trials that answer the clinical question: does this drug provide the benefit that the patients are looking for? Does we have an endpoint that reflects patients' burden, their experience of disease, and we can show that this drug provides a meaningful benefit to patients? So while we certainly are interested in accelerated approval, I know we're going to talk more about that. The starting point is, and our default for that reason, is a trial that looks at endpoints that are about how patients feel and function. Is it improving patients' lives, and can we directly measure that? But we recognize that, clearly there are big challenges there. Often these are small populations in some types of limb-girdle muscular dystrophy, maybe small numbers of patients. And so running clinical trials focused on clinical endpoints can be challenging. We're going to talk about ways that one can do that and how those can be informed, but they are challenging. And not only that, they can take many years to have enough patients in the study and to see the changes that have to be seen to demonstrate the benefit of the drug. And so while we focus on trials that look towards traditional approval with clinical endpoints, accelerated approval is a tool which we critically need and have to look to. But it's also a challenging tool. I mean, we have to recognize that what we're looking for, in particular in the area of a disease like limb-girdle muscular dystrophies, are biomarkers that become surrogates. And that can be a big challenge because we're going to rely on that endpoint, that measurement, to tell us that we can expect clinical benefit. That's the uncertainty that we accept with accelerated approval. We say, okay, we can look at this endpoint of this surrogate. Does this protein go up or down? And can we then expect an outcome? But the data to support that is a challenge. I mean, it really is a challenge because the last thing we want to do is approve a drug because it increased some protein level, but turned out not actually to make a difference in patients' lives. It didn't really reflect clinical benefit. So the challenge is really developing the evidence, the support for that endpoint, that surrogate, and being able to have confidence, not assurance, because we accept some uncertainty, but have some confidence that it will predict benefit and that we can ultimately show that benefit. And that's quite a challenge. And the way I look at this is it's something that we haven't, I think the companies and academic academics haven't engaged with sufficiently. I really think the problem we face now is that these come to us often without the kind of background research and development and evidence that gives us the confidence we need to be able to utilize them. So I think, as Dr. Marks said, when you have a pathway where you're using a gene therapy and increasing a protein, and you can show that protein went up, that may be sort of a lower hanging fruit, if you will. I don't want to suggest it's easier, but a lower hanging fruit. But when you have a drug that has a sort of complex role in the pathogenesis of the disease, the biomarker, the surrogate, the thing that we're looking at to predict outcome may not have such a clear relationship to the benefit that we're looking for. And then we need to really dig into the science, the animal models, the epidemiologic information, experience from many other settings, basic science work that really can give us that confidence. So accelerated approval is an incredibly useful tool, but it's only useful when we can get the evidence that we need to utilize the endpoints that we can use to approve drugs using accelerated approval, and that's the challenge.
Could I ask you both then to reflect on, and I appreciate that complexity. I'll also stir in, and I appreciate that if you're an individual living with limb-girdle, what I'm going to say next is not going to feel accurate. But in limb-girdle, especially within the different subpopulations, we're talking about small populations, and comparatively from a regulatory perspective, probably slowly progressive diseases. And so you recently, Dr. Stein, helped author, led the authorship of a guidance that may provide useful here and have some really important lessons for us to draw from, which was FDA's guidance on clinical evidence of efficacy that does talk about other flexibilities that FDA has that would have applicability potentially to limb-girdle. So could you maybe both talk about some potentially other principles of clinical trial design that we could be thinking about within the limb-girdle community as this community thinks about how to move some maybe trial designs forward?
I mean, maybe it just goes to start with just saying, I think just building off something that Peter said before, I mean, having patient communities get together and discuss and really understand what matters most in the disease really is important, because the time, I hope this doesn't stir anyone up too much. The time for people to get involved, patient groups to get involved in drug development is very early on, with helping to understand what endpoints are important for those drugs to be, or biologics to be developed. It's not when like we have an advisory committee meeting and, you know, people, I mean, it's true, people jump up and down, et cetera. The work is over then. And that's when, you know, we should be judging things dispassionately. But the time to get involved, I think, really is early on to understand what really matters, because I've witnessed drug development programs where, you know, somebody developed, you know, to try to get to a given endpoint, and that endpoint didn't actually matter to patients. For one genetic disease that, you know, that causes cardiac arrhythmias, it turns out that the doctors were like all excited to try to suppress the arrhythmias with a gene therapy, right? So that people didn't have sudden death. But the patients and their parents said, I don't care about, you know, being, you know, sudden death is bad, but we care about the fact that the major problem that they experience is incredible fatigue day after day, right? So address the fatigue, and you know, that's what they were more interested in actually seeing measured as an endpoint. Not that, you know, and actually in some ways it's an easier endpoint to measure. And now using other technologies, you can deal with the arrhythmia endpoint as well. But it's just a point being that understanding what matters is really important. I think patients are the ones where that needs to come from. Doctors, we don't always, you know, we have what's important to us, and it may not align always with what patients experience.
You know, I couldn't agree more. That understanding the patient's perspective has been such a transition in drug development. I mean, I, like Peter, have been in drug development and or regulation for many, many years. I started maybe almost 30 years ago. And I can tell you, when we started, the idea that the patient would inform us as to what was relevant was sort of, that would be quite novel, right? And now that's what we all are absolutely committed to. How would one think about designing a trial, not thinking about whether the endpoint made any difference? In terms of flexibility, I mean, interestingly, the regulations, right from decades ago, already gave us a lot of flexibility. We talk about the reliance on an adequate and well-controlled trial, and I'm not going to bore you with all the criteria for an adequate and well-controlled trial, but suffice it to say that there's a lot of different designs for an adequate and well-controlled trial. The one that we recognize is the most robust and precise is a placebo-controlled trial, and we do rely on those, and they are important, and we can talk about that. But actually, there are many other types of designs. Active control trials can be adequate and well-controlled, and we can rely on those. The externally controlled or historically controlled trials can be a form of an adequate and well-controlled trial. And there are other forms of it. Dose range trials can be adequate and well-controlled. So just to say that even going back decades, we did have a fair amount of flexibility, and we've used it more and more. But even going back 20 years ago, there were approvals based upon externally controlled trials. I think the other thing that comes out of the guidance is a recognition that in rare diseases, we need to accept more uncertainty. And we can, if we're going to approve a drug for a disease that is, like type 2 diabetes or hypertension, affecting millions and millions of people, the level of uncertainty that we're going to accept is going to be exceedingly small. And that's because the tools are there to get to that point. We can run trials with thousands of people in type 2 diabetes. I did that for many years. And so we can get to a level of precision that there's virtually no uncertainty that the drug has the effect we expect it to have. We can't do that in rare diseases. We can't design trials that give us that level of precision. So we, on the regulatory side, have to accept some degree of uncertainty. And that's part of also what patients need to tell us. How much risk are they willing to take, both with regard to the safety burden, but also to this issue of uncertainty. Now, that said, we also recognize the harm of approving drugs that aren't effective. It's not just that we're risking, and this is the primary harm, we're risking a patient taking a drug that has only risk and no benefit, but it also means that that area is going to seem to be fulfilled, and other companies that might be doing research might not come into that area. So we recognize that we have to consider these trade-offs. We have to meet the unmet need for these serious diseases like limb-girdle muscular dystrophy. We have to be flexible because of the limitations of what we can find out in the context of clinical trials or a clinical development program. And we need to recognize the harms of approving drugs that are not effective, but also recognizing the need to fulfill an unmet need. So those are all the considerations. And I think the patient voice in helping us understand patient perspectives on the safety issues and the acceptance of some uncertainty is really very important. But the guidance, I think, tells us that we can accept some uncertainty, but it's just a matter of sort of how much uncertainty and how much flexibility we can apply.
So let's maybe follow that thread a little bit. Let's talk a little bit about the concept of totality of evidence, if we can. So when a disease is rare, endpoints are almost always going to be novel. So let's maybe dig into some maybe the tools that we have in rare disease. We at the Every Life Foundation recently had a researcher, G.K. Raju, who's done work in oncology with FDA and done work now in rare disease, did an assessment of the outcome measures and endpoints that are being utilized in infectious diseases and oncology and in rare diseases, and found that there was really a striking contrast between the number of times endpoints have been utilized in oncology and in non-infectious, non-oncologic rare diseases, which is not a surprise. And so recently there's just been a lot of discussion in the rare disease community around the importance of secondary endpoints in regulatory use. And so can you talk a little bit about the need for the totality of evidence and how that can be used?
So this is a great question. When I was at J.P. Morgan a couple of weeks ago, I got this question in a variety of ways. We talk about primary and secondary endpoints, and I think I want to just back up and say that rare disease, this issue of being able to tolerate uncertainty is an important one that means that, and it actually does back up and at least for me, have an impact on what we have to do with our statistical constructs. So when we're developing a drug for type 2 diabetes, we tend to, because we tolerate so much less uncertainty, we tend to actually require strict statistical constructs where there is a hypothesis that's tested, and generally if something doesn't meet its primary hypothesis, we get pretty upset. And that also is an issue that has to do also with the fact that we're sometimes working with molecules that are small molecules, or they're drugs that act indirectly, not directly, as some of the gene therapies. Gene therapy is a little different because you're not always, because sometimes we do have to use surrogates in any number of diseases. But if you can measure what you're replacing or what you're knocking down, if there's too much of something, you have a handle on the science behind this in a way that allows you that if you do miss a primary endpoint but get all your secondary endpoints, you might feel, look, especially if you can understand why a primary endpoint was missed, you might say, oh, that's okay. When can that happen? There are any number of composite essentially scores that have been developed, and sometimes the composite doesn't actually reflect what the benefit of the drug actually is in the population, or the gene therapy. And so I think we are very willing to look and say, hey, does everything make sense here? Does the science make sense? Is, the gene products getting made? There are real positive clinical effects that seem to be occurring. The surrogates seem to all be in the right place. And yeah, if it fails its primary endpoint, it's not great, but we can live with that and try to get the data, perhaps if we give an accelerated approval on the back end. We wouldn't do that probably for a diabetes drug or heart failure drug, but we have the latitude to do it here, and we're going to have to if we're going to make the kind of progress that we'd like to. I think the difference is, and I might as well get fired now. The difference is we can probably, the amount of uncertainty that we can accept for populations in the hundreds of thousands is very small. And so you'd like to only once in a blue moon, 1 in 1,000 or certainly less than 1 in 100, but 1 in 1,000 or somewhere in there, have made a mistake and have to pull something. On the other hand, in this rare disease space, in the next couple of years with gene therapies, if a couple things aren't confirming out, a couple in 100, it probably means we're not leaning in hard enough. And remember that things in confirmatory trials can fail for 2 reasons. It's because the product doesn't work, or because the trial was not designed properly to capture the effect. So I think we can tolerate that uncertainty from a public health perspective. Again, we'll do it very thoughtfully, and it will be done always, at least at our center, with safety first and foremost, because we can't go back and have bad things happen, because it doesn't take too many bad events to occur until people just don't want to use something. And trust me, some of what I've seen in the vaccine world over the past few years, where it doesn't take much to get people to get worried about using something, we just don't want to have that happen with our gene therapy. So safety will always be front and foremost, but with that, once you have that, we probably are willing to take a little more risk with effectiveness.
I think we look at it exactly the same way. I want to just comment
Your mic might be off.
Is that better?
Yes,
wow,
it
okay.
is.
Wow.
I saw everyone leaning in.
Sorry, let me start from the first thing I said. So I do want to come back to this concept of totality of evidence. because it's a concept that we both embrace as well as reject in certain areas. So as we think about what we use to approve a drug, we think about substantial evidence of effectiveness, not totality of evidence. And that means that we're looking at an adequate, well-controlled trial, and I said, the kind of flexibility we have there, and either two adequate, well-controlled trials, which is sort of our traditional approach, or one adequate and controlled trial and what we call confirmatory evidence, and we can come back to that at some point. And that's what constitutes substantial evidence of effect, not sort of a totality of evidence. However, and I think this is exactly where Peter was going, is where we can use a totality of evidence approach is in interpreting whether we do have a supportive, adequate, well-controlled trial. And even if the primary endpoint fails, there may be enough evidence within that trial to still support that the trial did support the effectiveness of the drug. However, when we see a failed primary endpoint, we're starting with a level of concern and suspicion, and so it requires a lot of very careful analysis and consideration, and that's where totality of evidence comes in. Let me give you an example. We have a meeting within CDER where we bring sort of challenging problems to discuss each week, and rare disease programs are frequently discussed in that forum. And we had a situation where a primary endpoint failed, and when we looked at the primary endpoint, it was just poorly constructed. It wasn't that the drug wasn't showing efficacy on the primary endpoint. It was a lousy primary endpoint. It was not the way to have designed a primary endpoint. We looked at all of the secondary endpoints, which were very meaningful. Patient report of how they were feeling, their physician's report of how they thought their patients were doing, other scales that were used were strongly supportive. And there was a unanimous agreement that the evidence from that trial was very compelling. So it is a totality of evidence in a careful, thoughtful way that we apply. Now, the same thing applies to whether a surrogate is fit for purpose or not. We recognize that developing a surrogate requires basic science information, epidemiologic information that can glean from clinical trials, from the bench research that's done, and we put all of that together in a totality of evidence or a weight of evidence approach. So we do use that concept in very specific ways, but the criteria and the approach we take to deciding about whether there's substantial evidence is a little bit more discrete, and we don't in that situation say, hey, there's this little trial over there and that little trial over there and this little piece of evidence and that little piece of evidence. So hey, in toto, that's good enough. That's not the approach we take. We need to see an adequate, well-controlled trial and additional evidence that is substantive, that really meets our bar for substantial evidence. So I just wanted to make that distinction.
No, that's great. So you answered the question of where the PROs and the COAs fall in that equation. Where would you say, or maybe not to put you on the record, but maybe how are patient preference studies proving?
There's a record here?
There's no record. But how are patient preference studies proving helpful to date? So are they lending themselves helpful in clinical trial design? Are they providing useful further downstream in regulatory? Where do they provide context that's been beneficial?
Well, what I'd say is that, and we have one of our experts here on these kinds of studies. You'll hear more about this, and I may have to look at Michelle.
She's taking notes, by the way.
Yeah, I may look at Michelle. So if she cringes at my answer, I'll know I didn't quite get it right. But what I'd say is, rather than sort of put it under the category of patient preference studies, what I would say is that the information that we get that builds the PRO in the first place is based upon a detailed and extensive process that harvests patients' input. I mean, it starts with, and I think people are probably familiar with this, it goes through a very rigorous process, talking to patients about, just in an interview type format, what are the things that matter to you? Talking to multiple different patients from different aspects of, different manifestations of the disease, different stages of the disease, pulling all that together, and then working further and further to develop the instruments that reflect exactly those factors that matter. So I think in that way, the patient input is the primary way that we build the endpoint in at least clinical, where we're looking at a clinical endpoint that we use in clinical trials. So patient preference a bit different, but patient input is the sine qua non of building endpoints that are relevant in clinical trials.
So you were talking about confirmatory evidence, and I don't know whether you put this in that category or not, but I think we all would hope you would. We saw this morning, Dr. Johnson gave a presentation and talked about, I believe there were 9 or 7 natural history studies right now underway or being supported in the limb-girdle community. And we've seen this year natural history studies become very important in some regulatory decisions in the rare disease space, which I think has really added more wind to the sails of rare disease communities as they look at how the natural history studies are being funded and designed and what core data elements are being pulled into them. So could you both talk a little bit about the natural history studies, how you're thinking about them? I know we have some data scientists in the room that are really investing and spending a lot of time on these studies.
Yeah, so at least for us, I think like everything else that we do, what we collect should be fit for purpose. I think in some settings, natural history studies are very helpful. In others, they're less so. When there's tremendous variability so that it's, you have so much noise, we have to find some other hook to figure out how we're going to get confirmatory evidence other than natural history studies. On the other hand, when it doesn't have to be homogeneous, but when there's less variability, these can be incredibly helpful, because then with that natural history study alone, without having to have concurrent controls, if you have a gene therapy that shows you're just off, because many of the gene therapies, the beautiful thing about gene therapies is unlike, I've worked on drugs where the oncology drug, the difference was, a difference in 5% in response rate at 3 months. And that's what those are the kinds, and those are meaningful. But for gene therapies, we're often seeing like on-off thing, alive, dead, or able to walk, not able to walk, able to sit up, not able to sit up. And so if we have a natural history, especially in a disease where we're not going to normally see improvement with a gene therapy, where we're going to see stabilization, it's just very helpful for us to have those natural history studies, because then when a study's conducted, it's nice, again, for small populations, it's very hard to get people to want to get randomized to in a gene therapy trial. And oftentimes, you wouldn't be able to randomize enough people to deal with the noise inherent in interpatient variability. So these natural history studies can be really important. Again, fit for purpose. I try not to say that every rare disease group may not want to go out and conduct natural history studies because they may not be able to get data that's helpful. But when you can get data where there's some reasonable consistency, it can be tremendously helpful for us in the gene therapy space in facilitating these single-arm gene therapy studies.
Yeah, I think that really applies very well to the drug space as well. With respect to confirmatory evidence, I think the role is a little bit less there, but there is a role in the sense that if you have an adequate, well-controlled trial, a natural history study can inform us as to whether the observations in that trial are consistent with other observations about the natural history of that disease. But of course, the natural history studies have so many other functions, as Peter mentioned, potentially as controls for a single-arm trial. And certainly in the space in gene therapy where you might expect a large effect, that is a sweet spot for a natural history comparison. Less so where we may be dealing with a drug that has a meaningful effect. It might have a 10 or 15% change in the course of a disease, which could be very meaningful, but is very, very hard to detect when you have a noisy external control. And a lot of that is about the quality of the collection of that data, if it's very carefully collected, and if a lot of the variables that might change that natural history or affect the natural history are collected so we can better understand and control that variability and match it to the patient population in the single arm trial, it can become useful. It does require really high quality data, carefully collected, with careful analysis. What we often see are natural history cohorts that were never intended for this purpose. And so a lot of things that we would need to know to match the population in the trial we're looking at. They just weren't collected. The endpoints weren't collected in the same way. The diagnosis wasn't done in the same way. The staging wasn't. So that we just don't know how to compare those things. But if it started early, and if particularly in communities like this where there's a commitment to really building resources to develop and support trial development, program development, then you put together a natural history study where right from the start you've collected the right things and potentially had discussions with the FDA division that may be involved, or with experts who really understand natural history studies. If the right things are collected in the right intervals, it can have huge value. But again, when that's very noisy or disease has very variable courses that are not terribly predictable, even in that setting, it could have limitations.
So we've talked a little bit, we've touched on the accelerated approval pathway a little bit at the beginning. And I know we'll talk about that a little bit later this afternoon. And I am sure, like most rare disease communities, that is absolutely a pathway that the Limb-Girdle community is hoping they will identify surrogates that are able to be utilized in clinical trials. The statistics that come from FDA and those that have analyzed the use of the pathway show that benefit is ultimately confirmed about 90% of the time, the products that go through the accelerated approval pathway. And in instances when products that have been approved through the accelerated approval pathway do not confirm benefit, then they are withdrawn. And we actually saw that happen just last week. There was a very noteworthy product that had been approved through the accelerated approval pathway, not for a rare disease, that was withdrawn from the market. The industry partner has withdrawn that, the marketing of that product, which is how this is supposed to be working. It opened up a field for that patient community. In a congressional caucus briefing for the Rare Disease Caucus, a former FDA Cardiology Review Office Director, Dr. Ellis Unger, made a statement that really resonated with many of us, and he suggested that there should be a 70% chance for accelerated approval drugs to confirm their benefit as an appropriate benchmark, and instead the agency comes closer to 30%, is what he had said. And so but what you've talked a little bit about is you've referenced what I will say type 1 versus type 2 error. You've talked a little bit about that here. Can you talk first for those who don't know what type 2 versus type 1 error is? Can you talk about what that is? And then can you talk about ways that the scientific community can be de-risking decision-making as we're developing data that comes before regulators?
So type 1 and type 2, the issue is the two things you can do in a trial that aren't good is you can say there's an effect when there's not one, or there can be an effect and you don't see it, right? And that's type 1 and type 2 error defined essentially, simply. I think the issue is, and unfortunately when you have very small populations, it's easy to have type 2 error. In other words, it's easier not to find something when there's an effect there. On the other hand, when we make type 1, we really don't like type 1 errors because that leads us to put stuff on the market that sometimes has to be withdrawn. And that's the kind of the natural tension that you see us dealing with in bringing things forward. I think for us, I'm not sure whether those numbers from Ellis, I'm not sure I'm with him on those numbers, but by and large, I'd say most of our accelerated approvals do end up confirming. It's in oncology sometimes where we've had, the most challenges here because they're an area of, it's in some ways a lot like rare diseases, a tremendous medical need where we're willing to take early data and lean on it. Unfortunately, sometimes it doesn't translate into the clinical benefit over the longer term that we would like. So we use surrogates because you have tremendous medical need. And we accept the fact that, in a given space, it's probably 90 to 95% of the stuff works out, but 5 or 10% of the stuff has to come off the market. And again, it doesn't always mean that the drug didn't work. It just sometimes means that the trial was not designed in a way that it could detect the effect. So I think we want to use the accelerated approval pathway whenever it makes sense. I think here, rare disease, tremendous medical need, it has an analogy to what's done in the oncology setting. Especially where you have people losing function that can't regain that function. That analogy is, in LMDG, GD. Sorry, the analogy goes pretty well, I think.
Yeah, I mean, I don't know where Ellis got those numbers from. I'm not sure I would necessarily agree with those numbers or the 30% number. I also wouldn't agree with the 70% number because I don't think there is a number. I think that's not a good way of thinking about it. Let me just explain what I mean. If I have a drug that has some pretty serious adverse events that occur not, with some frequency, and I have a drug that I know works and really provides an important benefit, we may approve that drug because we do conclude that the benefit still exceeds the risk, and that's informed by what patients tell us, right? Mm-hmm. Now, in that drug, I'm not going to take a 30% chance that the drug doesn't work when it has serious side effects. That would not be very likely. Let me give you another example. A drug that is a pill versus a drug which, let's say, is administered through an intraventricular bolt. Am I going to approve a drug with a 30% likelihood and put that in a pediatric population without confidence the drug provides benefit? No, and and we have to hear what the community thinks as well, but you can't say 70%. What you have to say is, what is the drug? How is it administered? What is the risks of that drug? And then let's figure out what level of uncertainty we can all accept. But it can't just be sort of a one-size-fits-all acceptance of risk, because that just doesn't make clinical sense. So Ellis was a brilliant regulator, brilliant guy, tremendous respect for him, but I think saying 70% is nonsense.
I mean, something in the rare space that I think I'm coming to appreciate is each disease entity, and it's probably even true in limb-girdle, different subtypes. It's its own fingerprint, and it may require us to take a slightly different approach to get over the finish line. I think we can accept that. In some cases, we may have a low-hanging fruit as a surrogate marker for an accelerated approval. In other cases, there may be a clinical endpoint that there's no reason to do an accelerated approval because, heck, you can give it a traditional approval right out the starting gate. So I think it requires us to look at each entity in a way that we care enough about the disease that we understand the disease, we understand what the therapy is doing, and we use our regulatory toolbox and regulatory flexibility appropriately to do the best by that individual disease. And I do think of them a little bit as their own unique, fingerprints.
But which just builds the case for making sure that there is natural history data and
that's exactly right, because every hook you can have.
That's
I think that Peter's already kind of alluding to it. You need for these small entities, you want every possible. It's like you want to have every possible screwdriver in the toolkit, and that so that you can adjust to what you need to get something over the finish line.
That's great. So speaking of the finish line, I'm getting the wave. So I'll just end with a question for both of you to reflect on. So this is for those who don't live in the White Oak room or don't work in White Oak. There's a lot of really exciting transition taking place right now. So FDA has announced its largest reorg in history is undergoing. Maybe that's not so exciting, actually, if you work there, but that's happening. And just last week, one of our heroes in the rare disease space, Dr. Janet Woodcock, served her last day at FDA. So there's just a lot of transition happening. But as I started off talking about 2023 was really a landmark year in rare disease with the numbers and types of approvals that went through and the implementation of the user fee agreements, which had some really important implications for our rare disease community. And so for us in rare, this is a time to be very cautiously optimistic as we lean in and continue to work with our partners in the regulatory setting. But it would be wonderful if you could talk about what you're each optimistic about as you look into the rest of 2024.
I'll let you go first this time.
I think I'm optimistic about the sort of our increasing effort in this area. I mean, we started the ARC program now more than a year ago, and it's really expanded. It's providing, I think, important internal education, external outreach, and really collaboration across our organizations to really help us think about and apply these tools in as I'd say aggressive a way as we can, but also to develop new tools. I mean, I'm really excited about opportunities that we have to do a better job working externally and working within our organization on enhancing what I call translational science, the early development of drugs and models, but also to improve how we can work on analyzing and designing small trials for patients in these rare disease settings where large trials that we've done for years and years just don't apply. So I think there's a lot of progress that we are making and can make in this space that will make the development of these drugs more efficient with clearer answers. We want to get to an answer that the drug works or doesn't work. And we've seen too many gray zone answers where the data is just not really clear one way or the other. I think we're going to see progress in that area substantially.
And I'll try to keep it brief. I agree. I think for us, I think we're really optimistic that we can accelerate progress. I think part of that, I'm optimistic. I think one of the things Janet Woodcock leaves behind, she had a very large impact on some of us in how we think. I think one of the things she kind of impressed me with was how important it is to build teams that work well together. I think I feel very good that we finally have a team in our center, particularly in our Office of Therapeutic Products, that's very much focused on really trying to focus on the patient, move things forward. And so I really look forward to trying to accelerate progress over the coming year.
That's great. Well, on behalf of all of us here and those watching the stream, we thank you very much for your leadership and all you're doing on behalf of our communities. So thank you.
Thank you. Tough act to follow. So my name's James, and my background's in genetics and statistics, and for the past 9 years I've been looking at natural history data and trial data in Duchenne to try to learn from it in ways that can help us improve drug development. And I'm really grateful for the opportunity to be here today and talk to all of you about what we might be able to do in Limb-Girdle. So kind of picking up from the discussion of accelerated approval. So certainly for accelerated approval, we need that biomarker that's reasonably likely to predict benefit. But we also need to have a confirmatory trial that's designed that gives everyone confidence that we can confirm that clinical benefit somehow down the road. So I'm going to focus today on how do we design those trials and what might be some tactics that could help in Limb-Girdle. And so of course by clinical benefit, we mean how the patient feels, functions, survives. Confirmatory study needs to be adequate and well controlled. We talked a lot today about the challenges in Limb-Girdle. I'm not going to belabor those. But we're going to talk through 6 different tactics that have relevance for Limb-Girdle. And we're really going to focus on what are the benefits and challenges and what could we do next to see if this approach will work. And if it does, great. If not, we want to find that out quickly so we can focus on other things. Randomized controlled trials. It's good to start there, gold standard. They have their limitations. And again, I think we all understand what those limitations are. But there's room to improve, especially by learning from natural history. And I'd like to draw some focus to one area, actually, that Joshua called out before lunch, which is how do we use these prognostic factors that we're learning about to make these trials better? And if you look at all the decisions drug developers need to make when designing a randomized trial, there's a lot of them that actually rest pretty heavily on prognostic factors. And it turns out that the stronger the prognostic factors we have, the better we can predict outcomes, the better we can make all those decisions. And by better, I mean we can make the trials more efficient, more power, shorter duration, fewer patients. And this is very well covered within FDA guidance. This guidance was actually released mid-2023, but I think it's reporting on something that statisticians have known for decades. And I think what this guidance is saying to me is, hey, you guys aren't doing this enough, but it can help and it doesn't have much risk. So I can't emphasize enough the importance of really using strong prognostic factors to design trials, right? The question Joshua asked is, if we know there's different subtypes with different progression rates, can we do something to make sure those are balanced in randomization? And yes, we can, through stratification. And if we're going to go to the trouble to do that, let's use the strongest prognostic factors that we have. So we've learned something in Duchenne that might have relevance for limb-girdle, of course, different diseases, but I feel like we were in a similar place in Duchenne where we had some knowledge of things that were prognostic, probably age is predictive, maybe the baseline measure of your motor function score, steroids, and there are all kinds of different cut points and rules for looking at these individually. But what we found is that it's really about putting them together. They're synergistic. So if you look at the bars here, the height is how much variability in motor function outcomes we're explaining. So higher is better. And the bar sitting there at 14.9 is adjusting for some stuff that kind of makes sense: genotype, class, age, baseline motor function. But we found that when we add multiple measures of motor function together, so not just this North Star score, but also time function tests, you get more than doubled explained variation. This goes right to the bottom line in clinical trial design. The more variation you can explain using baseline characteristics, the less noise you have to contend with in the outcomes to detect signal for drug effect. So I think for limb-girdle, trying to see what can we learn from natural history to find the best composite prognostic factors that we can, and then actually using those in trial design can help. We should say a few words about adaptive designs. I think they can be very helpful in rare diseases, but they have tremendous practical challenges. The helpful part is that when we're uncertain about our outcomes and about how what rate of change might occur over time, adaptive approaches help us manage that uncertainty, but they are logistically harder, and there's still a randomized trial requiring placebo exposure. And to do them well, we need to learn from natural history. Platform trials have come up today as well. I think the appeal here is tremendous for limb-girdle, just because there's so many subtypes. It was very encouraging to hear that many of these subtypes actually have very similar rates of change in motor function. That gives us some hope that we'd be able to pool these together into a platform trial where, of course, the goal there is to be more efficient in the use of a limited placebo set and be able to move more, kind of increase the throughput of testing for therapies. But there's still this challenge of how you handle multiple subtypes. They might be similar, but what if they're a little different? What if that causes bias? Since many of the treatments are targeted at a particular subtype or even a particular genotype, is it really fair to compare treated patients of one genotype to a mix of patients with another genotype? That's a challenge we've also looked at in Duchenne. And basically what we found is that if genotype effects are small or subtype effects are small, and maybe they're not small over a lifetime, but if they're small over the scale of a clinical trial, So if they have a small effect on like 1 year change, after you've already lined up everyone at baseline and adjusted for their functional status, if the genotype's effects are small over that follow-up period relative to the size of treatment effects, then you can do mixed genotype or mixed subtype control groups. And you can learn from natural history in a way to account for that genotype or subtype mix. So we can have confidence it's not causing bias. That tactic might be valuable here as well, and it seemed there was a wealth of natural history where we could learn this information. There was some discussion previously about composite endpoints and multiple endpoint analyses. I think the real attraction for these is when we have a lot of endpoints that we know they're all kind of getting at the same thing, like underlying motor function. They all have intrinsic clinical meaning. Of course, we need to quantify it and make sure we know what clinical meaningful change looks like on each of those endpoints. But if we're in a situation where we don't really know which one's going to perform best or be most sensitive to the drug effect in a trial, then maybe we don't have to take that bet. There are ways of kind of quantifying the totality of the evidence across different endpoints and pooling together the signal to have more power to detect drug effect than possibly any of those endpoints alone. The catch is you're also pooling noise, so there's also a risk that your composite could do worse than the best endpoint. So to sort this out, what we really need, what sponsors would typically do are simulations to kind of game this out, plug in what they expect about their specific treatment and mechanism of action. What do they think the effects are going to be on these different endpoints? And then marry up those assumptions with high quality natural history data that tells us how variable are these endpoints, how correlated are they with each other, how is the noise going to add up, and are we going to get more signal from noise by pulling across endpoints than by looking at them individually. Okay, external controls. So this is one that it seems really important to dig into. There's such a wealth of data that's been collected for limb-girdle, and it seems like there's questions about whether and how that could be used for external controls. And I'll just acknowledge that there's a lot of use cases for external controls. It could be, there's a single arm trial and you're lining up the external controls over a 1 year outcome period. I think a lot of the use cases will be longer term. Maybe we've got an accelerated approval, but now we're going to follow that first cohort of treated patients for 3, 4, 5, 6 years. We're not going to have a placebo that long. So we need a way of comparing them to some cohort to give us confidence, confirmatory evidence of clinical benefit. So of course the challenge is these are not randomized. So there's many things that could go wrong, many sources of bias. There could be differences in the populations, especially in baseline prognostic factors. The outcome measures, this is going to be, I think, the biggest challenge for limb-girdle. Ideally, we're looking at objective assessments, things like survival or tumor response. In limb-girdle, a lot of our endpoints are performance outcomes. And so we'll come back to that, but I think that's one of the challenges is that they could depend on effort or motivation, so it's very challenging when we don't have blinding. There's also legitimate concerns about differences in background standards of care that could vary across care centers, geographies, years. As I think Peter Marks was saying before, if there's a lot of variability that you can't explain in the outcomes, that makes it harder to do external controls. And a lot of this is going to come down to knowledge of important prognostic factors, right? We heard that we want the disease course to be predictable. Well, that means we have to know what the prognostic factors are and how good they are. So I think in Duchenne, we've got part of the way there. There's a really important piece missing that I'm going to focus on next, but I think there have been some really important takeaways from natural history. So what you're seeing is a figure where we took many different natural history sources, 7 data sources in total, spanning multiple years, multiple countries, and we asked a simple question: do one-year motor function outcomes differ between patients in clinical trial placebo arms and patients in natural history databases? And the figure on the left is just straight up comparison, and we saw that there was a little bit of a difference. But importantly, once we accounted for known prognostic factors, that difference pretty much went away. And this is very consistent even when we break out by individual data sources, break out by year, and these sources are different, but the outcomes are behaving similarly. So that gives us confidence that the training and all the effort that went into making these outcome measures consistently collected has really paid off, and that they are behaving similarly, at least in a placebo arm setting for a clinical trial versus natural history. But when we talk about external controls, we're not just comparing placebo arms, of course, we're comparing treatment arms. And I think this is where the most significant challenge comes in, and that challenge is open label bias. So for these performance-based outcomes or motor function outcomes, it's very reasonable, legitimate to be concerned about motivational biases. Sometimes bias can sound is kind of a loaded term, right? The bias can be unintentional. If the patients know they're on treatment, the assessors know they're on treatment, bias can creep in how these outcomes are assessed. And the prospect of that causes significant concern, because if you think about the direction of that bias, if people are more motivated or have more hope for improvement because they know they're on therapy, that would lead to bias that could make an ineffective drug look effective, even though it isn't. So I think this is one of the kind of limiting concerns to use of external controls. So I think given the wealth of natural history data that's out there, it's really important that we think carefully about this and take this concern seriously and think, is there a scientific way that we can address it? And so, one thought is, what if we could measure this open label bias? If we could, then we could account for it in an externally controlled trial, right? So maybe that top blue bar is what you're seeing in the treatment group. But if we had from external experiments or other data, we can get to how would we know this? But if we could measure that extent of open label bias, then we could kind of subtract it off. And that's kind of a penalty applied to the treatment arm because we think there's this risk of bias. It might take us a little longer to follow patients for the treatment effect to get big enough that we have confidence that it's real. But this could help us get past the concern about open label bias just being unbounded. And it could make us more scientific and rigorous about what do we mean a dramatic treatment effect? How do we really know when we've crossed that bar so we can make sure we're crossing it as early as possible and we can have confidence without waiting too long. So this is also a challenge, of course, for self-controls, right? Self-controls are very intuitive, very appealing. We follow the patient before they're on treatment, then we see what happens after they're on treatment. If there's a change in trajectory, we'd like to infer that that's due to treatment. But of course, those patients would also know that they're going on to treatment and would be subject to this open label bias. So again, if we can measure it, maybe we can account for it. So I think some worthwhile questions for us as a group are, first, let's suppose we could measure this open label bias in Limb-Girdle. And then we could do these externally controlled or self-controlled studies with performance outcomes and account for that bias. Would that be adequate for confirming clinical benefit? And let's assume everything else is taken care of, right? We've adjusted for the right prognostic factors. We're not worried about backgrounds. Everything else for external controls is taken care of. We're honing in on this open label bias issue. If we could measure it and account for it, would that be adequate? And then second, based on clinical insight, do we think we can measure this, and do we think there's a ceiling? Is there an upper limit on how much motivation could improve function? I mean, it's a progressive disease. It can't be cured by hope or motivation. Otherwise, it would probably be cured by now. And I think limb-girdle and these other progressive diseases are different from some of the examples where open label bias has really caused a lot of trouble. One of those examples that's been cited in previous briefing documents quite effectively, I think, is from coronary atherosclerosis with severe angina. Patients got this transmyocardial laser revascularization, and they did great on the treadmill, even for like a whole year after therapy. But in a randomized trial, no effect at all. But limb-girdle seems different than that disease. And I think, especially for the physiotherapists who see these patients, who understand how these tests work, it would be really great to hear their insights on, is there a ceiling? How could we measure it? How can we quantify it? And we have a lot of endpoints to choose from, too. Maybe some of them are more prone to this open label bias than others. That would be useful to know as well, as we think about whether and how we could use the wealth of natural history data that's been collected over many years to help. So I think Keith will smile if I keep this to 20 minutes. So closing remarks, we should do all we can to understand prognostic factors. They're going to help in so many ways. I think we have the data. It might just be a matter of looking at it the right way. What we found in other muscular dystrophies is like pooling together different measures of motor function really helps a lot. I think these are all kind of noisy measures of the same thing. So maybe you put them together, you're getting kind of a sharper picture of where the patient is on their disease trajectory and therefore where they're going. This is exactly the type of helpful thing to do with natural history studies. More data is always better. If there's ways to share or federate data across all the studies on that very nice slide we saw earlier, that would probably help here. Assessing consistency of these clinical outcomes. How do they look across different data sources, geographies, time periods? Are they really sensitive to standards of care, or are they pretty robust? That's something we also need to understand to see if the path for external controls is workable. Continuing to quantify natural history, especially across subtypes, especially looking at how these endpoints associate with each other. Things that might seem boring and quantitative, but that's exactly what drug developers need to do all the simulations and analyses they need to optimize their clinical trial designs. And then finally, if we could quantify open label bias and really work together to understand if external controls could be viable with a quantified measure of open label bias, I think that would be a significant area of progress. So thank you again, and I'd also like to thank all of my collaborators in the CTAP Consortium for wrestling with these issues over many years. Thank you.
Thank you so much. Next up, I'd like to introduce you for presentation number 6, entitled Clinical Endpoint Considerations by Dr. Lindsay Alfano from Nationwide Children's Center for Gene Therapy.
I'm sorry. Okay, so let me just start by saying clinical endpoint considerations is a broad topic that I will not fully cover, despite what everyone said earlier today, in 20 minutes. But I think a lot of the themes that I'm going to be discussing have already been brought up, so I get to kind of weave it all together, which is a fun thing. So first, great news is we have endpoints. As we've heard today, we have a variety. This is not an exhaustive list by any means. So what I wanted to highlight here is that we have endpoints and we have a variety of types of endpoints that have been developed hearing through natural history studies, hearing from patients, hearing your voice telling us what's easy, what's hard, what's important to you. So we have these performance outcomes that have been alluded to. I think the NSAID has probably highest word count of the day. So this assessment was developed and adapted from the NSAA, which we've also heard about in Duchenne muscular dystrophy, and it grades functionally relevant items on a 0 to 2 scale. So it includes some bed mobility, so rolling, which is nice. It's got some upper extremity items. It's got standing, walking, getting up out of a chair, some of these transfers that people are talking about that are really challenging, as well as getting up off of the floor. There's also time tests embedded within it, so we do get some of those continuous scale variables as well. There are other performance outcomes that we hear about varying frequencies within the neuromuscular space. Again, this is not all of them, but the performance of upper limb is a primary upper extremity assessment designed to kind of mimic some of those upper extremity tasks, like bringing a cup to your mouth, moving things across your workspace at work or at home, lifting things off a table. Again, to get an idea of functionally relevant scales that are reliably and consistently assessed so that we can measure change over time in clinical trials. So it's trying to take what's meaningful at home, at work, and put it in a clinical trial. We also have a variety of patient-reported measures that are in various stages of validation. They all seem to be working in some patient populations, which is great that we have options and we're not just kind of pigeonholed into one endpoint. These ordinal scale variables have pros and cons when it comes to measuring change over time. There's also continuous scale variables, which you get more incremental differences, but the question is, what's that meaningful change? So we have ambulatory tests. We have more functional tests, which do tend to be more transfer related. It's hard to come up with time tests of upper extremity function, so we've tried in various capacities to do like taking a shirt on and off or putting a coat on and off. We do that in other disease groups, but it does tend to be a pretty variable outcome. So we haven't mastered this one yet, so we would love to continue to hear from patients so that we can identify these time tests that are meaningful and based on functional skills that are really important. There's also a lot of work in technology-based outcomes. We continue to learn. I think that's my favorite part of kind of working in rare disease and in natural history studies and clinical trial readiness is there is plenty of room to improve. And I think we're all trying to work together to identify even more robust scales, continue to validate our robustly scales that are developed with rash analyses and have great psychometric properties to make sure that they're working in and across various subtypes so we don't have to reinvent the wheel. But when that wheel's not working, we have the opportunity to do that. So with that, the NSAD was one of the first assessments that was validated specifically for limb-girdle type muscular dystrophies, which was really exciting. This was a hugely collaborative effort. We've also started taking assessments that are validated in other neuromuscular disorders to apply to this limb-girdle space. So the performance of upper limb I mentioned earlier, 100-meter time test. We've been trying this instead of some of those more self-selected walking speeds. We geek out on the details of these assessments because they all measure people slightly differently. They measure different constructs, which are important in different patient groups. And so we're really committed to kind of getting that right and knowing as much as we can before that trial comes, as we heard has been echoed throughout the talks today. So in pulling some data from one of the studies running through the GRASP Consortium, we're finding that a variety of our assessments are really reliable from day 1, day 2, which means that how you perform on day 1 is exactly what you do on day 2, which gets a little bit to some of that motivation and performance bias that James was talking about earlier. We like to see some consistency in the group, which are ICCs above 0.9 for everything but the 9-hole peg test, which we'll just drop that one off. That one's not our favorite, but the rest look great. And again, we look at this at a group level as well as an individual level. So what we want to understand is, do we see relatively no change between a group? That's the goal. But then also understand what's impacting maybe individual patient variability. So if you look at that change column, If you look at this change column over here, you can see some of the range of changes can vary on some of these quite dramatically in individual patients. And when we talk to patients about this difference, some of that's related to the travel burden of coming to our site. This is not your home, you've had to travel a long distance. Some of that's related to just learning and comfort, being in this setting with people that you don't really know and trying things that maybe you haven't tried for a while. So understanding what that day-to-day variability can be really helps us understand that longitudinal trajectory. But on the whole, these are really reliable assessments. However, there's always a caveat. One of my favorite things that I've learned from Dr. Mendell over the years is that we always have this linear trial plan. We tried to plan for all variables. We're going to go from point A to point B. We're going to find out if this treatment works. But in reality, we end up going on quite an adventure. We learn a lot of things along the way. And then we can apply those learnings to the next trial. So hopefully we won't have quite as many deviations from our path the next time. So natural history studies really set us up for success and help us get there faster. So this will look familiar to many of you in the room. This is one of my favorite graphs because this epitomizes rare disease. So we have rare disorders. No center has this many patients, but if we work together and collaborate, we bring together these huge data sets that then allow us to see the heterogeneity in our patient groups. And then as scientists, because we need to have a number to compare point A to point B, we have to draw kind of a line in the middle of that huge graph so that we can say, at a group level, are we seeing changes over time? And really no one falls along that line. It doesn't really address an individual patient experience, but it's in the middle. Grossly, this is what we see. And so to get to this level of information is hard as we get into rarer and rarer disorders. Understanding the differences in these patients, our data-driven trial design is dependent on who enrolls in our natural history studies. So we only know what we know. So if there's a group of patients that because of some unknown bias we all have that we're not seeing, but that could benefit from a clinical trial, we can't plan a trial that will tell us if a treatment is effective for them. Now, that being said, the gray box, because we have a large enough data set, can then be color-coded into different patient trajectories. So this is where we want to get. We want to get as much data together as we can so we understand, those in the red group are different than those in the blue and the green, and we can plan a trial that addresses all of that through stratification of all of these factors at baseline. To points that have already kind of been alluded to, the variability in standards of care, access to services regionally are really important. So a lot of our consortia tend to be more regionally based. And so we can make really data-driven decisions for that group, but trying to generalize that across the globe can be really challenging. And then as we continue to make progress, there's more awareness of disease, people are getting diagnosed younger. We also have changes in care and more proactive models of care, which then in turn can impact that rate of disease progression and what the disease looked like. So there's kind of this cyclical pattern of learning. So I won't harp on this because we've heard about all of the clinical trial readiness activities that are underway. They are kind of regional. We do have some alignment on COA in the current studies, which is really helpful, I think, for this future kind of pooling, federating data sets. But they are really focused on the more prevalent subtypes as they are kind of clinical trial readiness and where potential treatments may be targeting these groups initially. So what do we know? We're learning from everything in the rare disease space and hopefully continuing to progress and accelerate our knowledge faster, but we know that these diseases are progressive. So it doesn't matter which group I pull data from, the lines go in one direction. And I think that's a theme we've heard today. So whether that's FKRP related LGMD R9, I'm gonna merge a bunch of nomenclatures at this point, and whether it's from the group in the EU and UK or the Iowa group or our GRASP consortium, we can see that over time, these disorders lead to progressive weakness that impacts function. In our own internal NCH studies, we have a CalPAIN cohort. We see whether we're looking at gross motor function or upper extremity function, we see those declines over the age range. Our psychoglycan beta group, we see the same. And then looking at data from the COST study and dysferlinopathy, we know what this progression looks like. So it's good from a patient perspective. You are telling your teams, our natural history studies are informing this. We can then use that for our data-driven trial design. But what does that look like in ultra-rare subtypes? So this is data from our group and the only patients we knew of, 3 patients with LGMD R18. We can take learnings from other subtypes, but we are going to get to these more rare and ultra-rare patient cohorts, and they're just as important. We need to be kind of thinking proactively about how we can apply our learnings from these larger groups to these more rare groups, and also build awareness, elevate the standard of care, etc. So heterogeneity is a theme we've heard as well today. Our natural history and clinical trial readiness studies really help us understand differences in these subtypes. What we can say is the same and what we can say is different is important. So we see in these graphs that our patients that have calpainopathy have earlier shoulder girdle weakness compared to lower extremity when you're comparing them to patients with dysferlinopathy. That's useful information, especially as you're selecting outcomes for a trial. We know that upper extremity, if you're enrolling younger patients, is going to be more meaningful in patients with calpainopathy than maybe that dysferlinopathy cohort right away. We've also heard a little bit about MRI as an outcome. So this was just presented by Carla Bolano-Diaz with the Newcastle team looking at MRI across various limb-girdle muscular dystrophies, in which even muscle involvement is different across the different subtypes. Again, the more we know, the more we can predict. We can stratify clinical trial inclusion based on these characteristics. And it's colorful, so great for a slide. Again, we've talked about this genotype contributing to this variability. The more we know about this, the more we can plan. And what it's really helpful to see is that we have kind of the same sort of results across different groups. So we can see that those patients that have compound heterozygous mutations do tend to have that earlier onset and different, maybe not trajectory, but earlier onset of decline than we would see in the homozygous cohort across GRASP Consortium and the Iowa group. And I think this graph gets most frequently shown today, so woohoo. There should be prizes at the end. So now we're getting to trials. So we've talked a little bit about those primary endpoints. We've talked about totality of evidence, which I won't say anymore, overall evidence that's providing substantial evidence of effect. But I pulled some data from our GRASP Consortium to look at all of the outcomes that we're testing. And while it's great that we can see changes across the cohort over time, I think it's also useful to kind of visualize which of these outcomes is actually informative in this population. So this is particularly in LGMD R9 or FKRP related. So we can see 33 patients, so about 37% of our total cohort were non-ambulatory. So having ambulatory assessments as a primary clinical outcome is not going to be informative for that patient population. But we do see that more patients were able to complete the NSAD. We have less of a floor effect, a little bit of a ceiling effect, because it was a little bit easy for some patients. But then potentially we can measure those stronger patients here on the 100 meter where we don't have a ceiling effect. Similarly, to continue to bash on the 9-hole peg test, it is not useful in most of our patients and also highly variable. So we have learned not to make that a clinical trial outcome. And what I think is important to realize is when we are selecting our primary efficacy outcome, we are limiting our cohort. So it's affecting enrollment criteria because, again, selecting an ambulatory primary efficacy outcome means that that is not meaningful or useful for a population that's not walking. It does also impact the length of a trial. So what I don't have here is, how responsive to change these are, but the quicker we can measure change, the shorter a trial can be. But also generalizability of the clinical trial findings across the broad cohort. So if we're only measuring a change in ambulatory function, that doesn't mean that that drug's not effective for patients who don't walk. It just means we haven't measured that. It impacts the meaningfulness of findings if we're selecting, as they mentioned today, if we picked the 9-hole peg test as our primary efficacy outcome, and our treatment improves that, great, but that's not really meaningful for anyone. And then we want to make sure that once we get to this disease-modifying treatment that's been approved, we want to make sure the broadest swath of patients has access to it. So that can be impacted by those efficacy outcomes that are selected. So again, meaningfulness of endpoints, we've talked a lot about this today. Are there certain outcomes that are just inherently more meaningful? I'd say meaningfulness of these outcomes changes across disease progression. So what's meaningful for you today will be different than 3 years from now and was 5 years from now. I'd also like to controversially say that really all endpoints are surrogates for a patient experience. Of course, as a physical therapist, I'm biased, so our functional outcomes are closer to real function, but we're still trying to create the patient experience within the clinic environment or even within the home. Some of the, wearable or camera-based systems are still taking your particular experience and making it as consistent and reliably administered so that we can measure change over time, which does make it more of a surrogate of your daily life. We see the others that we'll hear about today are also surrogates, but I think, we acknowledge that endpoints, we have a lot to improve, but we've also learned a lot, and I do think we have, we know that LGMD is progressive, and every point and second on these scales means something and is related to true daily life, so every second endpoint is meaningful. When you look at the NSAD, for example, 1 point is the difference between being able to roll over in bed or not, or being able to bring your arm up to your shoulder to pull up your covers, or being able to stand up out of a chair versus not, or being able to get up off the floor versus not. And those are, patients have told me, are very meaningful functional tasks. So to conclude in my 2 minutes, we do have an endpoint toolbox that are at various levels of being validated across LGMDs. Many of them are quite psychometrically robust scales, which is great, and gives us the opportunity to understand what constructs they're measuring, how sensitive they are to change over time. We have meaningful time tests related primarily to gross motor function, but we have plenty of room to continue to learn and develop and validate new endpoints. And that's where I empower the patient community to continue to use your voice and tell us what's meaningful and what we're missing. The individual patient heterogeneity can complicate trial design, as I think we've also heard about throughout today. So if we have, really broad enrollment criteria, if you're birth through 99, any stage of kind of that disease progression, it's really hard to select the best or optimal outcome. But if we pick an outcome that's, rising from a chair, that you're going to have incredibly strict enrollment criteria for that trial, it's going to be really hard to enroll in a rare disorder where maybe 10% of the population can achieve that task. And then at the end of the day, how do those results really generalize across the patient population? So we're not going to consider the totality of evidence, but the substantial evidence of efficacy across rare and ultra-rare disorders. There's no best outcome that's meaningful. We do our best to gather as much information as we can in these clinical trial readiness studies to anticipate every pothole and hurdle we may encounter, but we're continuing to learn. But we do have outcomes that are meaningful to everyone. And so I think pulling that together will allow us to improve our clinical trial design, maximize enrollment, and get to approved treatments at the end of the day, because these outcomes are meaningful and every point really does matter. And with that, I just want to say thank you to all of the patients and families and everyone related that has participated in natural history and clinical trial readiness studies, because I don't think it's said enough that we really do learn so much from you being in the clinic. The cost to patients of being in natural history trials is immense. You're not getting anything by being in these trials other than you get FaceTime with our lovely team. We get to hear from you. You get that input, which is huge, but it also puts, all of these challenges in your face, which we totally understand is hard. And so we really, really, really appreciate it and hope you continue to participate. With that, I will pause No questions.
Thank you so much, Lindsay. And next up, we have the surrogate endpoints in LGMD, the subtype case studies. I'd like to welcome Dr. Douglas Sproul, CMO of ML Biosolutions, and Louise Verdino-Klapak of Sarepta Therapeutics. She's the Executive Vice President and Chief Scientific Officer. Thanks.
I think I get to go first. So. We're going to do it this way, I believe. Let me get the clicker. All right, so my name is Doug Sproul. I'm Chief Medical Officer at ML Biosolutions, and I want to thank the organizers for giving me the opportunity to speak this morning on behalf of ML Biosolutions. I will provide a case study of a potential surrogate endpoint for use in LGMD2I. It's also in the new nomenclature that was discussed this morning, referred to as R9. I'll use those terms interchangeably for the reasons that we discussed. This is a disclaimer. As a disclaimer, BBP-418, which I'll talk about a little bit, has not been approved for use in any kind of condition in any jurisdiction, including the United States. So I'm going to talk, obviously, about our biomarker. And to frame my remarks, I want to define what a surrogate endpoint is. It's a marker or laboratory measure or physical sign that is used in therapeutic trials as a substitute for a clinically meaningful endpoint that is a direct measure of how a patient feels, functions, or survives, and is expected to predict the effect of therapy. There's three criteria that can be thought of as critical in describing an effective surrogate endpoint. The first is biological plausibility. The second is the reliability of measurement of that endpoint. And the third is its association with clinically meaningful outcomes, as was just discussed. So I'm going to discuss the use of glycosylated alpha-dystroglycan, or ADG, as a potential surrogate endpoint for LGMD2I. So Dr. Matthews eloquently described this disease earlier, and I'm not going to get into it, obviously, in any depth, given her remarks already. But I would remind the audience that LGMD2I is caused by a loss-of-function mutation of a gene called FKRP, which is an enzyme critical in the glycosylation of alpha-dystroglycan. Improper glycosylation of alpha-dystroglycan leads to membrane instability, injury, and ultimately development of muscular dystrophy. So Lindsay pointed out this has already been used once, twice, now 3 times. But the point of this, from my perspective, is to emphasize an important point. You know, existing clinical measures are unfortunately unable to really capture disease progression in LGMD2I over the relatively short periods of time that are typically used in conventional clinical trials. So as this work from Iowa and other sources have shown, there is a marked decline in deterioration, an accumulating decline in function that's seen over time in patients affected by this disease. And that can be clearly demonstrated over longer term intervals. But over the course of short intervals, such as a year, which is more typical for a conventional pharmaceutical trial, the measured decline is relatively modest and certainly is subject to challenges with regard to the noise and variability of a measure on an individual date, which challenges the effective study of potentially beneficial therapies. And so in LGMD2I, a surrogate endpoint may be able to measure changes in an earlier time frame that would enable shorter trial durations and more rapid development of potential therapies. So I'm going to talk first about the 3 criteria for an effective surrogate endpoint, starting with biological plausibility. And as you know, as I've described, and as has been discussed more eloquently by others already, defective FKRP enzyme activity leads to reduced levels of glycosylated alpha-dystroglycan in muscle cells, which in turn leads to impaired stability of those muscle cells and ultimately injury and cell turnover. And the left side of this figure shows what happens in the untreated disease state. On the right side is a proposed therapeutic mechanism of BBP-418, and while the enzyme is dysfunctional, as Dr. Matthews mentioned, at least some enzyme activity is obligatory for life. And so that residual enzyme activity can be harnessed by providing supraphysiologic levels of the substrate for that enzyme. And if sufficient, this approach could lead to an improved or increased amount of glycosylated alpha-dystroglycan that, if sufficient, could slow or halt the ongoing injury to the muscle cell with resulting translation to clinical benefit. This busy slide shows some of the preclinical data. Ribitol treatment in the LGMD2I mouse model led to increased glycosylated alpha-dystroglycan that was measured both by immunohistochemistry as well as Western blot and was associated with improvement in functional measures used in those animal experiments. So on the left side, you can see the improved expression of glycosylated alpha-dystroglycan. You can see that in the middle column compared with the left column in the untreated animals. On the surface of myocytes. In the center, we see increased glycosylated alpha-dystroglycan levels in treated animals as measured using Western blot. And on the right, we see an improvement in functional measures, total treadmill running distance and running time that are used in assessments in mice. So this sort of establishes the premise that underlies this approach. A second key criteria is obviously the reliability and the ability to consistently measure a given surrogate. And ML Biosolutions has developed and validated a Western blot-based assay that measures glycosylated alpha-dystroglycan from tibialis anterior muscle biopsies obtained by a needle core biopsy. We use a calibration curve that was developed from healthy donor tissues to provide a reliable, sensitive assay with a greater dynamic range than a typical classical Western blot. And this allows, importantly, the measurement of very low levels of expression of glycosylated alpha-dystroglycan, which we see particularly in patients who have non-L276I homozygous genotypes, the patients with compound pathogenic heterozygous genotypes for whom expression can be quite low. And this assay has been shown in our studies to have robust performance and an acceptable coefficient of variance, which sets the stage for its use in some of our early clinical work. And so this figure shows some of the experience in treated patients. On the left, we see what happens using this assay in the context of patients treated with BBP-418. The leftmost column is the untreated baseline state, in which you can appreciate is that green streaking and a very minimal amount of yellow, which indicates the marked reduction in glycosylated alpha-dystroglycan in that patient prior to treatment. In the middle, which is following 3 months, in the right column, which is following 6 months of treatment with BBP-418, you can see that increased yellow signal indicative of an increased amount of fully glycosylated alpha-dystroglycan. This yellow signal corresponds with the functional laminin binding form of glycosylated alpha-dystroglycan, and the antibody is the same, binds to the segment of the matriglycan segment that binds to the laminin chain. And so in some ways, this assay could be considered a functional assay from that perspective, given its role in the cell. We can also look at the effect of glycosylation on the protein binding pattern, which we see that streaking green on the left seems to be coalescing around a tighter yellow band with treatment. A third, and obviously probably the most critical pillar of any surrogate, is the association with clinically meaningful outcomes. Can the impact as measured using a surrogate endpoint predict clinical benefit? So in the context of our clinical studies and those of other sponsors in the space, the effect of a therapy can be assessed across the complete range of pathology, albeit imperfectly. First, one can measure glycosylated alpha-dystroglycan, which is indicative of pathology at the cellular level. Secondary markers such as creatine kinase and troponin can be used to assess injury at the level of the organ, muscle and the heart, respectively. And the effect on function can be measured using validated clinical endpoints and PRO tools and other measures. So this data was generated from the GRASP Consortium-led natural history study. And obviously I'd like to take the moment on the podium to recognize all the patients, investigators, and staff that contributed to this important work. Amongst untreated patients followed as part of that natural history, we observed that glycosylated alpha-dystroglycan levels reflected the known genotypic and phenotypic distribution seen in this disease. So in the forest green on the left are patients homozygous for L276I. It's a group of patients who typically present somewhat later with a less severe and rapid progression when compared with patients with other genotypes, which is shown in the lime green. And mirroring that known differentiation in the field, we see that patients who are homozygous for L276I have approximately 10% of glycosylated alpha-dystroglycan levels compared with a reference standard, compared with approximately 5% in patients with other genotypes compared with that reference standard. We also see early evidence from our open-label phase 2 study that suggests that glycosylated alpha-dystroglycan levels in muscle is responsive to treatment with BBP-418. And on the left, we see a rise in glycosylated alpha-dystroglycan levels in the L276I homozygotes, again, that darker green, from a median of approximately 16% to approximately 40% following that sustained over the 15 months of observation. Similarly, we see a rise in patients with other genotypes from approximately 6% of that reference standard to approximately 10%. We are also seeing a translation to the organ as a whole. We see a more than 75% decrease in creatine kinase, which indicates that at least on the cellular level, there's a reduction in ongoing muscle cell damage, which suggests an improved stability of the muscle cell membrane. We're also, and this is, obviously interested in seeing how myocyte stability would translate to clinical outcomes. And while this data set is small, it's open label, and obviously subject to those sort of biases. We are observing in our phase 2 study the changes in glycosylated alpha-dystroglycan levels seen at 3 months preceded and were associated with subsequent changes in clinical measures. And this is the 100-meter time test as an example. So we see evidence of that association, which is shown on the left. And on the right, we see the trajectory in treated patients, which is shown in that red dashed line. The darker blue line is the trajectory seen in patients who were followed untreated in the GRASP-led natural history study. So in summary, I've presented glycosylated alpha-dystroglycan levels as measured from tibialis anterior muscle biopsy samples as a potential surrogate endpoint in LGMD2I. I described the biological plausibility of this approach, noting that impaired glycosylated alpha-dystroglycan is on the direct causal pathway in LGMD2I. Our assay directly measures the functional form of glycosylated alpha-dystroglycan. I described the reliability of measurement of the fluorescence Western blot assay to measure glycosylated alpha-dystroglycan. This is a validated assay with robust performance across patients and studies. And I've described some of the early data that suggests that an association with clinical outcomes could be pursued. Glycosylated alpha-dystroglycan levels reflect the known genotype and phenotype distribution seen in populations affected by this disease, with lower levels in more severely affected non-L276I homozygotes. Additionally, glycosylated alpha-dystroglycan levels increased with BBP-418 administration, and these increases were associated with decreases in creatine kinase and improvement in clinical measures. So with that, I thank the organizers. I want to particularly thank the patient community across the limb-girdle muscular dystrophies for putting on this amazing conference, and particularly the patients and their families who have contributed so much to this work and the work across the field. So with that, I hand the baton to my esteemed colleague.
I have to move this way down. Okay. Just wait for that to come up. There we go. So I'd like to thank the conference organizers for giving me the opportunity to speak today on surrogate endpoints as well. I've been working and researching the field of LGMD for nearly 20 years. The first 3/4 of that was in the academic space at Nationwide Children's, and the last almost 6 at Sarepta. And so it's wonderful to see the field at this stage and to see so many stakeholders in this room: patients, caregivers, academia, industry, FDA. So it's really wonderful to be part of this. Thank you. Here are forward-looking statements. These can be read in detail at our website if you so choose. So today I'm going to be talking about LGMD2E or R4, which is caused by deficiency of beta-sarcoglycan, either a complete absence or severe reduction. And I'm using it as a case study. And so everything that I speak to today will apply to our broader platform of limb-girdles, of which we have a total of 6, and I'll talk about those throughout the presentation. So as we've heard today, the sarcoglycanopathies are a subset of LGMDs. When we look at sarcoglycanopathies in total, they affect 1 in 178,000 people worldwide. And so that accounts for about 15 to 20% of all limb-girdle cases. We see an age of onset from 1 to 15 years of age, loss of ambulation typically between 12 and 14. Respiratory involvement is very common, and cardiac symptoms are also common, but vary by subtype. We also see an elevation of CK, as we've heard earlier today. And so now looking at limb-girdle muscular dystrophy type 2E or R4, it's a very ultra-rare form of the disease. It's heterogeneous and rapidly progressing. If we look at the natural history, age of onset typically occurs around 6 years of age. 90% of patients are in the range of 1 to 14 years in terms of age of onset. As far as diagnosis, it varies by country. We've seen an acceleration of diagnosis over the past 20 years. Many of you in this room have helped do that acceleration. Unfortunately, molecular subtyping can still take long, and approximately 50% of cases are still successfully typed. In terms of loss of ambulation, this typically occurs by the 12th year. Again, we see a range in this as well. We see a steady loss of activities of daily living. As we heard, cardiac and respiratory complications are often the leading cause of death, which is obviously a premature death around the age of 30. Now, as we've heard, it's challenging to run a traditional trial for an ultra-rare disease such as LGMD2E. And what we're showing here is an illustrated example. When you start with a small population of 600 and you apply criteria to that, meaning only a number of those patients have a genetic diagnosis that can be confirmed. You have to look at the subpopulation that's amenable to treatment, able to meet the inclusion-exclusion criteria of a trial. And then in gene therapy, we have an added difficulty in that approximately 15% of patients have pre-existing antibodies to the gene therapy vector that we're using in this case, which is AAV-RH74. And so after you put the entire population through this funnel, that leaves you with a small number, in this case about less than 18, available for a clinical trial. And so doing a traditional clinical trial is just too difficult in these ultra-rare populations. And so where does that leave us? Obviously, limb-girdle muscular type 2E has significant unmet need. There's no one else working on this specific subtype. And we need to find a treatment, and we need to find options. And so what does this require? This requires us to think not just outside the box, but like there is no box. We're just throwing the box out in the recycling bin. And so biomarkers are an essential role in advancing therapies intended to treat these ultra-rare diseases. And so Apologies, but I'm going to read the definition of a biomarker just to make sure that we are level set. It's a naturally occurring molecule, gene, or characteristics by which a particular pathological or physiological process or disease can be identified. Provides objective indications of a patient which can be measured accurately and reproducibly, as we recently heard. So in the case of LGMD2E and all of the other limb-girdle subtypes that we're working on, of which we have a total of 6, beta-sarcoglycan is serving as both the surrogate endpoint and the therapeutic agent. So we're actually delivering the full-length beta-sarcoglycan gene. It's naturally occurring and provides objective data that can be measured accurately and reproducibly. And so let me just give you a little bit more detail about how we're delivering the gene for LGMD2E. The product, which is not approved, is called SRP-9003, and we're using this to deliver the instructions for making beta-sarcoglycan. So we're delivering the beta-sarcoglycan gene under the control of a muscle-specific promoter called MHCK7 and delivering that with an AAV vector called AAV-RH74. And in the absence of beta-sarcoglycan, as we've heard today, this sarcoglycan complex, which you can see highlighted, does not form. So not only do you see a reduction in beta-sarcoglycan, but you see reduction in all of the sarcoglycans. So restoration of beta-sarcoglycan is important for improving and stabilizing that entire complex, which I'll get to in a minute. And so starting to identify the preclinical evidence of surrogacy, I'll go through the preclinical evidence followed by the clinical evidence because it starts with the preclinical studies. And so we've done a number of studies to test SRP-9003, which I'll call it from here on out, to test how that expresses the protein and then how that leads to downstream biological function. And so this is a single study looking at a systemic dose in both low and high dose of SRP-9003. And this is a complicated diagram of what it shows is that we not only restore beta-sarcoglycan, but we restore the rest of the sarcoglycans. So just walking from left to right in the first column, and each column you'll see wild type on the top. So this is a normal control mouse. Underneath that, you see the beta-sarcoglycan knockout mouse where you don't see any expression of beta-sarcoglycan. And then you see animals from the low and high dose. And so not only are we restoring beta-sarcoglycan, but then in each subsequent column are the other sarcoglycans. So all of those are actually restored to the membrane following delivery. And we can quantify this, which is shown on the right. So you can see a dose-dependent restoration of that complex, which is a composite of all of those sarcoglycan proteins being restored. And so that restoration of the protein expression on the membrane leads to improvements in muscle health. And we look at this in a number of different ways. And so the first is looking at fibrosis. As we know, when you get muscle deterioration, you get infiltration by fat and fibrosis. And so the images on the left are a staining called picrocereus red, which stains for collagen. So in green, you see healthy muscle, and then in pink, you're seeing this fibrosis. So on the left, again, we're starting with wild type. In the middle, we're seeing the knockout mouse, so you can see a lot of infiltration by fibrosis. And on the right, in both skeletal muscle on the top and in the diaphragm, we're seeing an improvement in the muscle health. And then on the right, this is simply quantified. We use a unbiased computer algorithm to quantify the amount of fibrosis in the tissue. And so this improvement in fibrosis and overall muscle health also protects the membrane and leads to a reduction in creatine kinase following intravenous delivery. So as we've heard today, creatine kinase is an enzyme that's leaked into the serum upon muscle damage. You can see in the knockout mouse in the middle there in purple is significantly elevated, and following treatment you see a significant reduction in the amount of CK. And so finally, all these improvements that we're seeing in muscle health also lead to improvements in function in these mice. And so what we're seeing is two different assays. On the left, this is a specific force assay where we simply measure the force coming out of the muscle. Again, in the middle, you see the knockout mouse is significantly reduced compared to normal, whereas the treated is significantly improved and no different than wild type. On the right is a fatigue assay where we simply constantly stimulate the muscle and then measure the force over time. And again, here we can then see a significant improvement in that assay in mice treated with SRP-9003. And so how does this then translate clinically? Because in order to validate the beta-sarcoglycan expression as a surrogate, we need to see how this translates. And so our clinical development plan in LGMD2E, or beta-sarcoglycanopathy, is characterized by 4 different trials. The first trial, our Study 101, which I'll show you data for after this, is an open-label study. Patients will be followed for a total of 7 years. 6 patients were enrolled, 3 patients in each cohort, low dose and high dose. We've recently published the 2-year study in Nature Medicine, which I'll show you some of the data today, but we're continuing to follow these patients. The next study, called Journey, is our natural history study in which we're studying LGMD2E, 2D, and 2C, or the sarcoglycans. And we're using this study to obviously characterize natural history over time, and then also acts as a lead-in study to our gene therapy studies. Our Voyaging study, the next study, our 102 study, this is also an open label study. In this case, we looked at a larger age range as well as ambulatory and non-ambulatory patients, looking at all across the population to show safety and benefit across a wide-ranging population as well. And then finally, our study, which is Emerging, which is our phase 3 study, which we just began enrolling. This is a 5-year single-arm study, open-label study. There's a 6-month run-in. And this is in approximately 15 patients. We just began screening, and this is a global study. So now showing the evidence of surrogacy across our studies. So in Study 101, as I mentioned, we looked at 2 doses. It's for a total of 6 patients. And here we're seeing is expression of beta-sarcoglycan at the membrane post-gene therapy. So we're showing both pre and post in all patients in both the high dose on the bottom and in the low dose. We quantified this expression as we did preclinically, and we also quantified the amount of restoration of the sarcoglycan complex. So on the left is a graph of cohort 1, the low dose. On the right is the high dose. And for each of these, I'll just walk you through it because it's complicated. So on beta-sarcoglycan is the left 3 columns, so baseline is purple, 60 days is in blue, and 2 years is in gold. So we were able to do 2 different post-follow-up biopsies and saw sustained expression over time. And so what you see for each of those sarcoglycans, we see an increase in the assembly of that complex, and it's in a dose-dependent manner. You can see on the high dose on the right, we see significant upregulation of not only beta-sarcoglycan, our surrogate, but then also upregulation of the other sarcoglycan proteins. And so then we next looked at CK as well, and to see how this translated from our preclinical studies. And here again, we saw very good translation and a significant reduction in CK in these patients. So in both cohort 1 and cohort 2, we saw over 90% reduction at day 60. We saw a sustained effect at year 2 in all our patients. And on this graph is what we're seeing. As you can see here, this is individual patients over time. So each patient responded. In year 2, cohort 1, we saw an 83% reduction. In cohort 2, a 67% reduction. So this is sustained over time. So from what we've heard throughout today, long-term follow-up is going to be necessary for these ultra-rare diseases, and we're looking at the accelerated approval pathway to look at a surrogate very on, which is reasonably likely to predict clinical benefit and correlate that with long-term outcomes. It's difficult to correlate a quantitative relationship with beta-sarcoglycan expression with outcome measures in a short amount of time, given the small size of the patient populations and availability for clinical studies. So clinical assessment over longer periods of time, 5 to 7 years, is really the way in which you can contextualize the change, and especially how a patient feels and functions over time. And so we'll enable the functional benefits to assess those over several different domains in our clinical study, which we've done over a longer period of time. So just to summarize a few points, what I think you can appreciate is that LGMD2E is a slowly progressing, low prevalence rate disease caused by a single gene defect. It's very clear that we're treating the root cause of the disease in terms of biologic plausibility. This is a monogenic disease, and it can be addressed by simply inserting the wild type gene and producing the corresponding protein. We've demonstrated the empirical evidence for this surrogate through transduction, expression, localization, and biological function, both preclinically and demonstrating that clinically as well. And so non-clinical extrapolation is important for diseases like LGMD2E and all of the other LGMDs that we're working on in order to determine the likelihood of therapeutic effect. Reasonably likely surrogate markers and accelerated approval with post-approval confirmation is really the viable path forward for these treatment in patients. And so just to put a fine point at it, I think we've quoted the guidance, about 20 times today, but I think it's nice to hear things in multiple ways. And I think the accelerated approval pathway gives us a unique opportunity for patients, which we can't conduct large clinical trials like we can in other indications. Here we're delivering a full-length gene, producing a wild-type protein. Accelerated approval would enable us to get to patients faster, to provide functional benefit, and then monitor that over time in order to achieve traditional approval. And so this is all consistent with everything that we've talked about today. I do want to just highlight the fact that, as I mentioned, this is the tip of the spear. LGMD2E is a pathfinder for us, and all of the limb-girdles that we're working on also use the same vector, and so we can leverage that for other forms of limb-girdle. So I just want to stop and thank everyone here, all the patients and their families who participated in our studies and across, and really this entire field. It's really very much appreciated.
Thank you so much. We're going to have a little quick break now and come back at 2:45, and we'll get started with our clinical and our LGMD endpoints and clinical trial design roundtable discussion. Thank you.
Good afternoon. My name is Keith Flanagan. I'm the moderator of the incredibly giant roundtable. I'm going to do quick fire introductions, a little orientation, and then let Michelle show a couple slides. You've already met Kat Bryant. We have Michelle Campbell here from who's the Associate Director of Stakeholder Engagement and Clinical Outcomes for FDA CDER OND Office of Neurology. We have Joanne Donovan, the CMO of Edgewise. Ang Nguyen, the VP and Therapeutic Sector Leader of Ask Bio. Sophie Olivier, the CMO of Atomio, who has joined us from France and must leave promptly at 5:15. Louise Rodino-Claypack, EVP, Head of R&D and CSO of Sarepta. Laura Ruthenbach, the co-president of the Jane Foundation. Doug Sproul, CMO of ML Bio. Dr. Nicole Verdun, the Office Director of FDA CBER OTP. And Dr. Matt Wickland, Professor of Neurology at the University of Texas at San Antonio. So a quick word just for process orientation. We have an incredibly ambitious agenda, and the next hour and a half, the 10 of us aspire to put all the pieces together. What we're going to do is go through a series of topics: traditional and accelerated approval, surrogate endpoints, opportunities and challenges of placebo and single-arm designs, natural history, totality of the evidence, a couple other issues. The way it will work is I will run through some questions, sometimes directed to someone first, and then we'll have to raise hands if you want to be called on. So it's going to be a little bit of a scrum. If the spirit moves you, please raise your hand. We're trying to squeeze as much as we can in a short amount of time, so we're going to try and be very concise. With that, Michelle Campbell is going to present a couple slides to sort of level set about the regulatory framework.
Thank you, Keith, for that. And I also want to thank, on behalf of myself and my other FDA colleagues that are in the room today and those who are watching online, we want to thank the earlier panel of the patients or the persons with lived experience, their caregivers, and the parents who talked about that. I do know that's a very hard thing. You do not understand how much we really take that information back to us. We incorporate it into our everyday advice that we provide to sponsors when they come to us. So I have a lot of notes and lessons that I've learned from you today that I'll be able to take back to my colleagues and share, and hopefully we'll be able to incorporate what we've learned from you today already into advice that we can give to sponsors. So real quick, as Keith said, I wanted to kind of level set some of the things that we've actually talked about earlier today and what we're going to be spending this panel on. And that is our approval pathways. And I think both Dr. Stein and Dr. Marks really talked about this, these concepts that we're going to share real quick pretty well in their Fireside Chat and really giving you the application piece. But this is to kind of put into terms the framework that we're working with when both for CDER and CBER, we're having to make a regulatory decision and providing advice into drug development programs. So right now we have 2 approval pathways. The first one is known as the traditional pathway. And this is substantial evidence of effectiveness demonstrated on clinically meaningful endpoints, how a patient feels, functions, or survives is where we often talk about, or a validated surrogate. And the drug is safe for use under the conditions prescribed, recommended, or suggested in that proposed labeling. And that labeling is a very critical thing that at the end, if we are to make an affirmative regulatory decision, it's what we are going to inform our clinicians to be able to have that conversation with patients. And so we need to make sure we put all of that information that is critical for that into that label. The second pathway, and this is something that we've been talking a lot about today, is this accelerated approval pathway. It may be considered for serious or life-threatening diseases with an unmet need, substantial evidence of effectiveness demonstrated on an endpoint that is not itself a direct measure of clinical benefit. So it's not that clinical outcome assessment of feel, functions, or survives, but instead are reasonably likely to predict that clinical benefit, so a surrogate or intermediate clinical endpoint. There is a subsequent need, and you have to confirm the clinical benefit is required. And with accelerated approval, and I think this was really well articulated in the Fireside Chat earlier, is that there's the acceptance of some levels of uncertainty for the use of a reasonably likely surrogate. However, substantial evidence of effectiveness for that surrogate or intermediate endpoint is required with the adequate determination of making sure we have that safety information. So while today we've talked a lot about efficacy, safety is also that critical aspect when we're reviewing drugs and gene therapies and cellular-based therapies that we have to also review as well to make sure we can make a decision. So in the surrogate endpoints, there are 2 types. We have the validated surrogate endpoints, and that's an endpoint that's been supported by clear mechanistic rationale of the clinical data and strong evidence that the surrogate endpoint predicts clinical benefit. Examples to think about it, blood pressure. So bars of millimeters of mercury is a surrogate endpoint, hemoglobin A1C. We have science, we have the evidence over time that's been accumulated to demonstrate that those will predict clinical benefit. What we're really talking about, a lot of things that we talk about in the rare disease space are these reasonably likely surrogate endpoints, which is an endpoint supported by clear mechanistic and or epidemiological rationale, but insufficient clinical data to show that it's a validated surrogate. And so this is where we could see these being used for our accelerated approval pathway. Note the best glossaries noted there. I note that because that is a glossary that's a joint effort between NIH and FDA, and we try to align on terminologies with biomarkers and clinical outcome assessments. So this is what we leverage and use. I have it as a resource for you all if you are interested. And really quickly, well, I don't think we're going to go too far into the standards of effectiveness. It was mentioned during the Fireside Chat. So we do have this legal standard that's established for drug approval of substantial evidence of effectiveness. It refers to both the quality and the quantity of the data. And it is defined in the Food and Drugs Cosmetic Act about evidence consisting from adequate and well-defined or well-controlled investigations. And so I think, as Dr. Stein explained it today, you know, we often think about adequate and well-controlled sometimes as those 2 trials, but that we know in our rare disease space that's going to look different, and how we can implement and take and really use that ability to apply what our federal laws tell us we have to for our standards for effectiveness is when we talk about circumstances of one trial and maybe we have some confirmatory evidence that can work. So just setting the landscape, I'm going to turn it back to Keith so we can get this conversation started.
So the first question for the panel is there anyone who's a proponent or a champion of pursuing a traditional approval rather than an accelerated approval? Does anyone think that patients are going to enroll, or that it will be possible to get investors to support the wait for a hard clinical endpoint? Okay.
Hi there. I think what you're saying, a hard clinical endpoint, is different than a clinical endpoint. I just want to, maybe that distinction is, you meant to make that distinction?
Maybe that was a little inflammatory.
Okay, because
I retract the word hard.
okay, because what Lindsay was showing us in terms of being able to look at some of these clinical parameters in these diseases in a selected population, I think is feasible. I think that it all kind of goes down to how much signal do you have? How much noise do you have in that signal? Can we identify a reasonable sized group of patients that would be able to show a clinically meaningful difference in something like the North Star, the NSAD that we are looking at? And I don't think that's totally infeasible. It may be it's like what Louise was talking about, when you're down to a group of 15 patients, but for some of these groups, that the larger groups, it may very well be feasible.
The larger groups. We have a lot of other ground to cover. Does anyone have a comment on?
How do you define that cut point between those two thresholds? You're saying certain diseases too small, certain big enough. What is that threshold? What is that cut point? We have Alzheimer's on one extreme. We have,
I mean, in these kind of diseases with 5,000 patients, with 3,000 patients, can you do a trial with 100 people and see a difference? And I would say that is a reasonable threshold. You can't do that in certain subgroups that are very small.
But it depends on how much effect you're going to see. The larger the effect, the less the numbers you need. So, they didn't need very big numbers for SMA, right? I mean, I don't know all this, but I mean, it was like, because the effect was so strong, right? And I think the challenge here is, are the treatments we're giving, how much effect they're having? And if they're having, especially if you're just slowing progression, and you're not completely stopping progression, then of course your numbers are going to have to be a lot higher in order to be able to see a difference, because your effect isn't as strong. But I mean, eventually you have to show clinical benefit, right? So the thing is, you still have to end up doing the traditional one, but it's just, at least it's a way to get it out to a lot of people with accelerated approval, and then you can do the longer trial without having the placebos and things like that in order to prove the clinical efficacy.
Is it okay if I move on to the next topic?
Sure.
The next topic is how much and what kind of data is needed to support the surrogate. So in a prep call, Dr. Wickland, you were being somewhat playful, but you're like, look, you show protein expression and you're done. There's a lot of reasoning embedded in there. Can you sort of unfold what the thesis is and what the evidence is supporting it?
Yeah, that was a glib but honest answer. So in
most of the recessive LGMDs, there's a paucity or complete lack of the protein product that needs to do what it does. And in preclinical models, if you replace that protein and the protein goes where it's supposed to do and does what it's supposed to do, you restore the phenotype of the wild type or close to the phenotype. And so in my mind, if the challenge is that you're missing a protein because of a gene defect, and then you replace the protein, that should be what you need to do. Now, there are many steps between A and Z in there, and that's why we have people doing all sorts of work along that line. But pretty much if you can show that you've replaced the deficit, you should eventually be able to see some benefit from that.
Does anyone else want to dive in on that? Sophie?
Does it work? Yes.
Yeah, now it is. No.
It's okay. Okay, thank you, Dr. Wicklund, for this comment, but it's particularly relevant for LGMDs with null mutations. Yes, with null mutations, when you have absolutely no protein expressed in the disease, but we have some LGMDs where the protein is expressed already. It's dysfunctional, but it is expressed. So we need also to find other surrogate endpoint, not only based on protein expression, or we need to show that at least the transfection of gene therapy was effective. We can see gene expression, we can see mRNA, but an increase in the protein expression may not be enough.
So I'm going to let people continue to debate this.
I think what you're talking about is in cases where you have expression of a mutant protein, correct? So you have to differentiate between the protein that you're delivering versus the mutant protein. Just want to clarify.
Yes, in this case, sometimes it's possible when your transgene is different from the native gene. Yes, by RNA-scope, we can discriminate the two. But what I wanted to react was the comment that showing protein expression may not be enough all the time.
I think what both of you are saying are not in contrast to one another. They're just two different examples where it's a null versus in a background. But your point's well taken that there are some cases where you have mutant protein, you have to show differentiation between those proteins or addition to it.
I guess I have the question with protein expression, it's about, are you making enough, right? And so I guess my question to the regulators is, how do you prove that what you're making is enough? I mean, is it just in the animal models where you've done dose responses, or, you know, how do you know what's enough to predict a clinical benefit?
I mean, I think it's a good question. For us, it's really a combination of factors. I think showing protein expression, showing that the incorporation of the transgene, and then also I think that we are, and I'm sure you've heard this a lot today, we are really open to doing this a lot of different ways, right? I think that's one of the great things about the accelerated approval pathway is it does provide us some flexibility where it's needed. And I think specifically in muscular dystrophy, this is a place where that flexibility is needed. And so to me, it's really a combination of protein expression, of clinical evidence of clinical benefit. And I do think that, you know, as we just heard in the talk before the panel, there's significant heterogeneity in the disease, which sometimes makes it harder in terms of development. And so in those spaces, biomarkers, we are more than willing to also lean on that data as well. And to have it's really more to me a package approach than it is like one thing versus another, if that makes sense.
Yeah, I would just add on to that from the other center's perspective is that what is also extremely helpful that it needs to be demonstrated is really what is the biological plausibility. So how do we tie that back? And I think someone's last presentation, the biomarker, when you pointed that as that's an important consideration of, why do we think the science is saying this is the right thing? And what do we know about that? And this is where your natural history also comes in handy to help inform that. I think that one challenge that hasn't really been brought up that needs to be thought about when it comes to the use of biomarkers or surrogates to support accelerated approval is what was your assay validation work and that science and validation work that needs to be done to really demonstrate that we actually know what we're measuring. We can truly say that and we can replicate that because there are times in trials that the intent of it was not to be used as the surrogate endpoint, right? It was developed for something else and then based on how the trial happens and reads out, we may now be going down a different conversation pathway when you come in and present to us. And sometimes the pitfalls is really that validation work. So you want to make sure that you're spending that time to really make sure you're developing your biomarker rigorously to be able to support its intent as something that's reasonably likely and support the accelerated. But leveraging, I agree with Nicole, we look at everything. We're going to look at your non-clinical data. We're going to look at the PK/PD data. We're going to be looking at a lot of things, not just solely one thing. And I think that's really important because in our rare disease, whether we're talking an accelerated or traditional pathway, it is really what is the data story that's being presented to us from all of these different things. And I think that's what was highlighted in the Fireside Chat earlier was that we are not looking at a single thing. We're looking at everything and to see if things are moving in the right direction and really can we tie it back to some type of biological plausibility? Does this make sense there? And do we have that evidence and rationale to support it?
I have a question for the regulators. All these concepts are really something that we love to hear as pharmaceutical company, biotech companies. However, at the time we meet with you for a pre-IND meeting or at the time of IND submission, we are still not forced. We try to do things well as well. So we want to present a clinical trial that has hypothesis, sample size calculation, a lot of things. We make a lot of assumptions to design the clinical trial. We expect such magnitude of effect. And at the end of the day, after 1 year in the clinical trial, we may have what you call substantial evidence, but sometimes we are not exactly where we expected to be. So we present to you at the time of the IND submission a very nice package with a well-designed study, with a sample size calculation that is, yes, well documented. But 1 year later, we are a little short with this, and we feel uncomfortable, and we ask ourselves, can we file? Can we meet the FDA and for a pre-BLA meeting? And we are uncomfortable with this uncertainty.
So I want to say that I appreciate that. I really do. And I think it's a very honest answer and an honest conversation that we have to have. What I would point to is really that we have to foster communication. I don't want to oversimplify that, but communicating with us, and not just at the point where we're a year in and things don't look so great. I mean, please come and talk to us through that development program. That's really what I would actually prefer, is to hear about those things. And even when it comes to some of the CMC challenges that some of our files have and are not able to appreciate until they get started with the clinical development and potency assays and otherwise. And so I hear you. I think that through communication, we can probably get through things, quicker than we can if we wait a year or 2 years or 3 years, which sometimes happens. And I agree with you that it is not a perfect science here, especially in rare diseases where there's just not a lot of patients even to be able to study. I thought that the slide that we had, again, right before this talk, really illustrated that point where you have, antibodies in some patients, already a small population. There's just a lot of challenges. And so I do think that requires additional touch points and additional communication.
Yeah, I don't think you're gonna find, I hope not, at least not in Office of Neuroscience. We want you to come talk to us. I think any review division, because all the therapeutic review divisions in CDER, someone has a rare disease, right? And so in CBER too, right? We want you to come talk to us because we know that we can't ignore those challenges. We don't want to either. And so if we hear about them earlier, and I agree with Nicole, thank you for bringing up that question, because I know that's a scary question to ask with us here, but it's a real question that is what you're faced. And I think it's coming and talking to us early, like when you're starting to notice that, because is there something that we could do, right? Is there, all right, do we have to add another 6 months? I'm making this off the top of my head, not saying this is the advice would be given, right? But is there opportunities to still collect good scientific data? Is there things to think about? Is there what evidence may you need to be able to bring in? That we will never know those questions unless you come to us. And I do think if it is waiting till that pre-BLA, pre-NDA meeting, we are too late. We are definitely too late. So when we say early and often, we really do mean it. And this is why, because if we have the opportunity to provide advice that may allow us to gather additional information. I'll give an example. It could be something as simple as, okay, can we get some exit interviews from your population in your trial to help us explain meaningfulness, right? Wasn't thought about at the beginning of the trial. This is what you're sensing. That's adding to this accumulation of evidence that could come in to talk about that trial experience and what's really happening. If we're worried that we're seeing maybe something's a modest effect, or we're maybe going to have interpretation issues anticipated. So definitely come and talk to us, because the earlier will allow us to really think through and try to really see what's really needed. We obviously are always optimistic and trying to find those paths forwards. And so earlier the better.
Perfect. I'm going to move on to the next question, which is, what are some of the general opportunities and challenges of using a placebo for LGMD? Doug, your case study concerned to us specifically. Are there any general lessons that we can take away from the case study? And then for the group more broadly, any thoughts concerning adaptive or other design features that can be used to mitigate the ethical and other risks of keeping patients on placebos too long?
Yeah, so I think it's obviously a very complicated topic on a number of levels, and we heard this morning from a number of patients and advocates on the challenges of being in a placebo, that you are committing to participation in a study, that you may be receiving an ineffective drug while your disease continues to progress. And that is the risk that anybody in any trial for any condition obviously suffers. It has become a standard in the tradition, and there's a reason for that. There's obviously a lot of examples of clinical studies that have been conducted in open label fashions that have ultimately failed in more controlled environments. And so, it's certainly a very controversial and complicated aspect. In our clinical program, reflecting that and reflecting the needs of our program and the regulatory requirements, we are conducting a placebo-controlled trial, but it is something that is a significant burden that we are asking our partners in the patient community to hold. And one of the major questions that's been raised on several times in today's discussions is the role of other data sources and other data sets. And that is an area where, and this is, there's not going to be any one size fits all, obviously, but having the ability to be creative in how one develops placebo arms, how one develops comparator arms, how one utilizes pre-treatment experience can be critical in addressing our needs as a sponsor, our regulatory partners' needs to confirm the effectiveness and the safety of a product, while being as minimally burdensome on the patient community as possible. And that's one of those areas where I think having a dialogue, but also having greater flexibility and to be creative and engage with creative solutions is going to be really critical in the years ahead.
I'd like to make a comment on that. So to give you an idea, when patients are required to do a placebo, this is the burden that the patient experiences. This is a 3-year process. Patients are not receiving the treatment, they're receiving the placebo, but yet they're required to go through the same things that patients who are getting the treatment so that it looks blind to everyone. And imagine like a patient's getting basically a sugar whatever or some null treatment, but they're getting muscle biopsies, they're traveling to centers every 3 months in some cases with muscle biopsies, blood tests, this type of test, that type of test. Then they're traveling all the way back home. There's patients getting off of work, there's families, there's kids missing school. People are missing like pay from work, and then they're going through all these arduous things for a placebo control. When patients go through all of that, it's no wonder that patients want to drop out of something like that, because the burden gets too great. And then a serious rare disease like LGMD, it's really, to me, it just makes it so unethical when you start to think that patients are not receiving something that could make their lives so much better. And that's just how patients view it. You know, I'm just giving you a patient perspective, and, you know, but just really kind of think about giving somebody a muscle biopsy when you know that they do not have the actual medication. You know, does that, you're taking another piece of that muscle out of somebody's body where they hardly have any muscle.
You know, I want to thank you, Kat, for that perspective. And this is something that we hear often every day from our rare disease communities that have progressive and often fatal diseases, right? We hear this, we know this. And I'm not here to sell you why placebo is good. I want to talk to you about why we need to think about it, though, and how, when applied, and why it can be important. So first of all, it's the safety piece, is that I understand every one of you will tell me, I'm willing to take that risk, Michelle. I'm willing to take that risk. I don't care. I'll do it. I have this progressive disease. And I appreciate hearing that. But we, at the same time, is we do have to balance that safety piece, right? Because I don't want something to develop that's actually worse than what you have. I really don't. I don't want something that we can potentially prevent. And so we do need the safety, and the placebo arm is really partly critical for us to get that safety data, to make sure that if we are seeing things in the treatment arm, is it really related to underlying condition, or is it because of the treatment? Because we have to make sure we put out effective treatments that are safe as well. The other piece is, you know, for me in CDER, right now as we're looking at therapies, and, you know, I'm hopeful that one day we're going to have therapies that are going to have such great magnitude of treatment benefit and clinical benefits, this will not be a question anymore, right? We have these blockbuster ideas. I want that so badly. But right now, we're probably most likely in a landscape where we're looking at things that may have modest benefit. And the placebo arm is actually really critical with that, because one, the trial length would actually, if we had an open label, would probably have to be much longer to be able to see that benefit because we have no comparator. And so that placebo arm is important, where can we get something out as soon as possible? I think could there be options where we explore what people deem as adaptive trial designs to maybe limit, for lack of a better word, with air quotes, what that placebo time would look like. And those are the conversations we want you to come in early, and talk to us about what could that look like. Because obviously, if we do have something that has a potential benefit, we would like to also get it out as well. We're not sitting on therapies. We want to see success just as much as you do. But the placebo can be really important. But I respect that's your want and desires, and I understand that completely. And if I was in your position, I'd probably want the same thing too. So please know that when we're reviewing trial protocols, and my colleagues who come in and sit across the table from us are now virtually in a little square box, we have these conversations about trial length, burden. So how many assessments and biopsies are you really doing, and is that needed? We do try to minimize as much as possible patient burden. And these are everyday conversations that we have internally with our colleagues on the review teams. And then when we talk with industry, when they come and talk to us for those meetings and the advice that we give. So like I said, I'm not here to sell you on that. I know that I'm not going to change minds, but I do hear you. Please know we acknowledge that. And we do really try to be open-minded as much as possible. But we do still have to be able to collect data to be able to make, in the end, that regulatory decision.
So we'll go to Nicole, Laura, and then we'll move on to single arm / natural history.
Yeah, I just wanted, I do have a slightly different take on it. I think in CBER, where a lot of these therapies are quite directed and, specific to correction of a gene, I think we actually are open to not having placebo controlled trials and for those to be single arm. So I think that it really does depend. I do hear you, and I agree with you. So I do think that, again, come talk to us, because I do think that there is a space for that, and we have in several rare diseases now allowed the trials that are progressing to be single arm. So, yeah.
Laura and Joanne.
So mine might go into your next question, but I recently was in a meeting and someone suggested a study in which there was some placebo, but then they also brought in natural history study to supplement so that you could reduce the number of people you needed with placebo. That's not something I had ever heard about before, but I'm just throwing that out there for people who know more than I do, so that you're reducing the burden. And I really, we might get this into when you talk about the natural history, but, I really liked what James was talking about, is about quantifying the bias.
We're totally going to get on that next.
Okay, well then, because I think that would be a great thing, especially for using assessments that are across the LGMDs. And if you can assess, the bias is probably not going to be subtype dependent, so that if it's more assessment dependent. And so if we can, as a community, find ways to determine what that bias is, because, I'm not a patient, but I would think, there are some things you can't motivate yourself to move something that you can't do. So there's going to be a limit to what that is going to be. And if you can quantitate that, then that would, maybe make it more palatable to use natural history, because you wouldn't have the ascertainment. You wouldn't have the bias.
Well, Joanne, next topic.
And from a sponsor's point of view, I think it behooves us to use all the data from natural history studies. As James said, if you look at the prognostic factors, then you can improve the power of your study. Improving the power of your study decreases the number of patients and ultimately the number of placebo patients that need to be in a study. And I think the other factor that we have control over, or try to have control over, Kat, is to make things as least burdensome as possible, to eliminate biopsies when we don't need biopsies, to have enough safety data that we can decrease the number of visits. So it's every 6 months instead of every 3 months. It's a burden, but it's not as much, as well as supporting the families to come in any way we can financially to make sure that, while you pointed out very eloquently that there is a cost to doing this, to minimize or eliminate any financial cost of doing that.
Ang with AskBio. I just want to have a commentary is that it's really interesting, our conference today, because cell and gene therapy, I think, is the full realization of the Orphan Drug Act, where by regulatory science, the regulation is less than 200,000 people. There are opportunities now to really approach novel trial designs. But I want to have a comment is like, we're hearing people saying, oh, the FDA has to do this, has to do that. I mean, as a former reviewer, I would make the comment that it's also the responsibility of all the stakeholders in generating the data. And first and foremost, is that, I think Dr. Signorovich was talking about external controls, for example. The use of natural history study data should not be used as an IP mode against the development of new therapies in general. Right? But the way for that to happen is a push-pull mechanism. I'm also a former health staffer. I work on 21st century cures, and we try to do data sharing as a thing to support the development of new therapies. And the answer is that patient support groups, communities, and consortia, as well as industry, all of us have to push towards this goal of sharing data to make it fit for purpose. In order, we can talk about external controls as this sort of sine qua non, it's just going to fix everything. The answer is it won't fix everything if it's not standardized and spoken with in a very transparent way with regulatory authorities, not just the US, but with globally, in order to get the fit for purpose data that we need. And right now, it's just not happening, right? And so we're sort of stuck on this weird catch-22. And so my bid is that, we as stakeholders, not just industry, not just the FDA, right, but all of us have to support this idea that patient data is something which is, it's not an IP mode. That's just basically it, right? And how do we approach that as a society, right? How do we advance the regulatory science? Again, CGT, I think, is the full realization of the Orphan Drug Act. I think within our lifetime, the next 20 years, there's all these sort of safety packages, right? And the answer is, it's gonna get better because there are gonna be third, fourth, fifth generation capsids, better promoters, there'll be lower doses, less mutagenicity. Everyone at this table will work on that, right? And so I think the safety data package will be better characterized. We'll get better at it. But at the end of the day, if you really want an external control for efficacy, right, we're going to have, again, shared data and talk about it again in a transparent way, communicating effectively with the regulatory authorities, and having a clear set of leveling of expectations ahead of time. And that's the large vision. And I think maybe if we get some comment from Dr. Verdun and Dr. Branch on that, but I think all of us here believe in the mission of building therapies, and we have to advance regulatory science.
Sure.
I agree. I think you touched on a lot there. I think there are a lot of challenges, though, to that. What we see on our side, for example, there are several developers in a very rare disease space that sometimes is challenging. And so I 100% agree with you that we need to work together more. What I see in reality is that does not always happen. And so it's sort of like this race a bit to the finish line, which you can understand. And I also think what is a challenge is we really have to figure out ways, and I don't have the answer to this, but we have to figure out ways to make sure that what we are approving is effective, because when you also have a very small patient population with significant unmet need, that where there is something approved, that obviously that's something that's there where there was nothing before. A lot of people gravitate toward that, but we also want to make sure that we're continuing to figure out ways to progress the field and get therapies out there that are better and better as well. And so all of these, I think, are excellent things to discuss and to talk about, and challenges. I agree with you.
Yeah, and I want to add on. So I think what Nicole just says, it happens sometimes in the space, right, where there's a couple people, everyone's honing in, but they're not talking to one another, right? And so I think what opportunities and things we've been describing today is actually efforts that could we focus on things that would benefit the whole through work in pre-competitive spaces, right? That's where data sharing can come into handy. That's where a group of folks are focusing on a biomarker and getting the assay development done and things like that, then that can be put into the drug development programs that the sponsors are coming and bringing to us. And so that is a way then, that is a path. And I know there's folks in this room that are part of, and there's folks on this panel that I know are part of pre-competitive efforts. They do work, they can be successful, and it's always good to have, and it helps advance the regulatory science. And what it also can help is also balance, and it was hinted at, as Nicole said, is that drug development in rare disease is global. And so that's where we can bring in our international colleagues, our other regulators. I mean, we talk to our counterparts all the time at other regulatory agencies, but this is an opportunity to also fold them into pre-competitive space efforts that, again, with targeted questions and goals you want to go for, can help advance that regulatory science and ultimately help advance drug development. And ultimately, in the end, we'll be able to help our persons living with limb-girdle. So I really think that is an avenue to think about, is the pre-competitive space work.
Can I say one more thing?
You sponsor the conference.
Hey. Well, I appreciate especially all these comments. And since you brought all this up, I'm going to say, let me give you another perspective from patients. So patients will hear there's a natural history study, and it's sponsored by a company, and there's a potential treatment. So all the patients run to this natural history study. And then, a little bit of time goes by, and then they hear another one, natural history study, and it's the same disease, but a different company. And everybody's like running over to this one, hoping to get into a clinical trial eventually. We have patients that are in multiple natural history studies at the same time with different companies. At some point, do you think that's insane? Or is that a good waste of muscle tissue? Yes. Is it a good waste of blood? Yes. It's a good waste of patients' time? Yes. Because there are ways, and I know it's very difficult, and I'm not saying I have the answers because I don't, but at some point, we've got to think of patients or the data that a patient gives you, whether you're industry or whomever or researcher, that data that a patient is sacrificing is that patient's data. It's not your data, it's that patient's data. They own it. And you got to kind of understand that. And I think that is the trajectory that patient groups are going. I do believe that there is like a huge effort that's going to be in the future where patients are really going to be making this an important advocacy issue because of the fact that patients are having to do these multiple natural history studies and these multiple things, and it's exhausting the patient community, all because they just want a treatment so badly. And I know there's so many families in here right now that are in multiple natural history studies. And so I just want to share, think about that a little bit. And, I don't want to drop the mic, but I do think that that's a good point to make.
Can I run?
Yes.
This has been bubbling up for the past couple of minutes. And so the next question is kind of a stew of issues which we can all work on. Okay, we talked about placebo. What do we need to do to make a single arm trial robust enough? So James presented some technical suggestions, like, everyone knows there is the risk of there's open label bias challenges, and there's a lot of noise. So the first question will be to you, like, how can we use the tools in the toolkit most effectively in this space? Then, Laura, I know already that you have like thoughts, and Nicole, if you'd like to share. So James, Laura, Nicole, and then we'll see where it goes.
I think we need to think creatively about where the risks could lie. And the nice thing about randomization is takes care of that. But if we don't have that, we really have to use our imagination and I think listen to all the stakeholders and take seriously the risks of bias. But then we need to be empirical and scientific. I think a lot of those risks of bias we can test with data. And we should think of ways to do that will move things forward. I think where things get mired is if it feels like there's a lot of unbounded risks or things that it's really just based on opinion. And I think hearing that not really as opinion, but as a legitimate concern, and then thinking, how can we use data and evidence to either address that concern, or this is real science, or show that that's a fatal concern, right? I think both things are possible. The data I've seen in other muscular dystrophies, I think gives me hope that this is possible and the concerns can be addressed. I think some of them have, but some are remaining. But I think the key is to listen to the risks and be scientific about addressing them.
I'm not sure what you expected me to say, but I was thinking, the Jane Foundation we've been doing a natural history study in our population since 2012. And our official natural history study is coming to end in June of this year, which is kind of sad. But we feel like, just continuing to do the same thing over and over is not the way to go, and we've learned a lot from that, and we want to know. So one thing we're trying to move into is to try to, how do we collect this data in a more as part of care management, right? I know that the PTs are really using and are learning that a lot of the assessments in the NSAD can predict clinical progression, where people are going to go. So it's not just we're collecting natural history data. It actually can be helpful to predict the study. But when I talk to most clinicians, it's not their fault because I know they don't have a lot of time, but most only do, PT, when there's an issue with falls and those kinds of things. It's not a standard assessments they do, except for Nationwide, who does it for every patient. And so, finding a way, I know they take a lot of time, sometimes they take special equipment, but if we could identify, the best things and then use them in clinical management, but also use them to continue to collect across, the NSAD is being used across the LGMD, so even people who aren't genetically subtyped, but then you're collecting the data as part of their normal assessments, and they're not having to travel, it would be local. I'm just throwing this out here. I know the clinicians will have a lot to say about this, but, trying to do that, but also, it has to be standardized, right? So you have to you have to be trained, right? So everybody's doing it right, or doing it in the same way so the data can be compared, But even with natural history studies, you want to be sure if you're using the same assessments that they're all being doing the same way, because that's the only way you're going to be able to combine the data and have it be comparable.
Can I offer something there? So I might offer that it's a mistake to end that natural history study. And I know it's not my business, so I can make that editorial commentary. But when we talk about surrogate endpoints, when we talk about clinical endpoints, nobody lives their life caring about what their NSAT score is. Nobody lives their life caring about what their 100-meter time test is. It doesn't matter. What they care about is, can I get out of a chair? Can I transfer the toilet? Can I eat my food? Can I comb my hair? Can I bathe? And those things are linked. That's why we do these clinical endpoints. That's why we do all these endpoints, is that they ultimately inform the things that really matter. And the opportunity as we look in the longer term, so this is a huge long-term data set, is those early changes, those changes that occur 2012, what do they tell you about things that are unambiguously important endpoints that, not losing the ability to walk, losing the ability to transfer, those sort of things down the road. And that can be to me, the invaluable thing in helping our regulatory colleagues feel comfortable with the impact that we're seeing with these early therapies. And so it's a huge, the natural history studies, we look at them from the standpoint of what does it tell us about how we could mimic a clinical trial. But the other real value of these things is, what does it tell us about the longer term, and how can we use that information to inform the meaningfulness of what we're seeing in the short term?
Just to be clear, we're ending the way it currently is done, but we're still planning to collect natural history data.
There you go.
It's just not in a formal, we're going to try to collect it in the way I was discussing through clinical, yearly clinical assessment.
How about Nicole and then James?
So I would just add here, I think that natural history data is great when we have it. It's not a luxury we always have, but in terms of how much is in that natural history data and the collection of it and how much we're able to use. I would say that I think that for single arm trials, where we're using biomarkers and relying on surrogates, we're definitely open to that. We have to make sure that there's biological plausibility. That piece is important, right? It's an important part of the evaluation. So showing that there is biological plausibility. I think considerations around who we're enrolling in trials. So I think it's fine to, for example, enroll across a very large age range, and I'm talking in generalities, but sometimes it's helpful to sort of do cohorts in that regard, because there are differences there. And I hear that. I mean, I hear at meetings as well, there's this trial and it's just in children that are 1 to 8. And I made it past 20. I wasn't supposed to, but I still have significant morbidities and I want things for me too. And so can you encourage people to also not forget about us? And so I get that as well. But when you're trying to get to the finish line and show that your endpoint has an effect, sometimes we're choosing patients where you can show more of that effect in a shorter time. So I think we have to think through all of those things. What's also very important is, as we're thinking about the trial for an accelerated approval or using a surrogate, making sure that we've also done as much to think through the confirmatory trial and what that's going to look like, because that's an important piece also of the conversation, is making sure that we're able to not only show, have that surrogate and have a potential approval there, but that we're able to confirm that clinical benefit. And so we have a lot of conversation around that, but I think sometimes that piece is not as well thought out. It's really just, can we get this first approval? And I think that's important for the community as well, because we want to make sure that what we are putting out there, ultimately we can show is effective. And that it's effective when we're approving it, but that also long-term we have that data and that the data is collected in a meaningful way. So those are just some of the things I would touch on.
And confirmatory study will be our next subtopic.
Oh, great.
Can I add a comment? Is that, and thank you, Dr. Verdun. I think sort of.
I can't hear you. I don't know if you're on the mic.
It's a tech nightmare, I guess, than microphones. So Dr. Verdun, thank you very much for the comment. I think kind of the big picture, and what I love about limb girdles and this therapeutic area is how vested interests the academic consortia are and the patient support groups. I mean, for example, we've got Dr. Matthews has, in the labor of love, has run a registry, a study for 16 plus years. And I understand the burdens that the agency has in the sense like, where do you cut off the data in terms of making an interpretability and a decision? And I think, I'm just gonna put it out there and just ask, is that, well, in this framework that everyone in this room agrees that the accelerated approval pathway is something that's gonna have to happen, none of us wanna be in the front of the Washington Post, none of us, right? What does that look like to streamline pathways towards, you know, an approvable marketing submission? And is there a role for, and we've seen this in the oncology space for CAR T, like master protocols. And is there a role for master protocols? You know, maybe they're not necessarily run by sponsors like us, but maybe they're run by consortia or they're run by Centers for Excellence as a multi-stakeholder group where they provide the transparency and the level of expectation, what you think is going to be important for us to make those decisions. So should in a future state, as we get better with our safety packages, like that sort of efficacy signals are just more, they're easier to interpret. And I think that like the role of master protocols in oncology has been a game changer, right? And is there a role for that here in our space?
I think that there is definitely a role. We haven't heard a lot about master protocols, but I think it's a great idea, and I think that we should engage more to operationalize that. The natural history studies, thank you so much again for touching on that, because I wanted to add one piece. When we're talking about accelerated approval, often what we are hearing is acceleration over 6 months, you know, looking at 6 months of data or a year on a surrogate, right? And so the natural history data itself is definitely helpful and used in the entire development program, and especially when we are setting up potentially traditional approval or conversion of accelerated approval to a full approval, what the natural history of the disease is versus what potentially is we're seeing in that other trial that we're looking at longer term. So that data does work there. I think the biomarker piece, often what I see is the desire to have shorter term looks at efficacy and endpoints using some of these surrogates. And so it's harder to then, in that specific case, look at the natural history versus a 6-month look. And so it really just depends. A lot of this, I'm speaking in generalities a bit, but it really does depend on the program itself. And we're open to looking at all of those things.
And Dr. Verdun, thank you for that. And sort of just...
Your mic's not on. Okay. Oh, it's on? Okay,
there we go. Sorry. So, and thank you, Dr. Verdun. I think, you know, kind of the spirit of it, and again, everything is non-binding. Of course, we're at a conference, right? But ideally, we would live in a world where if there's medical plausibility, biologic plausibility around the assay, the expression of the transgene or product, whatnot, is there, and, you know, given the assurances of a master protocol, can show signals, right? And you're going to get 12 months, 18 months, years of data. You know that already. If you have those assurances, then I think potentially there are opportunities to advance the regulatory science and say, well, if this assay is validated and looks good, there's medical plausibility around it, right? We can live in a framework, you know, where no one's in the front of the post, right? And says, well, you know, we know that we're going to know if we made the right decision regardless. And I think that's the big thing, right? It's like, that's what we care about, is building the right therapies for the people we care about. Right?
So Michelle, I'm gonna let you answer the ideal world question.
Yeah, well, actually, in the Office of Neuroscience and CDER, we do have some familiarity with platform trials. The ALS one is probably the most well-known for us in neuroscience. And so we have a lot of familiarity with the use of a master protocol, different arms type things. I know there was discussion at one time. I'm staring at Abby because she was part of those discussions for Duchenne at one time about what would that look like for them. And we've actually had other rare diseases come and you try to engage with us in various ways about what could it look like. And there's a lot of conversation. It is a multi-complex, lots of really more brilliant people than I am in this room need to be, to think about this, because there's things to think about, is this repurposing of a drug that's already existing to see how it works in a new population? Is it where sponsors are willing to, have investigational drugs to be part of it? So there's a lot there, but we, at least in neuroscience over in CDER, have that experience because we have that trial platform that's going on, and I think we've had some lessons, and I know that the group who leads it out of MGH talks about their lessons learned from it, from an infrastructure point and what it takes to set up. So I definitely think there is some openness. Again, it has to make sense. I also want to say, in terms of the natural history, the conversations we've been having, I think there was a comment that was said earlier today about, and in the cat, what you didn't say, what I hear a lot too, is which natural history study should I do, right? Because I got to pick one. I got 4 I'm in, and whose data should I fill out first? I had that question a couple weeks ago from a friend of mine whose daughter has a rare disease, and I was like, I don't know. You have to make that's a hard decision, right? To where to invest that time. And so I think there was a point, and I want to make sure we highlight. I want to highlight it again. Is what was the original intent of that natural history study? Because not every natural history study was designed to help support a regulatory decision, right? They may have been just to do what the title said. What is the actual progression of the disease? Because we've just identified it and we don't know, right? There are people who use natural history studies maybe to help advocate for services for their group, or maybe eventually for access to things, and if we do have approval. So natural history studies have a lot of different purposes, and so I think the highlighting today, and I think Nick said it with his presentation, is that really the conversation is, what do we need really for a natural history study to be informative for drug development? And that can come in many facets. It doesn't just have to be that external control arm. It could be, what is my trial population? And so we've got some rapid progressors. Is that something we need to do a little deeper dive in? Is it, and maybe these are the outcome assessments we need to look at. But I will say, when it comes to natural history studies, particularly ones that have been going on for a long time, first of all, congratulations for maintaining one that long. That is a hard thing to do. And I always ask those who have done it, how did you maintain to get all of those study visits in annually of maintaining that? Because I do know it's very hard. But it's also being willing to adapt over time, and maybe having to drop things that you're collecting or adding new things because the science has evolved as well. And just making sure that if we are collecting very long longitudinal data, we are evolving with the science, and keeping that relevant, because that is beneficial. But there are many uses of natural history study. I know today the emphasis seems to be on that external control arm, but it could be helping as part of overall confirmatory evidence by really being able to be in that comparator of what is happening with that population versus the trial population, and can we do some propensity matching on a couple variables that there is some alignment on. So there's definitely, I think, more opportunities of leveraging natural history than just external control. And so we should keep in mind of all potentials, but I think in the end, we need to make sure that we are collecting data rigorously and in a systematic way, so we can be able to make those scientific discussions and decisions.
I was just going to add, just for a second, oh, did you want to go, Nicole?
Well, I was just going to clarify, sorry. I was just going to clarify one thing, because when we say master protocol, that can mean a lot of different things to a lot of different people, right? So, you have several different drugs that you're developing at the same time in one population, or, multiple populations, and you're looking at that same drug. So we do have a lot of examples where people have leveraged information to simultaneously develop things in multiple populations or otherwise. And I do think that is a successful model, especially leveraging in preclinical data in terms of the CMC data, pharm tox data, not having to repeat all of that. And we do have those examples as well. The platform designation per se, we also are very much in favor of that that platform approach. The designation itself is one that where you have an approved product, and then you can leverage that data to then have multiple other, you're developing something with essentially same AAV backbone or otherwise, and then can develop that in other disease areas and leverage that information. We don't have a lot of approved products in the cell and gene therapy space relative to other, to drugs and otherwise, but we're really getting there. The pace of things is quite fast. But so we also have that platform type of approach that we agree with, even in the absence of having that first approval. So I wanted to just make sure, because that's very important.
I'm just going to add, I wanted to comment on Eng and his comment a little bit too, and several others. He had mentioned Dr. Kathy Matthews and her natural history study, and it's a 16-year worth of data, and the name of her study is called Clinical Trial Readiness for LGMD 2I, started 16 years ago, the highest level of natural history study that you could participate in. And I don't know what other type of natural history study that exists in the world that would be bar higher than that one. I guess my thought would be, please tell me, everyone, what are we looking for that would be better than that caliber or the cause caliber? To me, it's from some of the best universities and the best researchers in our field. I don't know that we can develop anything better. It's elite. And so there's a lot of the elite ones, and those are just the two I'm talking about. But I just wanted to comment about that. And I will have to leave in about 5 minutes to catch my plane. But I don't want you to think that I'm leaving because I was upset over anything. But please, consider what I just shared.
And now you're dropping the mic. Okay. Quick open-ended softball question for Nicole and Michelle on confirmatory study, right? So a lot of the time today on the difficulties of doing a clinical endpoint study at all, and it doesn't get easier, and maybe it gets harder with the confirmatory study. Any general comments about how we can be creative and rigorous over the whole life cycle, including the confirmatory study for LGMD?
Yeah, I'll start. You guys will turn me off on the back. Okay, there we go. I think Nicole's first point, she says, think about it early. I think when you're thinking about your pathway of whatever you want to go, and maybe our industry colleagues will chime in, they may not always start thinking about which direction they want to go sometimes, but you want to definitely think about it early, and you need to think about what you're going to measure, right? What is that clinical outcome assessment you're going to use? And is it clinically meaningful? And that's something that I felt like maybe was a little bit missing today, was really talking about meaningful change on some of those scales you guys mentioned. And hopefully when we do the last session, Keith, I'll have some opportunities I think this community could explore in measurement wise to help enhance the data and information on the clinical side. And so I think you do need to think about it early about what are you going to measure, and is it clinically meaningful to patients, that change that you're going to see. And so that work, when we think about what would be needed under that traditional approval pathway, is what you're going to have to demonstrate. And so knowing what's important to patients, I think we heard that earlier today. How am I going to measure it? What am I going to select to measure? Am I hopefully going to leverage a couple things that are going to show me that spectrum of that lived experience from that? So I think this is where your natural history studies can be leveraged to help be informative, but it shouldn't be your sole source. You're probably going to have to go talk to some people, and you're going to have to find that out. But you definitely want to start thinking about that very early, because the implementation of it's going to be really critical to get that confirmatory study up and going and running. It's the critical piece you're going to need.
I think it's really, I will say it can be challenging to enroll in a confirmatory trial after you have an approval. And so that is an important piece of why I'm saying we have to sort of think through this together, earlier on in this review cycle. Some of the approaches are following patients that were enrolled in the accelerated approval study, and where there was a collection of a surrogate or a biomarker, and following those patients longer, and have incorporate more clinical endpoints that you need to follow longer to show effect. So that's an option. Another, it really just depends. It depends on the accelerated approval trial and the patient population, for example, like if there's a specific age range where there was an effect that's seen, and then potentially having, like, broadening that age range after after the approval, and then for the confirmatory study to show an effect on a clinical endpoint. So it's very specific to the development program, but there are several different approaches that you can use. But we really have to think through the enrollment piece and making sure that what we are proposing, we are able to get patients to participate and to enroll.
Matt had a comment, and then we'll move to the next question, please.
Actually, mine's just a very quick question, and it goes, again, of course, to the regulators. We heard a lot earlier today about discussions about accepting uncertainty in efficacy, and that was reassuring to hear. But we didn't hear a whole lot about acceptance of risk in terms of adverse outcomes. And when I talk to my patients who are wheelchair dependent or nearly wheelchair dependent, I'll often ask them, since one of the side effects of gene therapy can be liver toxicity, I say, well, if you had a 10, 20, or 30% chance of liver failure, and yet the disease or the drug was known to stop the course of disease, what would you choose? And they almost uniformly say, I'd go for it. They can replace my liver. So can you just touch on your talk about risk tolerance, since in the past we've heard sort of the 1% rule, and our patients are thinking 10, 20, 30% risk of liver failure and renal and liver transplant.
I agree with you. I hear the same, that the risk tolerance is often, directly related to how many options a patient has. And so I think that as regulators, we have to appreciate that. And if the patient community is willing to accept possibly more risk than we would have in a situation where it's a healthier population of patients, then I think we have to listen to that in order to advance the field. I think that there are ways where we can incorporate into our trials really robust and stringent ways to capture safety, and to, when I say that, the oncology space, we were discussing the oncology space earlier, has really been able to advance in a way using those types of things like stopping rules or DLT type of criteria, but thinking through those things very systematically and cautiously, and having DSMB type of committees in place to make sure that we are looking at things in real time. But I agree with you, as regulators, we also have to balance that and be open a bit more to what the patient communities want and are telling us that they are willing to take in terms of risk.
I just add on real quick. I think this is where Annie's question about patient preference was coming in. And that's because for gene therapies and cell therapies where we hear this, right? Particularly, we're still in the, can I say the infancy for gene therapies? We're moving along to adolescence, maybe. We're getting bigger. There's a lot of unknowns and uncertainties with the risks, right? And so patient preference studies are quantifiable ways to measure someone's risk tolerance in a way. And I'm being very crude, and my patient preference experts will even more coming to my knees for misquoting things. But one of the things that they can do, because it's based on what is your question, has been used to look at benefit risk and really in a quantifiable way, that 10 to 30, 20, 30% risk, are they willing to take if you know that in 5 to 10 years you're going to have liver failure or whatever? And so that is a quantifiable way to do it. What Dr. Stein talked about is also just talking to patients. And that is something is the qualitative work. And I think our patient-focused drug development guidance series is really important with this, because I hear all the time, I'm willing to take the risk. I'm willing to take whatever safety risk. But I don't often hear the specifics of what that looks like, right? Well, I mean, we do eventually, right? We're willing to take it. But I often hear the glaring, I'm willing to take this risk. But if it is, what if in 2 years, potential of death, that may be different, right? I'm making these up. I don't know what they could be. I'm hoping it's not that, okay, right? But those are those questions. And I think, one, that's making sure we understand if hopefully our mechanism of action of the drugs and our treatments and how it ties back to that biological plausibility. The other piece is the safety databases that you've developed, the sponsors have developed over time. So we're getting that accumulating data over time. And then really, we do have a lot of these internal conversations when applications come in about, is this something that can be mitigated as well? So we do have ways, for some risks that we can mitigate, and that's through our REMS process, right? And what does that look like? Is it, we want more safety data for another 5 years to be collected to help us understand this more. And so I definitely think that we are also trying to apply where we can, but we definitely want to make sure that we understand, we hear, I know you're willing to take the risk. I think what can be helpful sometimes to us is, what does that actually look like? And sometimes that's where I mean, sometimes we don't always get the specifics sometimes in those marketing applications, but we know when I come to PFDD and come to all these meetings, I hear that. And sometimes that can be helpful and useful when we're struggling with a decision if there is known risks that have been identified during the course of the trial.
So, do you have an additional comment? We have 10 minutes, and I think we should aspire to bang out 2 more topics. The next topic is totality of the evidence. So I'm going to read a question and then throw it open. In a disease and it stems on some of Dr. Marks's remarks from the Roundtable. In a disease this complex with multiple novel endpoints, it can be challenging to prioritize. What types of evidence can help assess whether the therapy has had a meaningful impact on the disease trajectory? For example, if it takes a long time to show clinical benefit and some of the outcome measures used are relatively coarse, can patient and expert clinician observations be leveraged to reduce residual uncertainty sooner? Who wants to take that?
I got this one. This is my bread and butter, Keith. And this is what I was struggling this entire meeting. I'm like, measurement opportunities we have. Yes. So I'm sorry, I'm very excited. This is literally my bread and butter of what I deal with every day is measurement, and how do we incorporate the patient voice and the patient experience into our endpoint hierarchy. So number one, it's an endpoint hierarchy. Let's look at everything. And I think that was adequately said in the Fireside Chat. We're going to look at everything you're measuring, right? Primary, secondary, we're going to look at it all. Collectively, it's telling us the story of that trial experience, right? And the more diverse, I mean, let me caution when I use the word diverse, because I don't want to go like, let's have so many different endpoints. It's not, we've lost the story now. But we're collecting data that makes sense, right? It makes sense to the disease, it makes sense to patients, it's important to patients. And so I am hopeful that we are seeing different type of endpoints. I'm hoping we're not just seeing all performance outcome measures. I heard a lot of them measured today. My first point is, where's my patient-reported outcome? Because I know I can get reliable, good self-report in this group. And I'm happy, because I don't always get that in neurology, right? So anytime I can get a PRO, I'm very happy, because that's honestly the only way I'm really going to know if something works for you, and your experience is directly from you. So I encourage the exploration of all of the clinical outcome assessment opportunities and ways to measure. And I also encourage looking at the use of digital health technologies. They're another piece that we know, because there are some things that are important to patients that aren't, a typical COA can't do. We know that the DHT may provide actually something better. Another thing I've heard today, and I think it's critically important, and I feel like in this room it makes sense, but sadly, sometimes people forget that, please, if you're focusing on a population that is no longer ambulatory, do not give me the 6-minute walk, right? That is not something should be done. But folks, sometimes this is understanding our population. So making sure that if you're looking at something that's going to have both ambulatory and non-ambulatory patients, we're able to collect that information, collect something that's important, that's going to make sense to the entire population. So yes, Keith, I'm very much open to exploring all endpoints. I'm really, it's really important, because in the end of the day, the data is going to come in to the respective review divisions, and we're going to look at it, and we need to see the story that's being told from that experience. And oftentimes it's when we can get these multiple perspectives that are informative to us to really see what is going on and what is really moving, and really they build upon that story. So that's why I get enthusiastic about it, because it is, like I said, it is my bread and butter of my everyday, and it is very important to us.
I just want to say I wholeheartedly agree, and I would like to add to that, as developers, we think in the same way, because it's really up to us to look at our own data and say, do we believe? And so the preclinical data that I showed, we have even more than that. It's very easy to collect a lot of preclinical data in a inbred null mouse, but just think about, clinically, you have expression, you have your clinical outcomes, but then you have a lot of other very informative, unbiased measures. When you start to see improvements in histology, you see improvements in CK, you see MRI changes, those things are very difficult to influence by bias. So collectively, it's really hard to ignore when you're seeing all these things triangulate into seeing an effect. And so I really believe in the totality of evidence, in terms of looking at these therapies. It's not just one thing, it's the whole package.
Kind of a closing big picture question that I talked about with Doug in prep. I think, Doug, the way you phrased it was the whole, primary objective of today's scientific workshop was to try to strike the right balance, right? Because you need rigor and persuasiveness, but we have to be able to do it. It has to be feasible. So stepping back, what's the big picture in terms of the next wave of drug development?
Yeah, so this is a soapbox question, I guess, as well, and it's one I really welcome comments from other folks on this panel, particularly my colleagues to my left. But the importance, as I see it, of the efforts that are going on right now, the work that all the sponsors at this table and elsewhere are trying to move their programs forward, is one aspect. Is there ways that we can accelerate or streamline the pathway to getting our programs, our products through to patients in an appropriate way? There's also the next wave through. And this is the big question, I think, from the community's perspective, which is, can we establish a framework that obviously every program needs to be its own bespoke development, but a framework that developers in early stage programs can look at and plan the relevant approaches they need to do to meet that bar. Can we come up with a way that can allow novel programs for ultra rare conditions to have a pathway forward to be able to gain the investment and gain the interest from the people who, when you put up the bucks to make this work, to move these programs forward? And that's the real question of the day. It's not so much what's already there, it's what's coming behind. And that's, for everyone in this room, that's the hope, is that the programs that are looming on the horizon and right here, right now, do amazing things, but the stuff down the pike does things that are even better.
In the same way, really, I'm sorry.
I would just say that the optimal word here is flexibility, that as regulators, we have to be flexible. I do think that we have the pathways currently that can get us to the finish line, but we have to be able to, in a flexible way, based on sort of what the disease area is, what the challenges are, be able to design something that works, and specifically a package that works, right? And so we've talked a lot. I know I have talked a lot today about just what that overall package looks like, but it really is a combination of things. And in addition to the biologic plausibility piece, but having multiple endpoints and different types of endpoints within a specific package, I think is helpful. And thinking through sort of the overall development a bit more, which is also something I talked about. But I think that we have the tools in place. From a regulatory standpoint, we have the desire to move the field forward and to get things to the finish line. And so I think that through partnership, we can get there. Those would be my closing remarks.
Thank you. I think that captures it really well. One of the things that I heard this morning reflects on the idea that what we want to do is change the trajectory of the disease fundamentally. And what I heard from some of the quotes from patients was, yes, stabilizing the disease would be awesome, but even reducing the deterioration by, I'm seeing nodding, by 50%. And it's not about whether it's 2 points or 1 point or 3 points. It's about being able to decrease the rate of deterioration and potentially changing a disease that progresses over 25 years to one that progresses over 50 years or 75 years. And that will ultimately make a difference in people's lives.
Please join me in thanking our brilliant and exactly on time panel. We now move to a little fireside chat. It's Dr. Marks. Dr. Stein had to leave, so Michelle is sitting in. Kat had to leave, so Kelly is sitting in. And that's the fireside chat.
Okay, well, thank you again for being here today. And, it's been a great day, an emotional day for a lot of us. And I am really grateful that yes, hi. I didn't know you were coming here too. Awesome. So we're just jumping in here. Based on all the testimonies and scientific updates that we've heard throughout the day today, what do you think that we as an LGMD community, a patient community, need to concentrate on for success in the treatment development? And I really appreciate, what you said, Michelle, that you took notes today, that you really heard what we said, and you're going to take that back to the office. And I think that is, probably the biggest outcome from our patient community is that we were heard. We felt that, it was really hard for so many patients to get up here today and caregivers to really pour their hearts out and to know that we shared the voice for the huge LGMD community today. And so thank you for making note of that. But again, back to my question is, what advice do you have for us as a community? What can we do to help work with, the experts in this room today to really make the difference?
So again, thank you for inviting us today. I know that my colleagues here, and I know there's some watching online, really do appreciate. We do learn from these meetings, and I do know it's a conversation I have a lot of patient groups. The number one question is, well, we talk to you, but what do you do with that information? And oftentimes that's very hard to see because we may not have a program. There may not be a pipeline, or we can't talk about what we have. So do know that we take back what we hear, and I did take notes today, and when we get something in, we'll reflect on them, and we do really learn. We do read the voice of the patient reports that are done, and we've read yours. And so no, it's there, but I think today was a really great conversation as you continue to advance the science and really what is happening in the therapeutic development world. I can't say enough, you are fortunate where you have opportunities in both drug-based, ASO-based, and gene therapies. I mean, that is an amazing opportunity that you have here. So many other groups wish they were that that at that point. And I think that's really exciting, and I think you should embrace it. And I think what our conversations today have been on is what do we have right now, and how do we leverage it via the natural history. And I think there's been some great conversations, and I think opportunities to leverage your natural history is going to be really important, how it can help inform drug development. So while it may not, might not have been developed per se for a regulatory intent, it could help inform perhaps, for example, your selection of your trial population that you may want to enroll. It may help us not exactly pick the right clinical outcome assessment for either something on the drug side or maybe for a confirmatory trial, but it may help us be able to say, well, that concept seems to be really important and we see that it's changing over time. How can I then pick something to measure that is going to be fit for purpose? I did mention that I think there are some measurement opportunities to explore all of the clinical outcome assessments that can be beneficial, including use of digital health technologies and patient-reported outcomes. And so I do think that there's some great efforts that can be continued on with this, and so encouraging that. And I think, in the terms of the biomarker work, I think there was some great conversations this morning on the clinical presentations and what are some ideas in this afternoon about biomarkers and what is needed to develop those assays and the analytic validity needed to really build that science and evidence. So when it does come into us, we're able to review it in that rigorous way to be able to make a decision from that and making sure we're thinking about that early.
Go ahead. Did you want to
No,
elaborate on that?
I agree. I mean, I think there's not a lot to add, except I think in gene therapy we will. I think Nicole said it before. I think we'll try to be as flexible as we can to use the totality of everything we have, which in some cases may be a combination of the biomarkers to get across an initial accelerated approval and these various outcomes assessments to convert over towards a traditional or full approval. And I think that's going to be the way it's going to probably have to work of necessity, because I think for some of those outcomes assessment, it's going to take, the 2-3 years to get there to feel confident. It's a little bit like you do a clinical outcome assessment at time points that are too close together, and you add a degree of uncertainty that it's actually almost negating what you're doing. So I think I think biomarkers in this case may be able to give us the bridge to actually get the data we need to feel comfortable ultimately. I mean, yes, it will meet an approval standard. Don't get me wrong, it will meet the approval standard of substantial evidence of effectiveness. It's just that the confirmatory evidence then can come in later on.
And Brad Williams, who couldn't be here today, did help us come up with some of the questions that he was, I wanted to acknowledge him because one of his questions that he offered was, what about developing outcome measures more reflective of quality of life and patient preferences that are clinically meaningful to the patient community? And what would you like to see to be developed? And do you have any thoughts or advice on actually implementing those?
So incorporating the patient voice and the lived experience into drug development is, as said earlier, the heart of what we do every day. And I was fortunate to join the agency at the infancy of patient-focused drug development and have seen it grow during my time at the agency. And so what we do have right now, and to see the growth is amazing. We do have actual patient-focused drug development guidance series that is actually for all of our medical product centers. So while you have 2 of the 3 medical product centers here, it also does is thinking from our Center of Devices as well of how to actually collect that information. So this guidance series is really meant to, how do we first just ask questions, making sure we're asking questions, what's important to patients, what's important to you. And we know in the rare disease community that that's going to vary. It's going to differ. And as the guidance series progresses, it talks about how do we systematically collect that data in a qualitative way. And then how do we turn that into measuring what's important to patients, measuring such as really into these clinical outcome assessments. And ultimately, how do we translate that to support the clinical trial endpoint, right? That's ultimately where we want to go, is we want these assessments to become fit for purpose and to be used to support those trial endpoints. And so being able to use patient-reported outcomes is one way. We also have other outcome assessments. Opportunities to collect that lived experience is important. I think a lot of times in the rare disease community, they think they have to start from scratch. They think that they have to go back and just develop, a brand new rating scale or a brand new ADL scale or a quality of life scale or something. We do not always have to do that. I think it's important, and I don't want to get into specifics, but there's in the third guidance, which is really how do you build the outcome assessments, it really talks about going back and looking to see what already exists. And there may be some gaps. There may be things that may not be fit for purpose, and we need some additional evidence. But it may be sufficient to start doing some work. I also think that in the outcome assessments, this is where we can apply some flexibility as well in the rare disease space. So, and as a measurement person saying this, my measurement friends would typically cringe with me. But I know I don't have the patient population size to go do a beautiful psychometric study. I know it's not possible. But I do know what are the critical elements of evidence I'm going to need to make sure that this is a reliable instrument and that I can trust the interpretation and the data that comes in. And so I may be asking for that as advice. The other thing is patient preference. And so patient preference can be a whole spectrum of things. And CBER actually has some great patient preference experts over in their center, as well as our Center for Devices. And patient preference really is another way to quantify, patients' thinkings on risks, side effects, a whole range of things. It does need to be done systematically and in a rigorous way. And there is a guidance out between CDERH and CBER related to patient preference. So it's definitely the incorporation of the patient voice is something that all of the medical product centers really do promote and do try to put out good advice through our guidances.
And the last question I have is actually on behalf of Kat, who had to take off, but she said, it's very discouraging to watch our disease progress over multiple years, participate in every natural history study, and see drug development happen too slowly. 15 years ago, she was able to walk up the 4-stair climb in 7 seconds, and now she can't walk. What can we do as patients to expedite treatments? And what are some key takeaways that you have, overall that you want to share with us today?
I think patients are critical because they participate in the clinical research that's necessary to do this. I think their families help participate in the advocacy that keeps the pressure on that we use all the tools available. And I think in particular, I think right now we're in the gene therapy realm, at least the heat is on to try to keep all of the levers moving, including what we need to do in manufacturing of these products, how to get them through the development process through, hopefully towards accelerated approval, and then how to get the confirmatory evidence. So I think the advocacy in trying to keep things moving through the process is really important. I think lowering the standard for products is, to me, not, we hear a lot of times, well, can't we have conditional approval like Europe or this or that? I think lowering the bar here is doing a disservice, especially for 2 reasons. First of all, with gene therapies, our current generations of gene therapies, especially with adeno-associated viral vector gene therapies, once you get them, you make antibodies, and that precludes you from getting other AV vector. Now, there are people who are working very diligently on trying to get over that problem, but for right now, that's problem one. But you just hit it on the head, which is that if these aren't, maybe in countries where you have nationalized healthcare systems, maybe they'll get paid for. But if we don't have a reasonable degree of evidence, they won't get paid for. So I think we just need to really double down on trying to get the throughput here. So I think patients, participating in research and their families, advocates, trying to just keep pushing that we move things through the pipeline.
The only thing I would add is, while we do not have a say in it, what I am hearing in the rare disease space is clinical trial infrastructure. And so being able to have the clinical sites for folks to even be able to go when needed. I know we're trying to move things to a more decentralized world for trial visits, but infrastructure and study sites is problematic. And so we can't have successful trials. We can't get to be able to work on advancing these therapies without that infrastructure. And so I think that's an important thing to think about, and also just the global nature of our clinical trials and what those hurdles need to be to how to put on that global trial is going to be also critical to think about.
Am I not supposed to be up here?
You're supposed to be everywhere, Keith.
So
It's good he's up here.
Great.
Hey, first of all, thank you very, very much. You work all the time. I don't know if you've had a good night of sleep in years. Everyone's very grateful for your time and engagement and leadership. Thank you very much.
Thank you so much.
I have the discussion today has all been about like how are we going to punch through a treatment, right? I would just invite you to close on an optimistic, visionary note. And it's not just about AAV, but you at least have spoken often about how we can't do this one at a time. We need ultimately to take a platform approach. It's not necessarily going to happen tomorrow, and AAV may be the horse in the lead, but maybe ASOs or something else follows. What is the long-term vision of hope here?
Yeah, well, I mean, I think we're on the cusp of making gene therapies. It may not happen next year, but I think we're at a place where we're going to gene therapy is going to transform from a one at a time thing to something that is going to be probably a very versatile platform as we move more and more towards use of these very sophisticated CRISPR-Cas9 molecules that are capable of correcting defects in the genome, adding pieces to the genome as necessary, and being delivered in a manner that is much more efficient than we currently can. Plus, being able to, and then probably most importantly, being able to be manufactured at a cost and a quantity that we simply can't do currently with our current generations of vectors, which require cell lines to produce them and then purifications, et cetera. These are much more, it's much more just synthetic biology, which should cost a lot less, have a lot more high throughput, and give us the versatility to really use the platform approach. But that's probably a few years off. Nonetheless, there are quite a number of academic and industrial sponsors really trying to make that a reality more rapidly. So I think it's a good time to be peering over here to see where these therapies could take us.
So I am hopeful for the community. I am hopeful that what we've seen in SMA and Duchenne is just the tip of the iceberg, and that I think we do need to have both types of development happening on the drug and the gene therapy space, because there will be folks, until we can figure out the AAV vector problem, if we have trials that, they can no longer be in the next gene therapy that may be approved, we're going to need the drug side perhaps, and we're going to have to go through that pathway. So I think we want to continue to look at dual development in both aspects. And I really do, I joke about it. I say one day I don't want my job. I want to be able not to have the job, because we have found cures and treatments for all of our patients, for what we do our job would be great. But I am hopeful for you. I think we've seen great strides in 2 disease areas. We have lessons learned, we're applying them. And I'm hopeful for you. And I think if we continue in this path of looking under both opportunities from both centers and what we regulate, will only benefit the community in the long run better.
That sounds great. Thank you so much. I really appreciate your time today, being here for us.
Am I on? On behalf of the Speak Foundation, we just want to thank everyone for being here today, from the patients to the researchers and all the stakeholders involved. This has been a really great day with some really important learnings, I think, for all of us. We will be sharing the full recording of the video on our YouTube page, so please just be checking the Speak Foundation's website for that. We are so grateful for everyone here today for working together to accelerate safe and effective therapeutic developments for LGMDs. We just want to thank our esteemed members of the FDA who spent their entire day with us today, either in person or virtually. Hearing from them today really shows that they're dedicated and committed to working towards treatments for our LGMD patient community, and we're so grateful and encouraged by that. So thank you. Thank you all so much for being here.